Today at the third aquaculture session of the HydroMediT conference 2024. While I am Melena, I'm here with my colleague Yannis, and we're from the University of Thessaly. And now we have 5 presentations until 11:30, all in the field of the aquaculture. Each presenter has 10 minutes for the presentation, followed by 5 minutes for questions. All the presenters are kindly requested to adhere to the allocation time, please. So I think that now we can start with the first presentation. The first presenter is Mrs. Papadopoulou. Please, Mrs. Papadopoulou, you can come for the presentation entitled Reproduction, Embryonic and Larval Development of Radial Cichlid in Aquariums. So the floor is yours.
Good morning. My name is Anastasia Papadopoulou, and I am an undergraduate student in University of Patras at Thessaloniki, specifically in the Department of Aquaculture and Fisheries. I would like to thank the organizers of the conference for giving me this incredible opportunity to present my undergraduate study. The subject of my thesis was reproduction, embryonic, and larval development of red jewel cichlid, Hemichromis lyphalili, in aquariums. So without further ado, let's dive into the presentation. The aim of the research was to study the reproductive process and the early development stages of Hemichromis lyphalili. Species of the genus Hemichromis play an essential role in the global aquarium market, especially for ornamental purposes, due to their bright red color during the breeding period, ease of maintenance in aquaria, and rapid survival and reproduction in captivity. Some more information about the species is that H. lyphalili belongs to the order Cichliformes, within the family Cichlidae. This species is found in Africa, more specifically in Lake Uzundwi in Ghana. The H. lyphalili is also known for their territorial and aggressive behavior, especially during the breeding season, which coincides with heavy rains and typically occurs in the months when temperatures rise. During the breeding period, the pair carefully selects a spawning site with the substrate characterized by a porous surface for depositing their eggs. As for their diet in nature, these fish feed mainly on insect larvae, microalgae, and mycelia. Our experiment was conducted with 4 individuals, 2 males and 2 females, that were split into pairs and placed in the aquariums to avoid any casualties before the mating bond is established. Stable culture conditions were maintained by 5 centimeter thick filter bed in both reproduction aquariums. The temperature in the breeding tanks was set to 28 degrees Celsius within the suggested range. The 4 individuals that we previously mentioned had an average weight of 6.45 grams and an average length of 7.0 centimeters. The total volume of the aquariums was 40 liters. The feeding protocol that we used is the following. The spawning fish were fed a diet equivalent to 7% of their live body weight 3 times a day at 4-hour intervals, 9 a.m., 1 p.m., and 5 p.m., with a different diet given each time. The first ration consisted of commercially available comfrey from the company Tetra Discus, with a protein content of 47.5%. We can see this fish food in the first circle pointing to the word granules. The second ration consisted of flakes from the company Oceanography with a protein content of 55.3%, which is the circle pointing at flakes, and the third of frozen mysis food from the company Oceanography with a protein content of 64%, and it's the last circle. The management of the experimental aquaria included a number of measures to ensure their proper functioning. First and foremost, monitoring and controlling the water supply, as well as providing an adequate air supply. Equally important is performing regular water changes, 5 to 10%, cleaning the air stones, and cleaning the surfaces with scrapers to remove any algae buildup. Every week, measurements were carried out for physicochemical parameters such as total ammonia, nitrate and nitrite ions, and pH value and temperature. The physicochemical parameters were measured with API test kits, as you can see in the first picture, and pH and temperature was measured with the pH meter seen in the last picture. The developmental time RTI, the developmental time RTI for each development event was calculated using this formula. These times were expressed both as absolute values, hours after fertilization, and as relative values according to the formula reported by Klimogianni 2011. The beginning of the stage was defined as the time at which 50% of the eggs had hatched, while the end of the stage was determined as the time at which the yolk sac reserves were completely absorbed. Statistical analysis of the water quality data was performed using an independent t-test with a significance level of 5% to compare the mean differences in treatment. The homogeneity and normality of the means were tested using the Levin test and Shapiro-Wilk test. In this diagram, we can see the hatchability and survival rate of the eggs in each pair. The study's findings showed the impact on hatching rates in the presence or absence of the pair, and the hatching rate was significantly higher when the pair of Ichthyology J1 was not removed from the breeding aquarium, exhibiting increased parental care to the fertilized eggs in compare with the pair J2, which was removed from the breeding aquarium during the incubation period, demonstrating lower hatching rates. This diagram illustrates the RTI time of each development event that takes place during the embryonic and yolk sac larval stage of the cichlid red jewel Ichthyology. The ontogeny study of Ichthyology from fertilization to full consumption of vitelline reserves seems a typical model of the Cichlidae species. On this slide, we are comparing the growth of fry J1 and fry J2 during the same developmental hours. We observe that the offspring of J1 is larger in size compared to J2 on the same days. This difference can be attributed to the fact that when the parents of offspring J1 are inside the aquarium and personally nurture their fry, they teach them how to feed, they protect them and make them feel secure, resulting in less stress and therefore better development. Observation on the early developmental stages of other substrate breeding cichlids are consistent with the pattern described in the present study. thus confirming the basic informity of cichlid development. Within the first 5 days from hatching, the larvae of H. liphalili adhere to the substrate with the sticky secretions from the cephalic adhesive glands and lie in hiding places that the parents guard. In this way, the adhesive glands in the natural environment help to prevent dispersal by currents and protect the parental larvae. During this stage, the yolk sac serves as an additional food source of nutrition as the larvae develop their external food abilities. In conclusion, it was observed that the hatching rate was significantly higher when the pair of J1 was not removed from the breeding aquarium, exhibiting increased parental care to the fertilized eggs in compare with pair J2, which was removed from the aquarium during the incubation period, demonstrating lower hatching rates. The blood red jewel cichlid H. liphalili has been successfully reared in captivity in aquaria, which promises high production yields, makes it even more attractive for ornamental fish farming. We included a little joke. Why was the fish that great at parenting? Because he never floundered when it came to raising his little minnows. This is the bibliography that helped me with my study. Thank you for your attention.
Thank you very much, Ms. Papadopoulou. Very interesting presentation. Is there any question from the audience? Nikos, do you want to ask something? I was a little bit confused about what's the difference between J1 and J2, but I just noticed that it was that the fact that the offspring was removed from the tank, right?
The parents was removed
from the tank, right? So that means that the parental care is too important for this development.
Yes,
is
of course.
That your study highlights, right?
Yes.
Is this a characteristic for the species, for the family of the species?
I think it's a characteristic for the species.
Right.
And yeah, for the species.
Okay, thank you very much.
I can't tell for the whole family.
Thank you very much. Thank you. Right. Thank you very much. So let's move to the next presentation, which is about co-culture cultivating sea lettuce with sea bream in aquaponics. Will be presented by Ioannidis, Vlachos, Lukovitis, and Mente.
Okay. Good morning to everyone. My name is John Ioannidis, and today I will present you a part of my postgraduate thesis with the title of the co-culture cultivating sea lettuce, Ova compressa, with sea bream Sparus aurata in an aquaponic system. The aim of the study was to investigate the growth performance and survival rate of macroalgae co-cultivated with the juvenile gifted sea bream in aquaponic system. More specifically, they estimate the culture time that macroalgae show the best growth performance and nutrient uptake of sea lettuce. We have 15 days cultivation, 30 days cultivation, and 45 days cultivation as a period time. So what is an aquaponic system? We can refer to an innovative, environmentally friendly, and alternative cultivation method for producing food in a closed-loop system. And it combines aquaculture of different organisms like fishes, crustaceans, mollusks, the plant cultivation, vegetables, pharmaceutical plants, and the symbiotic environment with nitrifying bacteria. So the plant absorbs nutrients from the aquaculture waste. Regards to macroalgae, seaweeds can be used for the nutrient reduction. They can improve the biofilter capacity, increase oxygen level, reduce carbon dioxide, and increase pH. The genus of Vulva is distributed worldwide and has a wide variety of species. The study of certain macroalgae has been applied at various scientific levels, such as nutrition, biochemical synthesis, cultivation, and environment. The experiment was carried out at the Laboratory of Aquaculture Aquarium Sector, Department of Fisheries and Aquaculture at the University of Patras. So, in total, were used 108 juvenile sea bream individuals and 270 grams of ulva. We divide them into 3 treatments. The first one was chosen as the ideal cultivation period in which sea lettuce reached the best growth performance. The second treatment was chosen as a medium cultivation period, and the third treatment was chosen as a high cultivation period. You can see the system in this photo. Oops, sorry. For the experiment needs, we designed and constructed three different recirculation aquaponic systems. We filled them with brackish water and let them cycling for over 2 months. Each system consists of 3 hydroponic plastic tanks, 3 fish tanks, and some filter. Also, you use an artificial lighting that was placed at a distance of 10 centimeters from the hydroponic tanks, with a photoperiod time 12 hours light, 12 hours dark. Temperature was kept at 20 degrees Celsius in all treatments. The fish were reweighed every 2 weeks, whereas the macroalgae individuals were reweighed every 7 days. The fish were fed with a method of ad libitum 2 times per day by hand with a commercial extruded pellet diet. Regard to the water quality parameters, we measured total ammonia, nitrogen, and carbon dioxide with chromatography test every 5 days, pH, temperature, and salinity every day, and the oxygen levels were adjusted up to 70 to 85% for the whole experimental period. At the end of the experiment, performance indicators were measured according to the questions: SGR, weight gain, condition factor, and survival rate. The data were checked for normality, homogeneity, and comparison of the means. Statistical analysis were carried out using the software package of SPSS statistics. For the results, this is the first table of the results, and more specific about the water quality parameters. We can see that the nutrient utilized efficiency, the nutrient that uptake, the macroalgae ulva is statistically significantly higher in the first system, 50 days cultivation. Moreover, the pH was statistically significantly lower at the third system at 45 days cultivation. The other parameters has no statistically significant differences between all treatments. Now we can continue to the second table of the results. Growth performance indicators for juvenile species and macroalgae. In the beginning of the experiment, we have no statistically significant differences between all the treatments, but at the end of the experiment, the final weight of the feces was higher in the third system, and also the final macroalgae biomass has significant statistically differences in all treatments, with a higher number of the first system. This is the first study that provides information for the co-cultivation of ulva sea bream in a brackish aquaponic system. Previous studies has been conducted on the co-culture of ulva in recirculation aquaculture systems with the sea bass or king prawn co-culture with green seaweed in a closed circulation system. pH value in the present study was significantly higher in a treatment A and B than in treatment C. The findings of the present study show significantly better growth in biomass for ulva cultured in the aquaponic system for 15 days in comparison with ulva cultivation for 30 and 45 days, respectively. The results of the present study agree with the previous studies on ulva. In conclusion, ulva presents a high ability to uptake dissolved nitrogen, especially ammonia, improving water quality. Ulva could also be a promising candidate for use as a nutrient medium in aquaponic system, and the results of the study indicate that Ulva compressa better utilizes the nutrients of fish waste grown in the aquaponic system for 15 days. Thank you for your attention.
Thank you, Mr. Ioannidis. Is there any question from the audience, please? Come on, don't be shy. Make a question. Yeah.
Do you think that the aquaponic systems has the potential to be commercially applied?
I think that the most important problem that stops this opportunity is the economical. So it needs a lot of power, electrical power, and the cost of materials is against of this opportunity, but with a lot of solutions like solar panels of cheap materials, we can achieve to this result.
Okay, so it's a field for extra research, of course.
Yes. In the other countries, like America or Israel, they have started business with these methods, hydroponics, aeroponics, and also aquaponic systems. With common fishes like tilapia, salmon truta. It's a common technique in the yes, for a country.
Thank you.
Right.
You're welcome.
Panos, Dr.
Question for you.
Okay. In Europe, aquaponic has big farms. It's already be used. In Greece, the main problem is that when you want to make an aquaponic unit, you have to apply for an aquaculture license. And the time for this is very big.
Okay.
So this is the main problem. Energy, okay, it's a problem, but you can use solar panels, winds, panels, everything. But the main problem is the license.
Since we have plenty of time, I can see, I can make some questions as well. I was wondering, is this fresh water or saline water?
It was in brackish water.
Brackish water.
20 per 1,000 salinity.
Right. Ulva is a plant, is a macroalgae that grows also in fresh water and brackish water,
right? You can adjust to a wide variety of salinity. There are some species that can grow in close to water condition, freshwater conditions, and some others to hypersalinity.
So I was wondering also if this is a problem for the further development of aquaponics in Greece, like usually aquaponics is in freshwater, right? So you have freshwater fish species, but here in Mediterranean, we are more keen in seawater fish species. So your study is very interesting in the point that it has used sea bream, which is a marine fish species, to an aquaponic system. So that will be this adapts the needs of Mediterranean aquaculture with aquaponics, right?
In previous studies, I have studied the sea bream in a lower salinities, 5 ppt,
right,
close to fresh water, and we can adjust other plants that tolerate to salinities, like Salicornia, Portulaca,
right,
species that can tolerate the salinities. And also, with the fresh water, we can adjust the sea bass. Is one of the species that can live and grow in total fresh water with zero salinity. But it needs a lot of time to
yes, fair enough.
adjust to these parameters.
And also, if I can have a last question, what's the rationale behind of having higher growth in 15 days and this the ulva growth is reducing after that?
Generally, algae, microalgae and macroalgae have a lower period cultivation. For example, microalgae has 7 days cultivation, 10 days around there. So we have lower cultivation time, and we can produce more biomass in less time.
What's the rationale behind it? Is the growth rate of this macroalgae it reaches the maximum in 15 days? Is that the meaning?
Yes.
And after that,
you can you should renew the cultivation with the fresh one, macroalgae.
Right. Thank you very much.
Very much.
So we can move on to the next presentation, entitled "The Effect of Macroalgae Isolated from Greek Transient Ecosystems in the Rearing and Lipid Profile of a Greek Rotifer Strain," presented by Mr. Koukouvini. Hello. So you can start with the presentation, please.
Hello everyone, I'm going to present you the effect of microalgae isolated from Greek transient ecosystems in the rearing and lipid profile of a Greek rotifer strain. Many microalgae have physiological and biochemical properties that make them ideal for commercial cultivation and used as indicators for environmental monitoring. Non-toxic species of marine microalgae are the main source of food for at least some stages of the biological circle of marine life, as well as for most species in aquaculture. In aquaculture sector, rotifers, brachionus, and microalgae are very important as they have a critical role in early life stages of many aquatic species. These microorganisms can provide essential nutrients that contribute to the development of juveniles through the adulthood. Rotifers brachionus, characterized by their small size, rapid reproduction, nutrient-rich composition, stands out as optimal live feed for shrimp and fish larvae, offering essential fatty acids critical for juvenile aquatic organisms' growth and development. On the other hand, microalgae, especially those with favorable lipid profiles, are significantly contribute to the nutritional quality of the aquatic food web, serving as primary diet for rotifers and then higher trophic levels. Recognizing the pivotal role of lipid composition in promoting optimal growth, survival, and overall health, the integration of brachionus rotifers and lipid-rich microalgae emerges as imperative for the success of sustainable aquatic practices. The aim of this study is to evaluate the potential 5 microalgal strains isolated from Greek transient ecosystems as food source for a Greek rotifer strain for the species Brachionus plicatellis. Also, the lipid profiles of both microalgal strains and the fed rotifers were evaluated. The 5 strains were examined, isolated, and deposited in the Athual Bank belonging to the Department of Ecology and Taxonomy of the Department of Biology of the National and Kapodistrian University of Athens. These strains originate from transitional ecosystems with variable salinities as well as salt marshes, mainly in northern and northeastern Greece. The selection of these ecosystems was based to the diversity and adaptability of the microalgae that have adapted to the different salinity conditions that may have affect their nutritional value as food for rotifers. A widely used strain in aquacultures is Dunaliella tertiolecta, and for that reason, and because of the immediate supply by the Athwal Bank, was used as reference microalgal strain in this study. The other strains were 2 Tetraselmis verrucosa, 2 Dunaliella salina, and 1 strain of Ochrosphaera. These strains were cultured in 5-liter flasks under stable conditions, and their cell density was measured daily by using a hemogytarometer type Neubauer improved. Cultures of a Greek strain of Brachiones plicatilis isolated from the lagoons of Mesologi and were carried out in 1 liter of seawater under optimal conditions. For each strain of microalgae used as feed, rotifer cultures were grown in triplicates and were daily fed at ratio of 15.24 cubic micrometers of biovolume of microalgae per 1 million rotifers. Also, rotifers and eggs were counted daily using a stereo microscope, and also the specific growth rate was calculated through the equation. The experiments lasted 1 week, except in cases where the population declined earlier. The total lipid content of microalgae and rotifers were determined by a sulfophosphovanillin reaction method, which allows the quantification of total lipids in non-human samples. In order to analyze their lipid profiles, microalgae and rotifer samples 2 hours after the enrichment were collected and through ultrasound-assisted transesterification were prepared for analysis using GCMS. About the results, as we can see, the lipid content of all examined microalgae was similar, with the average of 15.65% of their dry weight. The strain with a higher lipid percentage was D4 with 15.42% of its dry weight. These results are in agreement with current bibliography. And here we can see the growth of rotifers fed with 6 different microalgal strains. As we can see, the reference strain Dunaliella tertiolecta came up with densities until 265 rotifers per ml, but 2 of the examined strains came up with higher densities, such as 291 rotifers per ml for log and 267 for D4. Here is the SGR factor that, as we can see, the reference strain showed the higher SGR factor equals to 0.42, but the other 2 strains that came up with higher densities are following closely with 0.38 and 0.35. Here is the lipid content of enriched rotifers, and we create a baseline sample that is basically rotifers in starvation for 24 hours, so they have metabolized any nutrients from their previous food. The increase by the enrichment is from 33.5 to 182% of the baseline sample. Here are the fatty acids profiles of the rotifers and the enriched of microalgae and the enriched rotifers. And as we can see, the fatty acids that provided from microalgae in higher density, higher percentages are also coming out as higher in rotifers. Also, the ratio of total omega-3 to total omega-6 fatty acids are similar and directly affected by the enriched microalgae. The results of this present study were significant and in perfect sync with modern studies, which is remarkable considering that both the microalgal strains used as food and the used rotifer were strains isolated from Greek lagoons and salt ponds. The fact that two of the tested strains, log and D4, achieved higher rotifer concentrations compared to Dunaliella tertiolecta, which is commonly used for rearing rotifers in fish hatcheries, is very important and triggers the interest for further research and optimizing culture conditions and exploiting microalgal strains originating from the vast and dynamic transient Greek habitats. Also, the enrichment protocol led to an increase up to 182% in total lipid content of the used rotifer. Moreover, the fatty acid composition of the ratio total omega-3 to total omega-6 lipids of the differently enriched rotifer were directly influenced by the microalgal strain used as food. It was observed that the fatty acids were supplied in a higher percentage by the microalgae were also the ones that appeared in a higher percentage in the rotifers, namely palmitic acid and alpha-linolenic acid. These results are very promising as alpha-linolenic acid is the metabolic precursor for the synthesis of longer polysaturated fatty acids such as EPA and DHA, which is required in fish farming industry. Future projects to test and optimize different enrichment protocols in combination with screening for microalgae, reaching the requested fatty acids. That could lead to even more significant results as the rotifer used exhibited a remarkable ability to assimilate the fatty acids provided through the enrichment with the desired microalgae. Thank you for your time.
Thank you, Mr. Kolgas. Kovitis, I'm sorry. Is there any question from the audience, please? Can I ask something on your study? It was very interesting, actually, and I was wondering, how did you choose the 5 species? What's the idea behind choosing these species?
Of the macroalgae?
Of macroalgae, right?
We collect them from the transient Greek ecosystems, and because of their different salinities in these ecosystems, wanted to check how that affects their nutritional value and how that transfer to the rotifers, to the enriched rotifers.
Okay. Were the species chosen based on their fatty acid profiles as well?
Not in particular, because if we did that, we would choose the microalgae full of DHA and EPA.
Right. Because I've noticed some great values of DHA there, or I was wrong.
Yeah, there was one strain.
One strain was
MPA, but there was a problem in rotifer cultivation. They couldn't metabolize the outline of the macroalgae,
right,
because it's a naptophyte, and that way rotifers couldn't metabolize the DHA.
I see. Because on these tables that you show us on the fatty acid profiles, to my opinion, one of the major points is to highlight what happened to the EPA and DHA levels to the rotifers fed the microalgae, because this is the point that we need. We are doing enrichment of rotifers to enrich them in these fatty acids, and I think some rotifers, some groups of were enriched, if I'm not wrong.
In a small percentage. That's why we wanted to optimize the protocols of the enrichment.
Because you also mentioned that the linolenic acid is the precursor of EPA DHA. Is that happens in the rotifer biology?
No, they are not able to
bioconvert.
Yeah,
right.
The linolenic to a DHA or EPA.
So that means that we are not so much interested in microalgae in linolenic. Actually, we're more focusing on microalgae on EPA DHA.
That's right, but also we can use the macroalgae as that have a linolenic acid,
right,
to feed the higher levels of the biological circle.
Right, and also as you mentioned, as a balance of N-3 to N-6, this is also important, right? Right, thank you. Is there any question now?
So the next presentation won't be presented, will not be presented. So we can move on to the other one, entitled The Effect of Stocking Density of the Growth Performance of European Sea Bass and Salicornia, co-cultivated in the brackish aquaponic systems. Presented by Mr. Capetaños.
Good morning, ladies and gentlemen. My name is Kapetanios Manolis, and I'm here to present our study, which title is The Effect of Stocking Density on the Growth Performance of European Sea Bass and Salicornia co-cultivated in brackish aquaponic systems. First of all, we have to say that humanity is facing major challenges in terms of food security. More specifically, factors such as the consequences of climate change, the rising population, and the decline in fresh water are promoting the food industry to develop alternative, but at the same time efficient agricultural systems. Systems like that could be considered aquaponics. But what are aquaponics? Aquaponics is a polyculture method that combines recirculating aquaculture systems and hydroponics in a common system. You can use them to produce fish and cultivate plants at the same time. Research so far is focused on possible combinations between high value behavioral in fish and halophytes with high economic and nutritional value. Because, in order to be honest, food production sector is not a charity foundation. We have to combine high nutritional food and high economical profits. So, the aim of the study was to investigate the effect of three different stocking densities on the growth performance and survival rate of sea bass and Salicornia. Those two species are co-cultivated in a small-scale brackish aquaponic system in laboratory conditions. So, the experiment was carried out at the Laboratory of Aquaculture Department of Fisheries and Aquaculture at University of Patras. The experimental protocol was pretty simple. As you can see, the experiment duration was about 45 days. The feeding protocol was ad libitum by hand, a very common technique in this kind of experiments. The system salinities were boiled at 20 ppt, and you can see the average temperature. The temperature was about 20 degrees, an ideal temperature for the sea basses. So, three autonomous closed-loop aquaponic systems were used, and the total value of each aquaponic system was 400 liters. The main idea was very simple. A total number of 180 individuals of sea bass and in total of 24 Salicornia plants were used. Those species were divided into three treatments with different densities: 30 individuals per system, 60 individuals per system, and 90 individuals per systems. Or in other words, 10 individuals per tank, 20 individuals per tank, or 30 individuals per tank, respectively. The system, in a nutshell, was having three fish tanks per system. The filter was divided in three different parts: a mechanical filter, a biofilter, and a pump. And as a hydroponic technique were used the nutrient film technique. The fish were reweighted every 2 weeks, and in order to evaluate the experimental process, we used mathematical ratios such as SGR, FGR, DFI, and factors such as weight gain or survival ratio, as you can see in the slide. In order to compare the data were checked for normality, homogeneity, and comparisons of the means using one-way ANOVA. About the results. At the beginning of the experiment, there are no statistically significant differences in terms of initial weight and initial length. But at the end of the experiment, we have another story. The highest value of average final weight was observed in treatment with density of 90 feet per system, and there were statistically significant differences in weight gain in the 60 feet system. Another very important fact is that the food consumption, FCR, PER, and DFI values showed statistically significant differences between every treatment. A very interesting fact is that the FCR was ranged between 0.80 to 2.48, as you can see in the slide. But we are not talking about fish, we're talking about plants either. So there are no statistically significant differences in initial weight and initial height of the plants at the beginning of the experiment, but also in the final things were changed. The final plant weight and weight gain were significantly higher in treatment with a stocking density of 60 fish per system compared to the treatment with a stocking density of 30 fish per system and 90 fish per system. Also, the final height of Salicornia was significantly higher in treatments with a density of 30 individuals per system than in treatments with a density of 60 fish and 90 fish per system. As you can see, the highest value of the yield was also observed at 60 fish per system treatment. So, one problem that we have in brackish aquaponic systems is that we do not have the archive to make comparisons. So we use experiments with similar protocols or similar central ideas. As you can see, Maurizi et al. Used two different stocking densities for cultivating the common carp in a freshwater aquaponic system and reported an FCR ranging from 1.55 to 1.86, which was similar to the value of FCR shown in treatment B in the present study. I have to remind you that the FCR range from 0.80 to 2.48. Also, Chu and Brown studied three different stocking densities of Pacific white-leg shrimp, who cultivated with 3 halophytes, just like this study, in aquaponic system, and reported that when shrimp reared in a low stocking density, show higher mean values of weight gain and SGR. As we said before, Chu and Brown reported that the growth performance of 3 halophytes was better with increasing stocking ratio densities. Different fish stocking densities are a crucial parameter that influences Salicornia yields. And in general, Verma et al. Reported that an appropriate fish stocking density in the aquaponic systems has an impact on the high nutrient assimilation of the plants, a key factor at those kind of systems. So, the study provides information necessary for the rearing of the sea bass in a closed-loop brackish aquaponic systems co-cultivated with a Salicornia plant. The results of the present study, first of all, suggest that a high fish stocking density positively affects plant yield, and also Salicornia showed increased productivity at a density of 60 individuals per system, while the fish showed optimal SGR and FCR at the low fish density of 30 individuals per system. This is a part of the references that used for the present study. I want to thank you for your attention, and I'm here to answer every possible question. Thank you very much.
Any questions from the audience?
The audience is so quiet, eh? Come on. I have too many questions.
Of course.
Right. First of all, is that you study the stocking density, different stocking densities, or different biomasses?
Different stocking densities about the individuals. And that's the key point of the study, because in the aquaculture there are protocols. Every aquaculture has different protocols, have different stocking densities and different opportunities.
Where the fish, all the fish, they had the same weight, similar weights.
Initially, yes,
Yes.
in order to make comparisons between the treatments,
Right.
but not at the end. We want to see that the stocking density, different stocking densities, how they affect the food consumption and the growth parameters, because we know that lower stocking densities leads to higher body weights, higher biomass values, and there was a theory in general. But does this theory connect with the aquaponic sector?
Right, but when you are using 15, 30, and 60, if I'm not wrong, 30, 60, and 90, right? So you actually increase the biomass, right? The biomass in the tank, right?
The biomass was a part,
What I'm saying is actually
damage.
Right, yes. What I'm saying is that the plant growth, right? The plant growth, all these parameters of plant growth are majorly affected by the inputs from fish, which majorly affected by the feed inputs, right? And the metabolism of the fish. So did you calculate how much feed you insert, you supplied in each system?
We calculated the feed consumption of the fish, and the aim of the study was to focus on the ideal stocking density and plant yielding. As I said before, food production sector wants effectiveness, not only in terms of nutritional value and in economic terms also. So we have to find an ideal combination to say we have to find an ideal combination in order to provide them the best values of fish biomass and the best values of crop. So this study showed that these two things are not connected. Unfortunately, we see that we have the best values in terms of mathematics, of course. I mean, in 30 fish system about the fish, but at the same time, plants were affected the most positive way at the 60 fish system. That was the biggest, the most interesting finding in this research.
Yeah. what I'm trying to say is that in order to make comparisons between studies, the absolute numbers of 30, 60, 90 fish does not actually say something in order to compare with other studies. What is actually more important, to my opinion, is to signify the fish biomass and the feed input, what was consumed, what was not consumed. That's how you can also compare inputs between studies. And also you cannot compare HGR and FCR values of fish between treatments, between studies, right? It's meaningless for me. Like too many factors that are affecting these parameters. So you cannot say that I have a higher growth rate compared to other study, or I had a better FCR. What is most important is to signify, to calculate how much feed input was in your system, how much was consumed, how much was not consumed, if who could do, how much was metabolized, what's the nitrogen level in your system, and that will affect the growth, the growth of the plants, right?
In one way, yes. I mean, biomass is the key factor, but as I said before, and you said also, mathematical factors are range about the conditions. I mean, different feeding protocol could affect FCR and SGR in a very efficient way. I mean, we can feed them 3 times per day with a specific weight of a specific food.
Yes.
And the same way, and in other way, ad libitum would affect so many parameters, just like the stocking density, as I said before. I mean, it's complicated because we're talking about living organisms. We are talking about individuality and
Yes, yes. So sure. It's a complex calculations as well. But what I'm saying is that you probably the next step is to make you should try to find the whole allocation of inputs of the food, how much nitrogen, how much goes to the fish, how much goes to the water, how much absorbed by the plants. These are actually more strong factors that you can use to understand your results, right? The SGR and FCR values of fish are not so strong indicators, to my opinion, right?
That is, as you said, there are just factors to evaluate one specific experiment in specific conditions.
Yeah.
Unfortunately, I said before, the archive about Brachy's aquaponics is very narrow.
Yeah.
There are no data, so we have to make some cheats. So we have to see methods. We have to see.
I think you need more complex calculations, more in-deep calculations. I think it's difficult because you have a system that is affected by fish and plants as well. So,
of course, we're talking about a new whole ecosystem here. We're talking about plants, fish, and bacteria in the filter. We're talking about complexity level above the average.
Yeah.
Unfortunately.
I mean, how you can calculate the nitrogen absorbance of the plants? How you can calculate this, right?
Fortunately, the only way that we can do this calculation is to see the nitrogen input and the nitrogen output.
That's what I was saying. Yeah, exactly. How much nitrogen was in?
There are other studies that we try to do this, and the results were very interesting. To say, my postgraduate thesis was having this kind of calculation, and it was very interesting that there are very strong results. There are differences that you can say with naked eye, as we can say, about 40 to 50 milligrams per liter down. Think about absorption in real time, but you are not sure about this, if this absorption would actually affect positively the plants. You have to see their parameters that are not specifically. I mean, as I said before, there is individuality. There are plants that we have very strong root, they can absorb more nutrients and have a better result.
Exactly. There's a variability in the plant growth as well, right?
Fruit is destroyed or damaged, maybe the nitrogen will be in the optimum level. There are no absorption, so possibly we have a dead plant at the end of the experiment. So it's not representative.
Right. Thank you, Manos. Is there any?
Can I have to pose a question also? I was wondering, I've never worked on aquaponics or hydroponics. However, I was wondering, why don't you use a control group?
Group, it's not very easy because we cannot have a control group for the very simple reason we are not sure what control is about. Control could be such an aquarium with only fish, would be a hydroponic system only with plants. So the values between the control and the other treatments, hypothesis, I don't know how to express that, wouldn't be reliable. So, I mean, we have, as a main idea, the aquaponic cycle is that water makes the cycling economy between the nitrogen, between the carbon cycle, and plants absorb nitrogen, which is not good for the fish. So, can we calculate that nitrogen outside the control? We cannot do that because we have to change water manually. Manual water change in aquaponics is useless.
In terms of calculation, you can do that, but yes, as you mentioned, it's not a practice. It's not a practice in aquaponics.
Compare your results with both these like control groups you also mentioned.
Could be a control group, but we have to say that the manual control of aquaponics is something that it's not preferable because we provide sorry, I have a little dyslexia, sustainability of the system without human factor insert. I mean, if we have to change the water manually, there are no purpose to build a very complex system with 3 parts that we have coexist and having the optimum result. And for this reason, we have problem with the product label because we cannot consider aquaponic products as organics because of this reason. We do not have the ability to surpass blank references, blank definitions. For example, we cannot consider organic the aquaponic plants because there is a definition in European Union that says for that plants have to have roots in the ground, in the soil. Ground what? Soil what? What is soil? A blank definition? There are aquaponic systems, such like grow bed techniques, that we have soil, but it's not the symbiotic soil, you know? We are having a mean to support the plants in order to not flout, but there are no the soil that we're used to in an agricultural field. Unfortunately, we are victims of the bureaucracy. Unfortunately.
Okay, so thank you, Mr. Yeah. Yes, please.
I'm Manolis. Congratulations for your study. By continuing Elena's question, I'm sure that you use Hirayama equation in order to calculate fish density.
Yes.
Do you remember which is the highest value that Hirayama equation give to you?
No, to be honest, I do not remember that because we do not focus at this part of the research. I have to be honest.
Yes, but in order to have a control group, because you are studying the density of the fish, you can use the high value as a control group.
The highest value, you say?
Yes. And lower values for your study.
Okay, that could be a very good solution, I have to say.
Yes, and I will expect to see the values of nitrogen in the grow bed and the grow bed out.
It could be an addition that would help. That's true. But the aim of the study was to see something different. It was a missing link, unfortunately, and our aim is to correct the false words or the false problem or the problems.
Yes, because your results are not clear. You have great yield in 60 individuals, I think, for fish.
Yes.
But at the same time, this density so low biomass, the final way.
Not exactly low. Lower in means just a sec.
Yes, if you go back to your statistics, I think that it was the lowest.
Of course,
there were significant.
Of course, there were statistically significant differences. But in order to be honest, in industrial production, these differences are not considered.
Yes, but don't forget that you have only 45 days of cultivation.
45 days.
When you go to further days, this result could be very different.
And that's a good idea for the future because those kind of researches have very narrow period of time. 45 days, 75 days, or even 100 days. Unfortunately, in laboratory conditions, we cannot strain the wall, we cannot open the time period because there are difficulties that are above us, such like programs in the laboratories or other things. In the field, the things would be very different because we are talking about money, we're talking about about different time periods, like in the aquaculture, sea bream could take about 16 to 18 months from the larva stadium to the hatcheries in order to find the weight between 350 grammars to a kilo, for example. In these weights and in this biomass, the plant production could be very different because we are not sure about the yield and how many yields could have in this period time, this time, because a lettuce could be ready from the very first day to the very last in about 45 days, if I remember.
Yes, 45.
Well.
Lettuce is very quick to grow.
But
my question was about fish, not about the plants. How you explain that in 6 individuals, sea basses per tank, you have the lowest weight gain for fish and the highest yield for plants?
Of course, that's true, and that's the problem. I mean, not the problem with the symbiotic mean, but the problem that is just one research that can do that you can use for the first step. Maybe, as I said before, in another experimental conditions, but with the same protocol, we can see different numbers in the same treatments. So we have to find a way to combine the optimum plant selection and the optimum fish selection. That is a very fundamental sector that aquaponics studies have to focus on. In brackish aquaponics, because brackish aquaponics could combine high value in terms of price, I mean, fish and plants, especially now that we're talking about trends such as hygiene, such like superfoods or everything, and fish, very popular in the world, just like sea bream, just like sea bass, or even pagrus.
Right, right. Okay, thank you very much, Mr. Kapatanis, for too many answers that you gave us.
This meeting has come to an end. Thank you very much, all of you, for attending here. Our next meeting is at 5 o'clock in the same amphitheater for the aquaculture session. Thank you. Bye.
Test. Hello, everybody. I'd like to welcome you to this second morning session about marine diversity and conservation. I'm pretty sure that you're gonna listen very interesting talks. We have marine mammals, we have fan mussels, fishes, invertebrates, and so it goes on. So I'd like first to call for Ms. Sakritopoulou. She's going to give a talk about the novel, the method about the environmental DNA, and how she managed to detect a very endangered marine mammal species in the Greek waters.
Thank you very much, Mr. Gaffos. Good afternoon to everyone. So today we will be presenting part of our initial project that has to do with my PhD. We will be talking about the validation of the environmental DNA in the Gulf that we applied in the Gulf of Amphitheiater. My project basically involves three gulfs, Pagastytikos Gulf, Amphitheiater Gulf, and Thermaiko Gulf. So today we're going to be talking about the novel detection of the Mediterranean monk seal in the Amphitheiater Gulf via the eDNA method. Hello. So eDNA is a non-invasive sampling methodology that allows to detect individual, species, and measures genetic parameters proved to be essential for species conservation and management, and especially regarding threatened and vulnerable species, marine species. So far, the majority of species monitoring research has utilized eDNA as a complementary research method, along with visual and acoustic monitoring surveys. The latter, though, has been proved to be error-prone. The latter two methodologies has been proved to be error-prone compared to the eDNA methodology, where it's highly sensitive in detection and very valuable for animals that are highly also migratory species, or they live in large water volumes. Also, eDNA can not just detect, but also confirm previous records of species that have been unconfirmed using all these genetic methods and analysis. So what we have here. The method has been validated in a mesocosm sea tanks. Since then, a plethora of studies indicated a positive correlation of DNA concentration and estimated population density of marine mammals. So basically, DNA and population density goes together. And that was based to traditional methods. As a method can detect down to species level, even if the animal is absent while you're surveying, or it's in low DNA concentration in the water, or even if the acoustic receiver in an acoustic survey will fail to detect it, even if that acoustic receiver basically can identify the frequencies of the marine mammal species repertoire. So yes, eDNA sees more than meets the eye or hears the ear, and that's why you actually love it. In overall, basically, that means that as a method can be embedded successfully in a survey that has to do with acoustics and visual monitoring. So in this project, we applied, as I said, in three gulfs, the eDNA methodology, but today we will be presenting the preliminary results of Amphitheiater Gulf. Our goal was to apply the eDNA methodology in order to detect marine species in the area. And our focal point was basically the marine mammals. We started with sampling the entire Gulf in 8 stations, but today we will be presenting, as I said before, Station D, which is the deepest spot in the bay and the closest to the terrestrial activity. So we started, after we collected our samples in filter membranes. We basically, excuse me, yes. So we used the typical protocol of phenol and chloroform in our lab. The samples were soaked in lysis buffer along with protonase case overnight at 60 Celsius degrees and centrifuged twice using the analogy 25 to 24 to 1 in regards of phenol, chloroform, and isoamyl alcohol. For DNA precipitation, we used sodium acetate for 2 hours at minus 20 Celsius degrees. And there was a final washing step before the pellet was diluted in TE solution and stored in minus 20. Right. In terms of primers, we used 2 primer sets of 12S and 16S genes for final applicant product of 220 base pairs. For PCR, we performed it at 60 Celsius degrees for 30 cycles using concentration of 5 green GoTac buffer, 0.25 m of each primer, and distilled water in order to achieve the 20 microliters that required from the PCR. Negative samples were also tested, excuse me, and also to evaluate possible contamination over our process, our lab process. And we tested both the tested samples and the negatives in separate rooms and independently. And then we moved into Illumina barcode tagged primer generation that were finally loaded in the ion torrent sequencer. In terms of bioinformatics, it was a very certain process. So the fast Q reads were demultiplexed, checked for quality profiles, streamed and filtered using the data2 program via the R platform, and then frequency of each quality score at each base position of the reads were drawn and validated. Then the reads were truncated at position 210, and finally all the chimeras were removed and reads were blasted via the BLAST and plus engine against all the nucleotide databases in NCBI. In terms of results, so our method proved that the DNA is not only, what's going on? It's not only, I'm sorry, I don't know what's happening today. Anyway, so out of the 8 sampling stations, we focused on only 1 station, station D, where we obtained 2 samples of 3 liters each. And the results revealed 8 species, including our very own species, the Homo sapiens. From the initial evaluation of the sequenced applicants, a total of 646,630 total raw reads were estimated. However, a final set of 11,951 high-quality reads remained, out of which approximately 36% were identified as the Mediterranean monk-seal Monachus monachus. So consequently, the results point out that the eDNA is not only a valid detection method and monitoring method, but also a valid marine mammal detection method. This makes the eDNA essentially important for monitoring important species, such as the Dalmatian pelican that was also recorded in our study here, which is the global population considered as near threatened according to the IUCN. One of the most important things that I would like to address here is that if we biomonitor via the eDNA methodology indicative species such as the marine mammals that indicate ocean climate and ecosystem shifts, possibly a prognosis and early warning system production could be achievable and essential for especially Natura 2000 areas, such as the Anthracikos Gulf. And we know that we have a few of them, Natura 2000s, in our country. When combining also the detection of new to an era species to its Natura 2000 area under scope with the local environment history given by the public itself, then proper and improved policy management and conservation is doable. As indicated by the study, where local fishermen confirmed our novel detection of the Monachus monachus in Anthracikos Gulf, it appears that the eDNA method is a robust and solid genetic monitoring method that we can actually monitor and detect marine mammals in an area of high ecological importance and input. This novel detection opens the gates for a new scientific chapter for the Mediterranean monk seal in our country, and it will be exciting to see what comes after that. So it is extremely important then to identify if there is a local population in that area of the Ambracicos Gulf, of the Mediterranean Sea, within the Gulf, and if so, using genomics to estimate population size, effective size, and compared with previous studies, will also give us the opportunity to have an accurate situation, ecological situation of what's happening with, not just only with this specific marine mammal, but also how this is linked to the ecological status of such rich ecologically area like the Ambracicos Gulf. Finally, effective conservation management should be including direct measures of population effective sizes, level of heterozygosity in regards to depleted population on specific functions in ecosystem communities. I apologize for all the sardams. Usually I have less stress. Thank you.
All right. Okay, thank you, Elena, for this interesting presentation. Do you have any questions about monk seals? Please. Hold on, please. I have to pass you the microphone because the guys on the other side, they cannot hear us.
Thank you very much for your presentation. Your work is really valuable, so thank you very much. And I have two small questions. Which volume of water do you use for sampling and from which depths?
I can't hear you, I'm afraid. No? Louder and slower, please.
Okay. Thank you very much. Yeah, and my question is, how much water do you use for sampling and from which depths?
So we tried a few things according to the literature, the scientific literature, and we tried different depths. We started with surface, and then we moved in less than a meter. And usually, the more water you get, the merrier. So it's up to you how you want to design your survey.
But no less than 1 liter or?
I would suggest more than one.
Okay, and the second question, did you find location when using eDNA methods you can find that species, but using traditional methods it was not found?
I know what you're about to ask. So there were cases like in Klosek et al. 2019, where in the same survey, there was, they had to introduce a new species to an area that was the longfin pilot whale, right? So the acoustic receivers failed to record the repertoires, although it was seen by the marine mammal observer on the deck. So I think it's also a little bit of luck. And what is very fascinating with the DNA in general is that it never lies. It is what it is. So if you found it and you do not have contamination, then it's there.
Oh, thank you very much.
Any other questions, please? Yes, it's exactly.
Thank you very much. My question is how close we are to an accurate estimation of the population size, because you know the European directives require and mandate for quantitative data. How close we are?
We are very close, but you will have to wait for our upcoming peer-reviewed journal publication. So it's basically kind of in press. So I think we are really, really close.
Okay, thank you.
Thank you. Anyone else, please? All right, thank you, Elena. Let's move on, please, to the next one. So I'd like to invite Mrs. Kotromani. She's going to give a talk about and interpret the Microcosmos sabatieri and how it finds its spot on the phylogenetic tree of the Stolibobranchia.
Hello, everyone. I am Koutroumani Stella, and I am from the Department of Ichthyology and Aquatic Environment. Today, I'm here to present you our study about the phylogenetic classification of Microcosmos sabatieri within the Stolidobranchia order using the mitochondrial DNA CO1 gene. Let's walk through the aim of the study. The aim of the study is to elucidate the phylogeny of microcosmos within the Stolidobranchia order with respect to abiotic factors. This effort represents a pioneering work in the field, and we hope it will significantly contribute to future research and our overall understanding of the species. But first of all, let's explore Microcosmos sabatieri in greater detail. It is a solitary Ascidian endemic to the Mediterranean, mainly in the Western region, and especially recorded in the Aegean Sea, while it's also found in the Adriatic. In contrast to other Ascidians of the genus Pura, this species does not form dense colonies, but it does exhibit high population density and is easily identifiable in their respective area. Sabatieri inhibits various hard substrates at depths ranging from a few meters to over 200. Typically, it grows to about 10 to 16 centimeters, but can exceed 20 centimeters. Its external appearance includes a distinct wrinkled mantle, characteristic of the family of Puridae, and it has siphons with 8 red, or purple-pink stripes distinguished by light-colored areas, as shown in the image. As an ecosystem engineering species, Sabatieri hosts numerous epibionts, preserving local biodiversity, and that's also where it took its name, from micros and cosmos. It feeds by filtering seawater, and it is a hermaphroditic marine organism that reproduces from September to November. And at this point, I would like to state that the relationships between the families of Stolidobranchia aren't clear yet, as their phylogenetic relationships have not been fully defined. So, about the methodology that was performed, the sampling of Sabatieri specimens was made in the Pagaseti Gulf in the area of Agios Stephanos, where autonomous diving took place. After collection, the specimens were placed on ice and frozen to prevent degradation. We designed novel primers to amplify the CO1 gene, and the PCR products were then sequenced using the Sanger sequencing method. For the phylogenetic analysis, we used sequences from 72 other species within the Stolidobranchia order, aligning them through the AliView software. And Brachiostomal lanceolatum was set as an outgroup and belongs to Cephalochordota to help us root the tree. And we constructed the phylogenetic tree using the Mr. Bayes program and the Bayesian inference method. Let's proceed to examine the outcomes. Following the alignment of the PCR products, the mitochondrial CO1 sequence of Sabatieri was deposited in GenBank, database of NCBI. And here I'm proud to tell you that it's a unique and the first sequence in the world using with the succession number that you can see in the screen. According to the phylogenetic tree, 5 branches are observed. Of course, I will show you later the whole tree. In the first branch consists of the family of Molgulidae, which appears as a distinct monophyletic group. In the second branch, only the species of families only the species Cnemidocarpa areolata is found, which appears, in the second branch, only the species Cnemidocarpa areolata is found, although it belongs to the Stielidae family. In the third branch, species of the families Piuridae and Stielidae are distinguished. Studies have shown that the evolutionary relationships between these two families, the Stielids and the solitary Piurids, have not been clarified in previous analysis. Our study, the present study, validates the previous findings and reveals that the species within this clade appear scattered and irregularly distributed. Sabatieri is classified within the branch formed by these two families, primarily near the Microcosmus claudicans, Microcosmus polymorphus, and Piura scamulosa. Furthermore, it is noteworthy that the species Microcosmus camiger, Exasperatus, and Helleri have established their own taxonomic branch, separate from their initial classification within the Stielidae. So, let's talk about our findings. The results indicate that Microcosmus sabatieri exhibits divergence within its genus, suggesting a dispersal pattern within Stolidobranchia, influenced by ecological preferences that shape distinct evolutionary paths due to environmental conditions. For instance, while Microcosmus sabatieri is endemic to the Mediterranean, Stoloniferus, that's showing a significant genetic differentiations, thrive primarily into the Indo-Pacific region, notably Australia. Of course, death and light intensity are also influencing the Ascidian distribution. Another factor is the presence of epibiotic species on the Ascidian tunic. Large sponges often compete with Ascidians for space, hindering their growth. And Ascidians adapt their body form to fit the microhabitats topography and position their oral siphons for efficient feeding, which is vital for survival. Additionally, the growth habits, Additionally, the growth habits of epibiotics likely affect the tunic's mechanical properties and durability due to their weight, but this hypothesis is needing experimental confirmation. Additionally, interactions with closely related species and potentially hybridization events have likely influenced the genetic makeup of Microcosmus sabatieri and Microcosmus species. For instance, Microcosmus camiger entry into the Mediterranean and Microcosmus exasperatus limited distribution in the Eastern Mediterranean suggests the possibility of hybridization, supported, of course, by minimal genetic distances between these species. This observation is also spatially corroborated, providing further evidence, but though additional research is warranted here too. So in conclusion, the presence and the dispersal of Microcosmus species in the evolutionary tree suggest an ongoing determination of their evolutionary position. And our research provides new insights into the phylogeny and the phylogenetic relationships among cytons, emphasize the role of ecological factors in shaping evolutionary paths. We hope the study enriches the field of biology by offering valuable data on the origin and the evolutionary trajectory of these organisms, and also that our findings will guide the future research in the area, because as we already saw, it's necessary. I would be happy to answer any questions you may have. Here is the whole tree, and thank you.
Thank you very much. Do you have any questions, please? Yes.
Thank you. I'm Eugenia Lefkaditou from the Hellenic Center for Marine Research. From the Marine Research, we are reporting benthic species, but at depths more than 50 meters during the MEDIT survey. I don't think we have found this species. Is the species edible or not?
Yes, it is.
Is the one that we eat?
Yes, of course.
So it would be maybe easier to do genetics if you can find in the, if fishermen collect it.
Yes, yes. We can find it easy. But
I see that you collected through diving.
yes.
It's the only way. Okay. Thank you.
Thank you. Else?
Anyone else, please? Okay, thank you, Stella. All right, next one. I would like to invite Mr. Kondilatos. He's going to give a talk about the devil firefish and some population dynamics results.
Good evening, everyone. I'm Gerasimus Kondilatos. I work for the Hydrobiological Station of Rhodes, the Hellenic Center for Marine Research. My today's presentation is about the length-weight relations and the sex ratio of the devil firefish, Pterois miles, from the area of Rhodes. Okay. So the data I present here were collected within the framework of this project, the Expelias project, which is entitled The Design and Piloting Methods of Commercial Exploitation of Invasive Alien Species with a View to Contributing to Their Population Control. It was held exclusively in Rhodes. A few words about the Pterois miles. Devil firefish is a Lessepian migrant and invasive alien species. It was first appeared in the Mediterranean in 1991 in Israel. There was a big time gap before the second appearance, the second record of the species from Lebanon in 2012, and up to 2021, it had reached the southern Italy and Tunisia. It has already been described as a major ecosystemic threat in the Western Atlantic, mainly the Caribbean and the Gulf of Mexico. And there are high concerns also about the impacts of the species in the Mediterranean ecosystem. So the need for management measures towards the control of the populations are mandatory. The aim of this work and all the work on the species was to access certain biological characteristics for sustainable management. A few words about the area of Rhodes. Basically, on the eastern parts, it's Levantine Sea, and the western parts is Aegean. The area is characterized by two important hydrobiological phenomena, the cyclonic Rhodes red gyre and the Asia minor current, which transfers warmer waters from the easternmost Eastern Mediterranean towards the west. Once the alien species enter the Mediterranean through the Suez Channel and follow an unaided route, they will cross all the Levantine Sea and will enter the Aegean through the Rhodes Straits, the Carpathos, and the Kassos. So we are basically, in most of the times, the first to record such species. The fishing, the experimental trials were conducted in a depth range of 8 to 35 meters at 3 areas, pre-designated areas of the coastal waters of Rhodes. And we applied 2 different fishing gear, both static nets, the trammel nets and the gill nets. These trials lasted for 12 months. we used 2 fishing vessels for the trials. And finally, we collected 363 individuals. They were all measured for many morphometrics. And here we used the total length, total weight, and sex. Maybe a few more details on the sampling schedule. It lasted from April 2021 to March 2022. We did monthly 36 for each type of fishing gear. We did 36 trials. The gill nets and the trammelers were both of 600 meters in length and approximately 1.7 meters in height. And the mean soaking time was 1.5 hours more or less and 12 hours respectively. So we did all the length-weight relationships, and we found that all except for 3 have exhibited significant positive allometry. And we also found that there is no difference in the growth pattern between females and males, between sites, and between seasons, except for summer. Well, this is how the plot looks like. It was plotted for the 2 sexes, males, which were 179 in total number, and the females 194. Well, the male to female ratio was calculated at 1 to 1.03 male to female. And we also found out that beyond the 26 centimeters, the males are predominant. We also found out that there's no significant differences in the male to female ratio among seasons and fishing areas. We also did some comparisons between the seasons and the fishing areas. And we concluded that the males were predominated during spring and winter in site 2. And the females predominated in summer and autumn in site 1 and 3. The positive allometry in the length-weight relation indicates a growth pattern where the species grows faster in weight than in body length. The positive allometry during summer is attributed to the reproduction of the species there. In fact, we found that the species reproduce in rods, at least during July, August, and September. The growth of the species in rods does not differ from Cyprus. We got 2 more publications regarding the length-weight relations from the Mediterranean, one from Cyprus, one from Turkey. And we found no differences with Cyprus, but a strong difference with Ichthyology Bay, Turkey. In regards to the sex ratio, the 1 to 1.03 male to female is similar to the 1 to 1.07 from Ichthyology Bay, but it's rather different from the 1 to 1.55 ratio reported from Cyprus. Our results are very close to various publications from the Atlantic, where they have reported the 1-to-1 ratio. So in conclusion, the species has become well established in the coastal waters of Rhodes and feeds very well. This is the reference list. And do I have some time left? Yeah. I would like to also show you, because we're talking about conservation and we're talking about roads and alien species, some very important information that came out from the project. In part, in our trials, the non-indigenous species represented the 64.7 of total fish biomass in static nets. And that Pterois miles, along with Lagocephalus serratus and Fistularia commersoli, were holding the lion's share in terms of biomass in static nets in the Rhodian coastal marine waters. In terms of how to catch the species, it seems that for now, among the existing fishing gear, the most appropriate is the static nets. So looking into the archives, we have beekeeping in the hydrobiological station of Rhodes. And in terms of number of species, because one of the things that we are afraid is how this species and all the other invaders are impacting the biodiversity. When we used the fishing gear of boat sailing, as you can see the red column, the percentage of the non-indigenous species have risen since 2002 to 28.39 in trials we did this year. There is an increase in the percentages of the non-indigenous species, but there's not a clear, at least for now, for this table you see, there's not a clear diminishing of the number of indigenous species against the non-indigenous. However, we see a dramatic change and rise of the non-indigenous species in general. From 5, we have now been catching 19. And in regards to the static nets, we also see that the number of species, of alien species, has more than tripled. And the number of indigenous species also has tripled as well. But if we see that in regards to the biomass, we see a completely different picture. And if you go back to 2002, you see that the percentage of non-indigenous species was 0.2. And now, with boat sailing, this number has risen to 42.2. It's like an explosion of the species. And the same trend, as I say, happens with the static nets. All right, thank you very much.
All right, thank you very much. Actually, the supplementary file that you just presented was my questions. I'm right, okay. Do you have any questions, please?
Hello, Gerasimos, very interesting presentation. As I saw, the study areas were all in the Levantine coast of Rhodes.
Yes.
Have you any information if there's from fishermen what's going on the north coast?
Yeah. So in order to select the areas, okay, we did a lot of questionnaires. We covered a big percentage of the fishermen there, the professional fishermen, regarding on where and how many of these species fish and where. So the areas were selected based on their sayings, their answers. It was not randomly selected. They were based on what the fishermen believe that the species have greater numbers.
But
so it was a targeted fishing, yes.
So we suppose that there will be different with the northern coast of.
Yeah. For most of the alien species, there is a difference. I mean, we mostly found them in the eastern parts, in the Levantine, rather than in Aegean. And this is what we are looking all the time.
Maybe the comparison, you mean, I don't know how was the sampling with previous years by the Rhodes station. It was in the same area, or some percentages referred also to other areas. Maybe that's
a rough,
the percentage.
That's a rough idea. Actually, yes. I need to, be statistically analyzed and maybe go for a nice publication. But it requires time,
Okay, thank you.
Anyone else, please? It's very important to take the advantage of the fishermen because they are the first who spot any problem in the area. And then it's very important for us to get and grab the information. This is what's called local ecological knowledge. So that's what we did. We do the same with marine mammals. we always follow the fishermen, and they say that we see dolphins there, we see whales, and then we go and we validate the information. Okay, thank you, Akis. All right, last but not least, I would like to welcome Mr. Tsolakos. He's going to give about the status of the fan muscle.
Okay. Good afternoon, everyone. Thank you, Mr. Chairman. First of all, I would like to thank the organizing committee for the invitation and the opportunity to discuss with you the presented topic. Our study was about the estimation of the ecosystem services provided by the bivalve Pinna nobilis in Greece. As previously announced, my name is Konstantinos Tsolakos. I am a PhD candidate at the Department of Fisheries and Aquaculture of the University of Patras. My colleagues in this study were Professor George Gatselis and the Associate Professor John Theodorou. Pinna nobilis, whose common name is the noble pen shell or fan mussel, is a bivalve mollusk, a marine bivalve mollusk, a large species of Mediterranean, which is our subject here. A few words first, let's mention about the Pinna nobilis biology and habitat. The fan mussel Pinna nobilis is the largest endemic bivalve species of the Mediterranean with a maximum cell length of 120 centimeters, a maximum lifespan of 45 to 50 years, and it appears the fastest rate of cell growth reported for any bivalve, especially during the first year of the species' life. It inhabits environment between 0.6 meters and 60 meters in depth, but mainly in the zone up to 20-30 meters, which is the potential habitat of the species, mostly on soft substrates surrounded by seagrass meadows or on bare sandy substrates. The fan mussel has about 35% of its length buried in the seabed. The species also was known for its healing properties and was used medicinally during the ancient Greek and early Byzantine periods. I will try to be fast. Pinna nobilis mass mortality outbreak was first recorded in Spain in the summer of 2016. Unfortunately, in 2018, it also appeared in Greece, where mortality reached the 80% up at the we at last found that it was reached up to 95%, and in some areas reached the 100%. The cause of the mass mortality was the parasite Aplosporidium pinnae. Today, alive specimens exist at two sites, at Ambrakikos Gulf and Kaloni Gulf here in the island of Lesbos. Nowadays, there are many ongoing projects towards a common goal, which is to protect fan mussel. Such projects are the IMEDMAR, Life Pinna, Pinna SOS. I don't know if there is something else. A few words about what is ecosystem services. There is a range of definitions of the ecosystem services based on diverging views on how they are generated and linked to human well-being. Ecosystem services in general are the direct and indirect contributions ecosystems provided for human well-being and quality of life. Ecosystem services are distinguished into four categories: regulating, supporting, provisioning, and cultural services. I will provisioning services about food production, supporting services about nutrient cycling, regulating services, climate regulation or water purification, cultural services, spiritual education, or recreational services. The most important ecosystem services provided by bivalves, such as Pinna nobilis, as filter feeding organisms, are referred to the regulating services of nutrient remediation and carbon cycling, estimated of worth about 3 billion to 10 billion per year. The ecosystem services of the ecological importance of the fan mussel, Pinna nobilis, has been particularly noted as a birthing filter organism, as it contributes to water clarity by filtering large amounts of debris and retains a high percentage of organic matter. Additionally, Pinna nobilis, in terms of its ecosystem function, it provides a hard substrate in areas with a soft bottom, thus providing a surface that can be colonized by other benthic species, improving or even increasing the biodiversity of the area. Furthermore, it is mentioned as a good indicator of changes in marine ecosystems, which can provide information about the direction with human-derived activities. The fan mussel has not been exploited as a shellfish product, but has been traded in the past mainly through recreational fishing as a fine bait, as well as a decorative artifact, while it is noted for attracting divers. The aim of the present study is to quantify the biofiltration and the nutrient sequestration ecosystem services derived from Pinna nobilis in the Greek coastal zone. The materials and the methods we used is, first of all, the depth of distribution of the species, which extends up to 50-60 meters, but mainly in the zone up to 20 to 30 meters, which is the potential habitat of the species. To map the zone up to 30 meters, we used the estimation of the bathymetry in the marine territory from the available georeferenced bathymetry images at the node you can see on the slide. We estimated the fan mussel population structure in a closed population subject to constant mortality at all ages, the number of live individuals per ages NT and dead individuals per age NDT are given by the equations you can see 1 and 2, where T is the age and 0 is the number of specimens at age T is equal 0. The total number of living individuals n in a given space A for the ages Tn to T1 to Tx is referred to the abundance, from which it follows the equation 4 and the number of specimens at age T equals 0. I'm not going to say much about this mythology. I don't want to disturb you. The total weight of cells for ages T1 to Tx for the above population is equal to the equation number 5, where C and B are the parameters of the relationship length and weight of the cell. To estimate the total amount of nutrients sequestered in the cell, the total amount of each nutrient contained in the fan mussel cells in marine territory is given by the equation number 6, where P is the weight percentage of nutrient and AS is the total area of the coastal distribution zone of the fan mussel. And last, the economic footprint of sequestered nutrients, J to the species cell, is equal to the equation you can see on the slide. Finally, the total amount of water filtration F is given by the equation number 7, where F is the filtration rate in water volume per year and tissue weight cubic meters per year, R and V the parameters of the tissue weight cell weight. Finally, the total water volume of the coastal zone was estimated from the equation volume S equal dP multiplied by S, where dP is the mean depth of the survey zone. To calibrate the model, we used all these equations, parameters, and measurements and other data that are not yet published concerning the age, cell length, tissue weight, the filtration rate, total mortality, density, etc. The depth zone of 0 to 30 meters, as well as the presence of mussel, according to the provided sources, was mapped as you see in this map. And according to the results presented, the area of the 0-30 meter zone is 14,000 square kilometers, and the volume is 210 billions of cubic meters. The number of living individuals of the fan mussel is estimated at 1,400,000,000 plus or minus 300 million individuals, while the number of dead individuals of the fan muscle is estimated at 1,258,000,000 plus or minus 450 million individuals, while the total weight of the cells is estimated at 410,000 tons plus or minus 100,000 tons. The amount of carbon sequestered in the cells, and not, of course, allowed in the atmosphere, and not live in the atmosphere, estimated at 480,000 plus or minus 102,000 tons, nitrogen at 1,300 plus or minus 400 tons, and phosphorus at 166 plus or minus 47 tons. From the estimations of the carbon, nitrogen, and the phosphorus sequestered in the cell, as you can see in this table, we have to say that annually, at an economic level, the provided regulating ecosystem service ranges from 6.9 to 70.9 million euro per year. But after the episode of the mass mortality of the species in the Mediterranean, in the Greek maritime territory, the mortality was estimated at 101.1% according to Zoto et al. 2020. This means that the economic value of the ecosystem services derived by Pinna nobilis is reduced to 1.31 to 13.47 million euro per year. Finally, the total amount of water filtered by the living individuals of the presumed population of the Pinna nobilis species in the coastal zone is estimated to 2,587,000,000 plus or minus 800 billion cubic meters, which drops to 500 billions plus or minus 154 billions after the massive death of Pinna nobilis. Unfortunately, the remaining population is estimated to filter 115 to 1,300 billion cubic meters per year, which means a potential to filter the coastal zone of 30 meters depth from 0.5 to 6.18 times per year. Before mass mortality, the amount was 289 times to 32.53 times. That's a big difference. And discussion. The present study is an assessment of the provided ecosystem services by the protected fan mussel Pinna nobilis in the Greek Sea territory concerning the regulating ecosystem services of sequestered carbon and not released to the atmosphere, as well as nutrients, nitrogen and phosphorus in the cell of the species, as well as the supporting ecosystem service of water filtration. The estimate has been made by simulating a model incorporating population age composition, abundance and weight gain per individual and age for an hypothetical population of the species with a maximum age of 30 years. But we have to mention that the recent study suggested that mortality of species in 2022 was more than 95%, while the alive specimens recorded only at two sites, as we mentioned before, in Avrakikos Gulf and here in Kaloni Gulf, indicating that today the ecosystem services of species are yet lower from the estimated and are especially limited. The estimation of Pinna nobilis ecosystem services is based on assumptions that may be risky. A, natural mortality was considered constant at all ages. B, the increase in cell length follows the von Bertalanffy equation, whose coefficients are similar according to variation of habitat suitability. And C, the range of species was the zone of 0 to 30 meters depth of Greece marine territory. However, in order to control, I'm sorry, I was too fast. However, in order to control the risks, a significant number of uncertainties have been taken into account through the large number of prediction iterations with inputs of various combinations of the involved variables, practically covering those resulting from A and B. For example, natural mortality as input values to the prediction model ranging from 0.5 to 0 times its average value practically covers model failures from an unrealistic input value. However, although as uncertainties are taken into account and included in the model, they are transferred to the final results, increasing the range of variation of its predicted value. Finally, the uncertainties derived from the range of species probably are low given the confirmed presence of species in the whole range. Thank you so much for your interest and attention.
All right. Thank you very much. Do you have any questions?
Thank you. Indeed, very interesting. I would like to ask you, before the disease of this parasite, there was an estimation of the population range and density overall the Greek seas?
This is a very good question concerning that after the mass mortality, even though the Pinna nobilis is a protected species, it was never, as other species, were not measured for their population or for their ecosystem services. So after the mass mortality, scientists found out that we have to know what is the population we lost, we lost, and how to gain back what we lost.
So there was not, as I understand. So we don't know.
In many areas, we knew the presence. We knew the presence in many areas, but we didn't know the population.
Thank you.
Anyone else?
Now, I'm sorry, something more. Now we know that we lost something. We try to fulfill it.
All right. That's my question. Speaking of conservation, what's the next step if we want to save this species?
Next
and
step.
if there's a possibility for this species to be saved?
There are ongoing projects that they use right now the living populations to make to rehabilitate the places where they think they are more suitable for this species, and we hope that will be the next step for the species, for this species,
and
for
where
Pinna
they
nobilis.
where they find the individuals for that, and where they find the individuals.
Like Kos Golf, in Kaloni Golf, in some in Marmara, I think in Turkey, I'm not sure about it. In Croatia, in the north of Croatia, I don't know if there is any.
So it's like they move specimens from one region to the other.
That's the idea, to take them from the one place to take it to.
Have they considered the genetic pollution of this?
Genetic
pollution.
Genetic pollution from the transfer of the population. They were there. Right. They will transfer them in their places that were before.
Okay. Okay. Thank you very much.
Thank you.
Okay, guys. Thank you very much for this. I think this is let's break now. So please go enjoy yourselves.
Good afternoon. We can wait a couple of minutes. I think also. Okay. So, ναι, το άνοιξα. No, we are waiting for a couple of minutes, okay? Are all the presenters here? You are here. Mrs. Bilikova. Great. And Ziu. I don't know. Okay, so we can start, no? 2 minutes? Okay. Okay, it's 1:18. I think that we because we have later on. Yeah. So, good afternoon. I welcome you to this session, entitled Inland Aquatic Ecology and Resources. And we move to the first presentation by me. Yes, thank you. We are going to discuss about a quite popular issue during last 5 years, which is, can we move to the, just a moment, to the, okay. Okay, I'm going to the, yeah. We are going to discuss about the ecosystem services, and as I told you, is among the quite popular issues during the 5 last years. And the question is how we can integrate the approach and the concept of the ecosystem services in water policy and decision making. Let's start, no. Oriste? Anne. No. Ne, okay. So let's start with some statements as that the water has been defined as a common good. So we need the proper management. And of course, there are ethical issues embedded in all aspects of water management and water making decisions. And of course, everything related to water has to be done with the so-called nexus, meaning the water, energy, food, ecosystem nexus. What are the ecosystem services? As everybody knows, water bodies offer significant economic, cultural, aesthetic, but also the very well known to us provisional ecosystem services and also regulated ecosystem services as the nutrient cycle, the biogeochemical cycling, also the carbon and nutrient cycles, and also it offers recreational and educational value. This is the fundamental figure by Maes on 2016, linking the ecosystem services and the functions, the ecosystem functions from where the ecosystem services are provided and how they are linked with the socio-economic system and including the human well-being, the economical values, the health issues, and of course we have two arrows. The ecosystem services provide values to the socio-economic system, and from the other hand, there are drivers of change from the socio-economic systems affecting the ecosystem services. But what is the ecosystem services approach, ECA approach within water policy? There are some questions. Could ecosystem service approach be a new strategy for meeting the EU directives, such as the Water Framework Directive requirements, which is already an old-fashioned directive because it's 20-year-old, 24 years old. And what we need, that we need the mapping and the evaluation of ecosystem services and to identify how those ecosystem services could be involve people, could be provide us with decisions or what kind of actions. What are the water-related ecosystems who support the I think that everybody knows the sustainable development goals, water security and biodiversity, our regulated flows and extreme conditions, the purification of water, the replenish of the aquifers, the economic, all the ecosystem services, and also the water-related ecosystem services provide us with the way, with the mode, how we can sustain the global hydrological, carbon, and nutrient cycle. So it is the ecosystem services approach, or in other words, concept, it is already widely used in EU Green Deal, EU Biodiversity Strategy, and of course in EU Common Agricultural Policy. Until now, there is limited integration between Water Framework Directive and, of course, the other European directives as the Marine Strategy Directive, as the Groundwater Directive, with the River Basin Management Plans, and, of course, further, with the Program of Measures. The aim of this work is to evaluate how an ecosystem services approach could be used and value to the decision-making process, and address the issue of mainstreaming the ecosystem services under the light of biodiversity and nature conservation. I'm going very quickly to the methodology. We reviewed and mapped the 14 river basin management plans, including the 5 transboundary river basin districts, and we focused on a case study at the Nestos Delta Lagoon. We used the landscape population maps. We select the ecosystem services indicators. We used this sizes indicator and quantification system. We have field data from monitoring studies. And of course, we used the expert's opinion through semi-structured personal interviews. This is the study area, okay? And in the expert's opinion, we asked the experts and the decision makers, does including ecosystem services concept as a part of the program of measures, which is already running, pose a risk for nature conservation? This is the first question. And the second question is, how do we ensure biodiversity conservation by ecosystem services approach? Our results are focused first on national scale, on basin scale, and of course on the expert interviews. This is the map of with the 14 river basin districts. 5 are transactional water bodies, and the first note is that population water pressure in urban stress centers, and of course that the first pressure, the first driver in water management plans is the agricultural pressure, which is the 70% more or less in Greece. Here you can see the classification of the ecosystem services. For example, there are the provisional ecosystem services as the agricultural, as the abstractions of the water, as the drinking water purposes and drive. There are the fish populations, catches, or the need for the fish population conservation. There are the regulating ecosystem services, which are how are the nutrient cycle, what are the needs for environmental restoration, as the need for wetland restoration or the morphological alterations. And of course, there are the cultural ecosystem services focusing on research and development needs or aesthetic needs or in environmental regulation. If let's take a look to the spider diagram. We can see that, of course, the water abstraction and the provisioning ecosystem services are the lion, the big space for the ecosystem services, and focusing on our area, the East Macedonia area and Thrace, of course, there is a high environmental value because there are Natura sites, there are protected areas under the Natura 2000 network. There are at the same time, multiple economic activities, and of course, a number of the majority of population in a number of coastal areas, they are vulnerable to climate change impacts. Here you can find the drivers and the pressures in the area, flooding, point and diffuse pollution sources, habitat fragmentation, water availability, and of course, there is a gap in knowledge between the program of measures and the ecosystem services. Here is the status as we monitored based on field monitoring and based on a number of indicators. We have moderate ecological status. We have habitat fragmentation, many expanding saltwater intrusion in aquifers. We have sand extractions, okay? And you can see the linked ecosystem services in each one pressure and in each one restoration measures. Agricultural crops, provisioning water quality, provisioning chemical condition, regulated ecosystem services, biodiversity, regulating the cultural, moving to the water availability, for example, drinking water, provisioning, water abstractions for irrigation provisions. We have mapped and quantified. Here is the quantification of all ecosystem services. Coming to the expert interviews, we have found that important knowledge gap concerning monitoring, uses, and qualitative hydromorphological or biological and abiotic assessment. The EC inclusion, the ecosystem services inclusion, carries a high risk, according to their opinion, okay, to ecosystem services and biodiversity, because most of the people, even from the experts, believe that ecosystem services are how to abstract water for irrigation or for drinking water or the ecosystem catch or the fish catches. So nobody links that the ecosystem services approach is directly linked with the regulating of the ecosystem, with the cycles of nutrients, with cultural events and all this. And the first step in achieving a balance between provisioning ecosystem services and biodiversity is, of course, to promote both provisioning ecosystem services and the regulating ecosystem services. So, to our conclusions, synergistic actions that should be developed among uses as agriculture, as energy, as nature conservation, between the nexus and the ecosystem services. Program of measures which are mimicking natural processes as the nature-based solutions have been proposed in the river basin management plan, but usually have not been implemented. The current approach under the EU water policy is towards more a research perspective than an ecosystem-based approach. And of course, the administrative units, because 90% from our experts are from the administrative units, okay? So the administrative unit having the responsibility for the Water Framework Directive and for the Marine Strategy Directive should reconsider the structure in the river basin management plans in front of the third cycle, which is in front of us. Thank you very much for your attention. And any questions, please? Okay, we can proceed to the next presentation, which is evaluation of macroinvertebrate inhabiting in extreme habitat conditions. And we are speaking about Tuzla Thermal Spring in Turkey and by Dr. Inci. Sorry for the, okay.
Thank you. Throughout the presentation, we're going to see very brief explanation of macroinvertebrates and ecological parameters. And we'll see the features of the study area and how we carried out the study. And finally, I will share the results and discussion parts. So macrobiotic invertebrates are essential for aquatic ecosystems, transferring organic matter and found in various habitats like rivers, lakes, and even hot springs. And they are key ecological indicators for water quality assessment and are crucial for understanding aquatic systems. Understanding the relationship between environmental parameters and macroinvertebrates could help researchers to address stresses like pollution, flow changes, and habitat reduction. And they contribute to the biodiversity as well. As for the ecological parameters, ecological dynamics significantly influence species habitats and distributions with abiotic factors such as temperature, pH, salinity, and they play a crucial role for the ecosystem. Mostly the ecological parameters are in equilibrium in most environment and habitats, but sometimes it changes. So we can find pH very low or high, the oxygen can be very low and high, as well as the salinity. So it causes the extreme environments for the organisms, such as hot springs. So hot springs and thermal springs with high temperature and minor richness are a unique ecosystem for aquatic organisms and macroinvertebrates. And primary producers and consumers such as algae, diatoms, bacteria, and arthropods are limited in their ability to live in thermal springs because of the extreme conditions. And each hot spring and its biota has unique characteristics, altering biotic communities and affecting ecosystem level processes. Thermal springs are a unique ecosystem for many aquatic organisms. So as for our null hypothesis is the hot spring sources can affect the environment and the organisms living in the same habitats. So we test the null hypothesis. We ask 4 different questions, which is which kind of macroinvertebrates are present in the spring water fed by the thermal water source. And secondly, what is the seasonal distribution and diversity of macroinvertebrates, if there is any? And thirdly, whether there is a disparity in terms of biodiversity between the points that are not nourished by the thermal spring and those that are. And finally, we try to find out there is a correlation between water parameters and seasonal distributions. So the area studied is a spring water, approximately 130 meter long, 20 to 120 centimeter wide, and 10 to 30 centimeter depth. And it's flowing into Tuzla stream, and most importantly, fed by the hot water spring. So a distance of 6 kilometers separates the sampling area from the sea. Additionally, in the vicinity of sampling area, there is a geothermal power plant and a goat farm. The area where this study was conducted is characterized by spring water, which is formed by a combination of hot and cold water sources. So the sampling was done at 4 distinct locations, as you can see. The hot water sources, which is the station 4, and the cold water source, station 3, the junction of the points, station 2, and the point where it flows in the 4 seasons between January and October 2023. So physicochemical analysis of the spring water were conducted in each season. A special hand net with 0.5 millimeter mesh and 1 meter square quadrat area was used to collect aquatic macroinvertebrates. To analyze our data, non-metric multidimensional scanning, which is NMDS, was used to perform the similarities of the community composition among the stations and all seasons. We also performed a redundancy analysis, RDA, on the biological data and environmental variables. Of course, we conducted CDA analysis before the choosing which further analysis is the best, our data, RDA or CCA, etc. So this is the station 2. We collected the samples. Four sample areas were studied from the spring water fed by thermal water during 4 seasons, as I said before. The highest water temperatures was measured as 70 Celsius degree from the thermal water section, which is the station 4. And there was no specimen obtained from this station. So therefore, the analyses were conducted on the other 3 stations, S1, S2, and S3. Otherwise, the results could be nonsense. Upon examination of the temperature values, it has been determined that the location where hot and cold water sources are combined exhibits the highest temperature value throughout all seasons, except 1 season, spring. At station 1, it was observed that the temperature was in equilibrium throughout the year. So this indicates that the water emanating from the hot water source has an impact on the ambient temperature, resulting in an increase in temperature. So it has therefore been determined that it ensures the temperature remains a certain level throughout the 4 seasons. And the salinity is extremely high and causes the environment to be extreme as well. So the highest diversity belongs to station 1 with 5 families. Tipulite family was found in this station, different from other stations. And chronomids, which are a common occurrence and are abundant in numerous water sources, were observed in this study only in 2 stations, station 1 and 2, and in a limited amount. The dominant family is our study by 61% was AMPHITHEATER family. This family was found in all stations and was also dominated in terms of the number of individuals. So as a result of NMDS analysis, no differences was detected between stations and seasons. The main reason for this may be that the thermal water source with a temperature higher than 70 degrees Celsius feeds the river during all 4 stations, keeping the physical-chemical parameters, especially temperature, in a relative balance. Thus, a favorable environment may have been provided for the organism living here for 4 seasons. As for the RDA analysis, AMPHITHEATER and HydroPhyllida were the taxa situated close to the arrow indicating MF, conductivity, and salinity, whereas Ceratopogonida family was situated on the opposite sides, close to the arrow indicating water temperature. When we look at the literature, we see that AMPHITHEATER family can live in high saline environments as well. So let's back to the beginning, our null hypothesis. We had to test the hypothesis, we had 4 different questions. And our 4 questions was which taxa live in extreme conditions. After 4 seasons of sampling, we found the presence of 5 different taxa living in the environment, which is Ceratopogonida, Chronomida, AMPHITHEATER, HydroPhyllida, and Tipulida. Secondly, we investigated whether there is a seasonal change in these taxa. We observed an increase in the number of individuals in the fall. However, when we look at the NMDS analysis, we didn't find any seasonal and inter-stations differences. So as for the RDA analysis, it was observed that the salinity and AMPHITHEATER family and water temperature and Ceratopogonida family were related. So as a result, a relationship was found between water values and some taxa. And this is our working group team. So thank you for your attention.
Thank you very much. Are there any questions, please? Very interesting presentation. Congratulations also for the graphical presentation. It's amazing.
Thank you.
Okay. No questions? Okay, I have one question.
Yeah, of course.
It's quite important, this biotic homogenization of the invertebrates in this. How we can explain this? They are resistant on temperature or salinity, extreme high levels of salinity.
Yeah, extreme high level. Extreme high level. So it's kind of, we can say they have an adaptation to the extreme conditions. And we found correct information according to the literature, because we found out AMPHITHEATER family was the highest level according to the individuals. But Chironomid family, which are very common in the inland sources, but we couldn't find too much individuals in the study area. So the study area is a very saline environment, and the hot sources was very hot. I mean, it's like close to the boiling point of the water, but we couldn't find any individuals from the source, of course. And I think they have an adaptation for the extreme conditions. Maybe it comes from the genetic material, etc. So it has adaptations.
Okay, thank you very much.
Thank you.
Congratulations once again. Now, please, we move to the next one, which is identification of species of the genus Rutilus in Ukrainian waters, data from the analysis of cytochrome oxidase, subunit 1, and cytochrome B, mitochondrial DNA genes from Mrs. Belikova.
Good afternoon. Today I want to present our work about the identification of species of the genus Rutilus. They inhabit in Ukrainian water bodies using mitochondrial DNA data. Molecular methods now can help us to resolve some issues. For example, with managing of species that we have in our water bodies. And for example, we have different difficulties in identification of members of genus Rutilus because they inhabit various rivers in Ukraine and in Ukrainian reservoir, for example, as well in Dnipro estuary. And due to the different localities where they live, they inhabit different size, growth rate, habitats, and feeding preference, and it becomes too difficult to differentiate them. And in Ukraine, we have common roach, Rutilus rutilus, and Taran, Rutilus haeckelii. And most common idea that common roach inhibit fresh waters, and Rutilus haeckelii was found in estuary. But also there were some papers that some coexisting zone exist. That's why we need to investigate it. And according to our result, then we could manage how to deal with different population. And it will help us not overfishing some groups and to maintain genetic structure of populations that we have. And for example, such traditional methods of identification as morphometry and morphology sometimes leads to errors because that genus have very high variation. That's why it's commonly recommended to use not only traditional methods, but also molecular methods. And here we will present our studies about investigation of different population in different localities from Dnipro River and Dnipro estuary, which present different groups. We will use mitochondrial DNA data. And here we will combine then our results after morphological identification and how it connect with mitochondrial DNA data analysis. So here in our study, we investigate different groups from 3 different localities. The first one is Kaniv reservoirs, is freshwater. And here we have 2 forms, fast-growing and slow-growing forms. And the second one is Kakhovka Reservoir. It's also freshwater bodies. And the second point was Dnipro estuary. In first 2 localities, we mainly have specimens that was identified as Rutilus rutilus. And in the third one, in Dnipro estuary, specimen was morphologically identified as Rutilus haeckelii. And we want to compare results of
morphometrical analysis and mitochondrial DNA data. So for that purpose, we used two gene fragments. The first one is cytochrome oxidase 1. It's a well-known unique a universal barcode fragment. And the second one is cytochrome B. It's a very variable markers that was used also in some studies on roaches in different countries. And sometimes it's difficult to compare results because some people use only cytochrome oxidase 1 and the second one use cytochrome B. And here we want to compare results using two different markers and then observe which variation we can catch at different gene fragments. And our sequence that was obtained using the cytochrome 1 gene, we identify using BOLT database and sequence from cytochrome B, we identify through the NCBI database. For subsequent analysis, we use a standard technique that using always with mitochondrial DNA data, for example, as MEGA, POP-ART, Arlequin, DNA SP, and so on. And what we can obtain? So identification of samples using two fragments was the same for each samples. On the tree, you could observe that our samples create two separate clades. And the first one, it corresponds to Rutulus rutulus. And according to Larmozia, that was proposed in 2008, two clades. The first is Western Europe clade that corresponds to Rutulus rutulus. And the second one corresponds to Ponto-Caspian clade. And also when we used also reference data, separation was the same. And the Ponto-Caspian clade previously was described that it could contain not only Rutulus haeckelii, but also some another Ponto-Caspian roaches. And Lövin in 2017 proposed that species in that clade could be like synonymous to Rutulus lacustris, but also authors mentioned that without additional morphometric deep analysis and without using some nuclear DNA markers, it's not so good to determine this issue because we can just rename everything to Rutulus lacustris, and we need to provide also this additional data from nuclear markers as well. So as we don't have this analysis of all these roaches from Ponto-Caspian clade now, in our studies, specimens that was identified in Ponto-Caspian clade, we named Rutulus haeckelii according to fish-based database and also according to Eschmeyer catalog of fish from 2024. And then we proceed this specimen as Rutulus haeckelii because according to that database in water of Crimea and near Dnieper estuary, it must be Rutulus haeckelii. But we also want to conduct our research and investigate these differences as well. So also on pairwise similar skull matrix, we could clearly observe differentiation between Rutulus rutulus and Rutulus haeckelii that here mentioned in blue. And we saw that both of markers present the same result, the same identification of species. And according to this, 90% of specimen in fast-growing group and in a group of Kachovka preservers, 90% was identified as Rutulus rutulus. In a slow-growing form here,
it's about 77% was identified as Rutilus rutilus, and in Dnieper-Bug estuary population, 60% was identified as Rutilus rutilus. The remaining specimen was identified as Rutilus haeckelii. And it was to mention that in estuary group, not all individuals that was morphologically identified as Rutilus haeckelii was the same identified using mitochondrial DNA data. And it's also highlighted the importance of combined identification using as traditional methods and molecular data. And also the presence of both species in each studied locality confirms the sympatric habitat of these two species, and this contact zone is very interesting for further research. And we also investigate diversity using these two markers, and we observe that cytochrome B sequence was more diverse than this of cytochrome oxidase 1. And it correlates with the size of fragments. It's logical. And it shows that if we use cytochrome B alongside with universal barcoding element, it could benefit and allow us to study a relationship more deeply with more divergence, and it improve our results. And also now it's some thought that if we use more markers, it could provide some more correct delimitation of species. So we use 2 fragments now. And also on median journey network of these 2 species, we could see also clear separation of samples that was identified as a Western European plant late that correspond to Rutilus rutilus in gray ground. And it's differentiated by 8 mutation steps from haplotypes that was identified as Rutilus haeckelii. And the same result we observed using cytochrome B sequences. And as we could see, according to level of diversity of sequences of cytochrome B, we could observe more branched networks than in cytochrome oxidase 1. It's highlighted that it's better to use both of them. So what we have now, so we delimitate 2 species that we have in Ukraine and find a contact zone where species live, that 2 species live in one locality. Then we want to add our data about microsatellite diversity of that population to understand how they connect with each other. And also maybe some modern methods as SNP analysis and next generation sequences will be needed because that genus very variable, and it will be good to see the differences that we have. And since here we analyze specimens that was collected in 2019, 2021, before the war in Ukraine, then we could compare results after, for example, destruction of Kakhovka dams and how it will affect the genetic structure of wheat fish. It's very important. And we think that future research is crucial to understand in long-term perspective how our genetic structure of Rutilus will change in Ukraine. So thank you very much. I would like to ask the question if you have. Yes, in 2023. Yeah, and we collect data from 2021, and then if we afterward collect some samples, we will obtain how it
Is an irrigation though?
I think multiple course. It was.
Yeah, it
Yeah.
was.
Yeah. But we'll see.
Any questions, please? Generally speaking, it's a decline population, a population of Rotilos, or an increased population? Actually, it's a declining population. Okay.
Yeah, but Rutilus, it's
no, it's
okay
it's not extinct species.
Yeah, but in different reservoirs in Ukraine, we also monitoring the situation, how it changed through the years. So we'll see. Now it's a little bit difficult to analyze all of
the specimens.
Yeah, because of some art situation.
Yeah, and technical reasons, you mean.
Yeah.
Okay. Thank you very much.
Thank you.
The reduction of all spring. Okay.
Yes.
Hello, my name is Ziu Athina, and I'm a PhD candidate at the University of Patras. The present study is about the assessment of electrofishing application for the reduction of the Lepomis gibbosus population in the lake of Aos Springs. Invasive species are known to cause adverse environmental, economic, and social impacts. As a result of this, invasive species have been the subject of international agreements, initiatives, regulations, and conservation strategies aimed at preventing their spread, preserving biodiversity, and the timely eradication and management of the established populations. Lepomis gibbosus was first reported in Greece in 1885, and the first settled population was recorded approximately 100 years later on Aliakonas River in Aos Spring Reservoir, was introduced during the middle of the previous decade. Lepomis gibbosus prefers shallow waters with little water movement and enough vegetation. The temperature that it lives ranges from 4 to 30 degrees Celsius and survives and reproduces in a wide range of habitats. In Greek ecosystems cause a significant impacts on the biodiversity of the ecosystem that it shelters. The Lepomis gibbosus reproduces in multiple makes multiple effort multiple reproductive efforts during the breeding season. Males individual crowd in the same area and creates nests. Also provide parental care, sheltering, and caring for eggs and newly hatched juveniles in the nest. The hatching of the eggs lasts from 3 to 6 days, depending on the temperature. For this study, the method of electrofishing was selected because it's a non-lethal active sampling technique, effective in clean and shallow water, has the smallest number of negative effects on native fish population compared to the other removal methods. It is also preferred for removing Lepomis gibbosus from aquatic ecosystems because of the particular reproductive behavior of the species. The aim of this study is to assess the effectiveness of electrofishing as an active method to mitigate Lepomis gibbosus population and to identify the impact of electrofishing to the other native species. Some of the lake characteristics is that the Aos Spring Lake is an alpine type lake and it is located in the northern Pindos. Is oligotrophic and its altitude is 1,000 343 meters. The lake has a maximum depth of 40 meters and an annual temperature variation of 4 to 26 degrees Celsius. The fish fauna of the lake consists both of endemic and introduced species. Endemic species are Salmopharoides, Barbus prespensis, Squalius espeaos, and Albunoides bipunctanthus. And introduced to the lake are Ogorhynchus mycis, Cyprinus carpio, and Carassius gibellio. To evaluate the mitigation of Lepomys gibbosus population, experimental electrofishing from the coast was applied around the lake during the day, using a portable electrofishing device. Nests identification took place from May to September 2021 during the breeding season of the species. Electrofishing sampling took place from July to November 2021 at the places where nests were identified. And also electrofishing was applied from June to September 2022 and June to August 2023. As you can see in the map, electrofishing was conducted to confirm nesting areas from surveys that took place in 2021. The yellow dots represent the electrofishing surveys that took place in 2021. The red dots represent the surveys that took place in 2022, and the blue dots the surveys that took place in 2023. For each sampling, the start and the end time of the sampling were recorded. Samplings were done by a device operator, followed by one or two individuals. Collected specimens were transferred to a recovery basin and their species and number were recorded. Individuals of the non-target species that recovered were recorded and returned to the lake. And Lepomys gibbosus individuals were kept for further study in the laboratory. A total of 63 samplings were conducted. 25 trials occurred between May and October 2021. 13 trials occurred between June and September 2022, and 25 trials between June and August 2023. In total, were caught 1,060 fish individuals, with Lepomys gibbosus comprising the majority at 1,147 individuals. The remaining 453 individuals consist of 5 other species, such as Albunoides bipunctanthus, Barbus prespensis, Squalius espeaos, Cyprinus carpio, and Carassius gibellio. Recovery rates for non-target species were also estimated. And for Barbus prespensis, it was 55.2%, for Cyprinus carpio, 64.3%, and for Albunoides bipunctanthus, Carassius gibellio, and Squalius espeaos, the percentage ranged between 80 to 23%. Recovery rates indicates the percentage of individuals returned to the lake. Despite the relatively higher mortality rates observed in Barbus prespensis and Cyprinus carpio, these species, comprising 2.3% and 3.6% of the sample, respectively, so the representation suggests a sporadic occurrence in the study area. So the performance of electrofishing in terms of the total number of Lepomys gibbosus captures was estimated. Particularly, catch per unit effort was estimated for the length classes less than 5 cm, 5 to 7 cm, and greater than 7 cm, and for all lengths in total. The mean performance for individual with length less than 5 was 2.14 for the year 2021, 0.95 for the years 2022, and 0.7 for the year 2023. The mean performance for individuals with length 5 to 7 cm was 8.65, 11.81, and 2.5 for the years 2021, 2022, and 2023, respectively. And finally, the mean performance for individuals with length greater than 7 was 6.12, 5.21, and 2.93 for the years 2021, 2022, and 2023, respectively. The performance ratio for class lengths 5 to 7 centimeter had a decrease of 75.1% for the years 2023 compared to the previous years. Also, there was a decrease of 48.2% for the length classes greater than 7 for the years 2023 compared with the other years. And finally, we see a decrease in performance for all lengths in total. The average sampling time per day was calculated 1.2 to 1 hours per trial. The sampling time excludes equipment preparation and movement between regions. Equipment preparation takes no more than 15 minutes per operation, and movement between stations does not exceed 30 minutes, given that access to the coast by car is available at all stations. And it's estimated that a 2-person team can cover between 2 to 7 positions per day within an 8-hour duration. And within a 6-hour duration window, the team can cover 1 to 5 positions. The aim of the present study was to assess the effectiveness of electrofishing as an active method to reduce Lepomis gibbosus population of the impoundment of our springs. Electrofishing was deliberately targeted at the breeding grounds of Lepomis gibbosus during their nesting period, aiming to remove the reproductive population and destroy the nests in these areas. Removal of male individuals from the nests results in a complete failure of the nests. Larvae and juveniles of the species are concentrated in nest areas, making them an easy target for electrofishing. And deploying electrofishing in active spawning areas can profoundly affect the survival of larvae within or on the substrate, particularly during their vulnerable developmental stages. The other species, the mortality of the lake fish population should be considered minimal because the remaining fish species caught was a part of the lake's fish fauna, and 2 of them were introduced, such as Cyprinus carpio and Carassius gibellio. And electrofishing was not applied in areas inhabited or preferred by the species caught, nor did it affect their reproductive areas, indicating that the fishing is likely due to occasional occurrence in the species in the study area. So, the use of electrofishing under specific conditions, focusing on the breeding grounds of the gathering place of the species, is profitable for Lepomis gibbosus. Also, the use of electrofishing with proper management results in relatively small mortality in the other species of the lake. The time required per station allows all possible concentration points of the species to be covered in less than of egg hatching time of 1 reproduction cycle of species. Thus, this tool is sufficient for the management of the population of the pumpkin seed in the impoundment of our springs. The above results concern an important element for the design and organization of the programs for the elimination or reduction of the population of the species in the impoundment of our springs. Thank you for your attention.
Thank you very much. Are there any questions? So there is, we have a question.
In Canada, where its native place, they eat the fish, yes. But in Greece, I think it's not tasty. We have the sea with many tasty fishes. So no, they don't eat the fish here, of course.
Of course, yes. No. And now is it the dominant species in our lake?
It's not dominant,
but it's in high population.
Yes, it could be in high population. As I said, it's an invasive species that can reproduce easily many times in breeding season.
It's amazing for this lake. Huh?
Yeah, and the lake it's alpine type. It's really high, so
and in high ecological status.
Yes, of course.
Okay. So, if there are no other questions, I would thank you for your presence in this session.
Το βρήκα. Ευχαριστώ πολύ.
I'm just looking, is that so?
Ένα-δύο.
Okay. Hello. It's a pleasure to introduce this first speaker of the session on oceanography, Professor now Yannis Androulidakis, who is going to talk about his work on applications of Wave for Us operational platform to support first-level response activities in the Thermaikos Gulf. Yanni?
Hello, can you hear me? Yes. Good afternoon, everybody. Thank you, Professor Dragou. I will present some very recent upgrades that we did for our operational platform system in Thermaikos Gulf. This is in northwestern Greece. The main goal was to produce some services to support first level and managerial activities. I would like to say a few words about the Thermaikos Gulf. Most of you know where it is. It's in northwestern Aegean Sea, and it's a very important Mediterranean coastal ecosystem that accepts very strong anthropogenic pressures, especially urban pollution, ports, and there are a lot of aquaculture activities. And the most important is the strong river discharges. There are several outflows around the Gulf, the coast, and especially they are located in the, as you see here in the map, in the western part of the Gulf, where it is also Natura protected areas. These discharges control the quality actually of the Gulf because they have very high nutrient concentrations, and so they can form and induce strong eutrophication events. These are some examples here of red tides, and the upper left is a dirty sea phenomenon event in very close to Thessaloniki city. So our main goal was to develop an operational forecasting system and cover several components of the marine environment and provide services to first-level responders, to management authorities, and also to the broader public. Before I continue, I would like to show this slide. This is from our recent review that we did in the Gulf the last 50 years. And it's impressive that with the red line, you can see the number of studies conducted in the Gulf from the 70s until today. There is a peak in the 90s and 2000s. And then after that, especially after 2010, which is especially due to the economic recession in Greece, we had a drop of activities and research activities and monitoring, especially activities. And this is, of course, this made the need urgent to develop accurate tools. Of course, they cannot replace monitoring, but it's that should recover after this decade of recession. And so it was a need to support first-level responders, especially when we have an accident or pollution event. This is the whole structure of the system. And you can see that we start with meteorological forecasts. This is, in my opinion, the impressive, one of the impressive parts of this, because they have a very high resolution. 1.6 kilometers for meteorological forecasts is quite high. And then you can see the nested components of the system with ocean circulation, storm surge forecast, waves. We also conduct predictions of hydrological forecast because we need the river, which is very important, the river discharges. And we finally go to predictions, estimations of coast flooding in the lowland areas, and also oil spill forecasts in the case of an accident. This is operating daily with providing 3.5 days forecasts, and we also can provide on-demand predictive implementations when an accident happens or a search and rescue activity is needed to be done. I will present a few examples of this system. First of all, this first example is an accident of an oil pipeline, which is a permanent pipeline in the northern part of the Gulf. It's where you can see here in the, you see my cursor? Okay. In the Gulf of Thessaloniki, it's very close to the city of Thessaloniki. And this happened in November 2017, when we had an instantaneous release of oil mass in the sea. And you can see here the three, on the left is the meteorological prediction of the following days. And okay, now it plays. And we have prevailing southerly winds and southeasterly winds. And this type of conditions, wind conditions, they mainly form a cyclonic circulation in the eastern side of the Gulf. And the most important is a jet, a southward jet in the central area. This is also is in agreement with our previous monitoring and drifter experiments that we did in the Gulf the previous years, and it also supports this type of circulation in the area. And in this case, we have minimal exchanges with the rest of the Gulf. And on the right, you can see the prediction of the oil spill, which is mainly remained in the upper part of the Thermaikos. These are two snapshots, one day and 7 days after the release of oil. And on the upper panels, it's an estimation of the satellite oil spill from ocean color sentinel images. This is the spreading of the oil southward. It's similar, not exactly the same, of course, but similar southward spreading of the oil spill. And 7 days later, we have oil patches on the eastern side of the Gulf, and similar is the model. The agreement is not very good, but we have some uncertainties that if we knew them, maybe we could improve the forecast. This has to do with the exact oil mass. We didn't know that. It's not provided by the authorities. It's not easy to find it unless they want you to do that so they can provide it. Then you can start your forecast and the initial extent of the oil spill. So if we had that, maybe we had a better prediction. And this is a hypothetical in the same area, hypothetical continuous now leakage you see the floating oil. So after a 3-day prediction, you can produce a map of the affected area, the potential affected area. And also you can also provide estimates of the cost of the effect on the impact on the coastal on the coast with a beached oil that might be stranded along the coast. Okay, let's go to this is a third example. This is related to extreme weather conditions, a very strong storm that can produce severe rainfalls and finally strong freshwater discharges into the marine environment. This is an example in November 2017. This is a Medicay Numa. You can see the track of the storm, and that affected Greece, and especially the northern part of the Greece, where we had very strong precipitation rates. And finally, this is our prediction of the entire year of the discharges in all rivers of Thermaikos, and this is the peak in Aliakmonas River during those days. And below you can see this is a comparison of our estimations for 4 years for Aliakmonas again, with measurements in transport measurements in Aliakmonas. In this case, we have used different components of the system. Our final goal was to track the pathways of the riverine waters and estimate the potential areas that could increase the chlorophyll concentrations and produce algal blooms. So on the left, the two animations. This is the tracers that were released continuously in Aliakmonas delta, in front of the delta, and how they spread and evolved during the following days. And below, it's the salinity distribution. In both cases, you can see the very strong southward coastal current along the western coast. With similar patterns can also be were observed with the use of satellite ocean color images. This is satellite-derived turbidity in the same day. This is the particles during that day. And on the right is the salinity. And below, you can see 3 successive dates of the chlorophyll distribution and how the high chlorophyll concentrations are right after the storm along the same coasts, the southwestern coasts. Another example now is the detection of marine heat waves. Marine heat waves, as you probably know, it's a climate indicator of that has been introduced last years. It's actually a statistic variable, and especially in Thermaikos, we can see that it was characterized by other studies in the past as a hotspot for increased temperatures, sea temperatures. And you can see here the number of marine heat waves. Actually, it's not the number, it's the intensity. It's combined the number with the temperature. The last 13 years after 2010, there is an increase in the number. And this type of events can affect the marine environment and ecology. These are 2 examples here with mussels on the top and coral mortality on the below. These are were observed in summer of 2021, which was a very intensive summer. And that's why we also want to predict this type of events. This is the comparison of SST between our model, our system, and satellite averaged over the entire Gulf. And these are the 6 events we managed to detect based on the Hobday methodology. 5 out of the 6 events, which is okay. Could be better, 6 on 6, but 5 out of 6. And it's good that the dates were the same, at least the periods that they occurred. And you can provide in this way very valuable information, for example, to aquaculture farmers that it's the last event, last example here. This is also related to strong storms. This is our prediction of the storm with our system. This is downscale to Aegean and then from Mediterranean. And then this is the 1.6 kilometers precipitation and winds over the Central Macedonia. And this is the storms surge, the sea level compared with the tide gauge, which is quite good. And below we have the waves. And this is the coastal flooding of these days, which is different depending the type of the sea level is related. For example, here we have the wave setup is more important with the waves, strong waves in 24 of January. The next day, the surge was very strong over the entire area. And so this different type of flooding and different water level of flooding depending where you are in the Gulf. Our next step is to develop and couple a biogeochemical model. This has to do with the impact in the ecological state of the Gulf. Could provide us information about that. The uncertainty here is the knowledge of the quality parameters of freshwater outflows. I'm not sure how we can get that now. And then the other one is the compound flooding, also include the flooding from the flash floods in areas that we have both coastal and floods from the inland. Thank you very much. Oh, really? Sorry about that.
Yes, a couple of questions we can take. Yes, Adonis? Short ones, please.
First of all,
you have to speak to the microphone.
Hello, Professor Androulidakis. One question, or two questions, actually. The first one is about these marine heat waves. Have you got any kind of evidence that there were some meteorological conditions as well changed after or following the marine heat waves extremes, like, for example, increase in humidity in the air, or more precipitation and so on from the time series?
We have not checked that. This is interesting. What after the favorable conditions, but after the events.
After the events, maybe there is a lot of evaporation and so on. Maybe affect that area or other areas. That's one thing. And the second one is about this. You want to do combined flooding. I'm interested about how you're going to deal with the boundary conditions, both in the ones from the land and from the sea. I mean, how you can define the area that basically you have to. That's one thing. And the second is, have you ever thought about how to basically validate this type of flooding data? So, for example, how we can have some kind of evidence to make sure that our models work properly? Thank you.
I'll start with the last one, which is more in my field. I know it's we have used until now. We have used the ocean color images to do that. For example, compute the NDWY index, and but the problem with this is that during a storm, for example, you have clouds, so it's very difficult to find a cloudless image, but you can do it right after the storm sometimes, and you can with this index you can. We have a couple of articles that
very difficult.
It's very difficult. It's very unsafe.
Very unsafe. Maybe good idea would be to have some kind of information from local areas.
Yeah,
that shows the height of the flood, how far is.
This is the only way to have from the field. So you have to, if you could take information from the local people with mobile phones or videos, or the best way it would be to, especially in areas that you have strong hazards like that, risk to have tight gauges, like cheap ones or portal ones that you can put in several areas, places that they can measure the water level and give you such type of, but you need a lot if the area of the flood is.
I'm very sorry I have to stop.
Sorry about that.
But if there is anyone else who needs to ask something, but not as long as Adonis did.
Maybe was my answer was wrong.
Please, if we do not have any questions, we can move on. And perhaps have more time with Yannis after the session. Thank you, Yanni.
Okay, thank you, Nico. Nikos Kliris is the next speaker, all the way from Southampton. He's going to talk to us about the accelerated drying and salinification of the Mediterranean Sea, implications for the overturning circulation.
Hi, everybody. So today I'll talk about some climate change impacts on the Mediterranean Sea, especially salinification of the basin and some implications about the overturning circulation. So one of the consequences of global warming is the amplification of the water cycle. So wet regions of the planet become wetter and dry regions of the planet become drier. So the Mediterranean is a highly evaporative basin, and recent reanalysis data sets show a very intense drying of the basin, which is mainly driven by evaporation. So if you look at these 4 panels here, we have evaporation, SST, precipitation, and the net evaporation minus precipitation phase. So you can clearly see that SST drives this increasing trend in evaporation of the Mediterranean over the 45-year period. So this is ERA 5, relatively recent high-resolution reanalysis data set. The other components of the freshwater budget, so precipitation trend for the whole region is actually close to 0. You can see this gradient of higher precipitation over, for example, the Aegean northern parts of the basin with respect to contrasted with the decreasing precipitation in the southern part. So the E-P trend, which is quite significant, is about 18% increasing net evaporation over this 45-year period. So in comparison with the global amplification, which is about 2 to 4%, this is a huge increase, several times more. So, of course, this drying, atmospheric drying causes a huge trend in salinification of the basin over the same period. So using here the EN4 data set, the Toffee's Hadley Center data set, you can see the salinity anomalies for 3 layers, the upper 150 meters, 150 and 600 meters for the intermediate, and the bottom layer. So they all saw robust positive trends in the upper layer after the 2000s, and like continuous trends in the intermediate and deeper layers of the basin. We can also see some peaks here in both East Med and West Med basins related to the climate transient events, the Eastern Mediterranean transient and the Western Mediterranean transient. Looking at the warming of the basin, so we have simultaneously very strong trends in the warming of the basin in the same layers. So increasing temperature, again, after the mid-2000s, have strong increasing temperature in both subbasins and in the bottom layer. But the effect on the density of the basin is difficult to distinguish because you have these opposing contributions of temperature and salinity. So especially, so there is no significant trend in density in the upper layer. Maybe in the deeper layer, we have some increasing trends after the 2000s. As you can see most clearly some peaks related to the Eastern Mediterranean transit here, for example, but not clear trends in density. So what my research here is using this water mass transformation framework. Here, at first, I'm applying that in salinity space. So we have this relatively simple equation which says that the volumetric change in salinity space is controlled by either the water cycle and the interior mixing in the basin. So changes in the volume of an isohaline surface, for example, this volume here can become bigger or smaller. This depends only on the water cycle, so evaporation, precipitation in river runoff, and interior mixing. So the volumetric distribution can change following changes in the water cycle. So we're expecting after this very strong drying of the basin to see some strong volumetric changes. So this upper panel here shows the volumetric distribution in 1979 and 2023. So we have two peaks here. The blue one is the Western Mediterranean, the right one is the Eastern Mediterranean, and the dashed lines here are for 2023. So you can very clearly see this displacement of the volumetric distribution of the two peaks further to the right towards higher salinities. So this is drives a strong transformation of volumetric transformation of waters towards higher salinity. So this negative trend here indicates that we have a freshwater transport from more saline to less saline waters throughout the basin. So everywhere is negative. So this is a cumulative change, volumetric change. So it goes, when it goes down, it means that you are losing water, volumes of water, which are displaced further to the right towards higher salinities. And then this increases again and goes back to zero because you have water mass volume conservation in the basin. So you can see that actually at lower salinities, like the Atlantic water or Dardanelles water entrance, transformations are very small. And the big transformation happens in the interior of the basins, the deep waters of the Mediterranean Sea in both basins. We can do that in specific regions, like the main dense water formation regions of the Mediterranean, so the Aegean Sea, Roach, Gyre, and South Adriatic for the east, and the Gulf of Lions of the west. So we get the same pattern of very strong negative transformation, so salinification. More salty waters are formed in the two subbasins, both intermediate and deep waters. And this is, much of it is clearly driven by the climatic transit events in the two basins. Now we can do the same analysis, but for temperature. So see what's happening in temperature space. So again, the volumetric distribution, of course, goes towards warmer waters. So this is the volume of water, this is temperature. The whole distribution is moving towards much warmer waters. So we have transformation of cold to warm water. Much more in the Eastern Mediterranean, though. And so what is the effect on density? Now, doing that transformation in density space, where we have these two opposing trends in temperature and salinity, opposing transformations, we get actually a very small displacement to higher density, which means we have higher increasing density, especially in the deep properties, in the intermediate and deep properties. If you look carefully, you can see actually that we have opposing transformations. So the low density waters become actually a bit more dense, but the deep and intermediate waters become much denser. In order to illustrate further that, we can do the same transformation, but in different layers. So the upper layer will have actually decreasing density, and in the deeper parts, we have the opposite, increasing density. So one consequence of that is you have actually higher stratification because you isolate further the upper with respect to the deeper layer in the Mediterranean Sea. The obvious space to see water mass change, of course, and water mass transformation is in TS space. So this is the same analysis, but now using the TS space. So all these points here is for each point, we have the volumes of water for specific temperature and specific salinity. So we can clearly identify where we have the Western Mediterranean water or the Eastern Mediterranean waters. And we can calculate from the change in volumes of water, we can calculate the transformation rate. So the transformation rate is the volumetric change over this 45-year period divided by time to get the transformation rate in Svetra. So the transformation rate picture is quite obvious here. So we have decreasing volumes and we have a transformation towards both warmer and saltier water. So you can see here, this is the Atlantic water, becomes mainly warmer than actually saltier. So actually the density is lower, decreasing, goes towards lower densities. But as you follow the Atlantic water into the Eastern Mediterranean, so this change becomes along isopycnals. This is for the upper layer. So this is 0 to 150 meters. If you go to the intermediate layer there, things are much more clear. So you can see that the transformation from low density to high density here. So you have transformations leading to crossing isopycnal. So clearly showing in both basins that the transformation leads to higher density waters. This is the intermediate layer, and this is the deeper layer, again in the same pattern. So the main volumetric distributions show transformation towards, again, higher salinity, warmer waters, but a bit more denser, again, crossing the isopycnals. Of course, this is the overturning circulation. The two main factors controlling the overturning circulation is the preconditioning, salt preconditioning mainly, and of course, the winter heat fluxes. So just by looking at the heat flux trends at the same period, this is only for March, but you can clearly see this dipole here. So the ocean heat loss in winter time is much less, is decreasing in the Western Basin and is increasing in the Eastern Basin. And if you combine that with a bit of decreasing winds here and strong decrease in latent heat flux and sensible heat flux, we get this, we explain why we have this ocean heat loss dipole pattern. So increasing heat loss in the east, mainly in the east, and decreasing in the Western Basin. So if you combine this pattern with the increased stratification in the basin, you get much more favorable favorable conditions for dense water formation in the east with respect to the west. So the Gulf of Lions has this huge anomaly in ocean heat loss, mainly driven by latent heat loss. Just to finish with some concluding remarks, so the excessive salinification of the basin, mainly driven by evaporation, shows that probably sufficient to sustain the intensity of the Mediterranean overturning circulation, especially in the Eastern Basin. And our water mass transformation analysis show significantly saltier, warmer, and slightly denser waters, especially in the intermediate and deep layers. Both these changes are driven by the abrupt, of course, in the Eastern Mediterranean and Western Mediterranean transient events. Very important for the overturning circulation, our analysis shows that the transformation into lighter waters in the upper layer contrasted with transformation into much denser waters in the deep layer. So we have this stratification increase. And when we combine this with a strongly decreasing winter net heat loss in the Western Mediterranean, the Gulf of Lions, this should have probably severe implications for Western Mediterranean deep water formation. So ultimately, the interplay between the changing winter heat loss and the salt preconditioning and warming will define the future status of the overturning circulation and ultimately the Mediterranean climate. So I'll stop here. Thank you very much for listening.
Thank you, Dr. Skleris. Any questions?
Good. Yeah. First of all, congratulations. Very, very, very interesting talk. And it shows us where our sea is moving to. I was impressed that while the surface waters keep getting lighter, the deeper waters keep getting denser. Yeah. So is it a time scale thing, since the stratification is increasing? How do they get?
It's highly no linear process. So what's happening in the intermediate and deeper layers is that during winter time, during the climate transient events, this signal is transported very efficiently to the deep ocean. So this is annual average. So the transformation figures that I showed you. So during summer, of course, we have this highly stratified water. So this reflects two different time scales indeed. So what we observed in the deep ocean is signals that were transported to the deep ocean.
Yes, but these signals took place in the past. I mean, do we expect that they will be? We expect that these will be continuing? I mean, this is since the stratification is increasing.
This shows that. I mean, this is highly non-linear. So this is when calculating the trends, and you have this very strong multi-decadal variability. Some of these trends are actually just the impact of the climatic trends in events that transfer these signals very efficiently to the bottom of the Mediterranean.
So I think we're very happy because we saw a new application of the TS diagram, how the TS properties are moving with time. Thank you very much.
One more question.
I would like to ask you, I saw you show us in a slide the response of the Adriatic, the Aegean, the Gulf of Lions, etc. Can you show this again? I saw in this slide that the response of Adriatic was much less than the other areas that you had presented.
Yes.
And I would like to ask if is this related to the bios and the effect of the bios of the Atlantic waters that are entering into the Adriatic?
Yeah, I mean, I think this again shows that this is the impact of the Eastern Mediterranean transient. So for this period, the impact of the Eastern Mediterranean transient, so this that water mass transformation happened in the Aegean Sea, and much lower proportion in the Adriatic. So in this 45-year trend, much of this trend is explained by the Eastern Mediterranean transit, which increased, hugely increased the water mass transformation rates in the Eastern Basin, the Aegean Sea, and then the whole Eastern Mediterranean. And the contribution of the Adriatic was less in that period.
Thank you very much. So we can move to the next.
Just another final question, Dr. Skleris.
One more question. Very good.
Just, very, very nice presentation, very interesting results. I would like to ask you, generally speaking, do you expect any kind of changes in lateral movement of masses between the Western and the Eastern Mediterranean, considering that there are such changes in different type of, and of course there will be a lot of differences in sub-basins, in communication between sub-basins, but generally can have some kind.
Especially for the intermediate layer, we can clearly see that the Western Mediterranean intermediate waters become more saline following changes in the eastern basin. So some of the signal that we see in the west is clearly came from the east, well, related again to the transient events and the reconstruction of salinity in the water column in the east. But clearly we have much more saltier waters going from east to west, for example, in the intermediate layer.
Thank you. Let's move to the next one. The title is Operational System for Ocean Forecasts over 3 main gulfs of the North Aegean Sea, AGs. It will be presented by Dr. Vasilis Kolovogiannis.
Hello, everyone. So the presentation is going to be about an operational system for ocean forecasts over 3 main gulfs of the North Aegean Sea. I'm presenting on behalf of the research team, the members of which from the Laboratory of Physical and Chemical Oceanography, the Department of Marine Sciences, and Yannis Androulidakis is still with the division of Hydraulics and Environmental Engineering at Aristotle University of Thessaloniki. A couple of slides to set the framework and the motivation behind this work. So in the Northeast Aegean, we have these 3 enclosed basins, the Gulf of Moudros in Limnos, Kaloni and Gera in Lesvos. These coastal environments, they are fragile ecosystems, we know that. Exchange of water masses take place through narrow straits, so renewal time is limited. They are part of the Natura 2000 network, so these areas are protected. At the same time, they are extremely important for the economy of the 2 islands. Numerous activities take place in their vicinity, fisheries, saltworks, navigation, we have small ports, and communities along the coastline, touristic infrastructure, agriculture taking place at their watersheds. So one can understand that they're affected by numerous anthropogenic and environmental pressures. So we need to understand and forecast or project the marine conditions in these areas, especially in the context of the changing climate that is expected to have serious environmental and socioeconomic impacts. So in this context, we have developed high-resolution numerical simulations that operate daily in these areas, in these gulfs, delivering short-term forecasts on the marine conditions, currents, waves, temperature, salinity, surface elevation, etc. And this modeling structure or suite is part of an overall infrastructure, that of the Coastal Environmental Observatory for Northern Aegean, the AEGIS project. In this slide, we see the modeling chain and an overview of the system, starting at the top with the Copernicus Marine Service providing the models with open boundaries, data for their open boundaries. And ICON and OpenWRF models providing atmospheric forcing, wind, velocity and direction, pressure, precipitation, total cloudiness, relative humidity, temperature at the surface. Next, we can see the models that the system is using. SWAN is being used to simulate wave regime at the Gulfs. The regional ocean modeling system is being used to simulate circulation at the central North Aegean. These 2 are coupled, they are interlinked, they run in parallel. And you're going to hear more about this in the upcoming presentation by Yannis Mamoutos. ROMS also provides boundary conditions for the coastal circulation models at the Gulfs, where we use the Delphi3D flow modeling system. So the domains are, we have an overall central North Aegean Sea with a resolution of 2.5 kilometers. There is an embedded grid around Lesvos with 500 kilometers resolution. And then we have the models of the Gulfs. We are using curvilinear grids there So we can have very high resolution to resolve the exchanges at the straits. And here are the forecasts that the system produces. So we can know every day we can have forecasts 2 to 5 days ahead of wave height and direction, the currents, etc. As I said earlier, temperature, salinity, sea surface height, and therefore we can know processes such as transport of mass, renewal conditions, mixing, etc. And at the bottom of the slide here, we have with the dashed lines is a module that we have started building on it. It's the ecological part, let's say, of the infrastructure, so that we can have also at some point we can get forecasts of the biochemical conditions, chlorophyll A, nutrients, dissolved oxygen, etc., with the use of the Delft-WAC model. Here is some information on the machinery we're using, the machines. So if anyone is interested, we can revisit this point at the end. Now let's move to see some results from its case study, starting with Moudros. So here we have, in this scattergram, we have how the model, the simulated sea surface temperature compares with satellite-derived sea surface temperature. This information comes from the Copernicus Marine Service. Data are spatially averaged. Both series are spatially averaged over the Gulf. This diagram refers to the June to December 2023 period. So ideally, we would like to this blue, all the points to lie on the black line here. But despite the small overestimation of high temperatures and underestimation of low temperatures, we think that the metrics indicate that it does quite a good job. I have to stress here that these simulations are free, meaning they do not assimilate any information. And the numbers say that they are working nicely. In figure B, in these 4 panels, we have the horizontal distribution for the surface temperature and salinity, and the velocity fields for the surface and the bottom. These series are temporally averaged over the June-December 23 period. The black lines indicate the prevailing flows across the Gulf's entrance, and this is an example of the information that is starting to build for these areas. And here is an example of how the predictions are delivered to the user, one that accesses the platform. So we have, for example, we can choose to see temperature at the surface, salinity, and the current fields. Here is an example of a vertical distribution of temperature along this section, so that we may know how the column is going to evolve in the next few days, as this was at some point a prediction. Moving on to Kaloni to see some results there too. For the economy of time, I'm going to skip this figure here. Go straight to the examples of forecast products. Again, we can see how surface velocity, for example, is delivered, surface temperature. We have here a vertical distribution of the temperature also. These figures were drawn from the system a few days ago when I prepared this presentation, so they're quite relevant to the conditions that exist right now. So we can see here, for example, how the cooler and denser Aegean water tends to form a a subsurface layer traveling up the Gulf and conditioning exchange. And the warmer and lighter surface water tends to flow in the upper layer. Now moving on to Gera Gulf. Here we can see an example of a detail of the grid that has been applied in order to be able to, for the model to resolve the exchanges. I have to say that the hydrodynamic simulations for Gera Gulf are yet to be incorporated in the system. This is my fault. So it doesn't run operationally right now for Gera, but we can see some examples of hindcast runs. So for instance, here in this panel, we can see for a period of 1 month, with red is the what the model calculates as the tidal signal, and with blue we have the tidal signal that is calculated, that is measured by the tide gauge that is installed here at the Scala Sikudas. And here at these two figures is an interesting event. In figure A, we have for a period of 1 month, this is the February of 2020, we have with the red line is the measured wind speed, and the blue line is the pressure, the atmospheric pressure at the sea level. These are calculated from data by data from the National Observatory of Athens. So this series describe the conditions prevailing at Lesbos, and we can see that at the start of February there is a strong low pressure system affecting the area, and the response of the sea level to this event is captured obviously by the tide gauge and the modeling system. And the model continues to simulate the tidal signal down the line. Okay, so here's a slide showing an example of 3-hourly wave conditions over the 3 areas. Thank you, Yanni. So we have one that visits the platform can plan his everyday activities. He gets information on the wave height and the direction that will prevail within the next 4 days. Okay, so what are the next steps? Obviously, the system is very young. It needs to be. We need to perform assessment on all of the components. Mounting of the repaired CTD to the oceanographic buoy that is deployed in the Kaloni Gulf will restart providing continuous observations that are very crucial for model validation. We need to forecast freshwater discharges, at least in Kaloni Gulf. And this is a task undertaken by the Coastal Morphodynamics Management and Marine Geology Laboratory of the department. We are planning to incorporate all the latest information regarding the bottom quality of these basins. There are very high quality bathymetric data, the bed forms, the vegetation, all these features that determine the bottom roughness, which is a crucial parameter when simulating coastal environments. And as I mentioned earlier, we have started building the biochemical component. Here is an example of how nitrate, phosphate, and chlorophyll A concentrations evolve at the surface of Kaloni Gulf for a period of 2 weeks in in October 2004. This is how the platform looks right now, if one accesses it. So who can benefit from this infrastructure? Obviously, the scientific community, as high-value information on how the coastal system works is produced. It can be used for the training of young scientists, but we don't want it to be just a scientific exercise. We want it to be appealing also to the responsible authorities, supporting them in first-level responding, the design of protection and mitigation measures, and in taking informed decisions. And of course, we want to engage the local communities. We want it to be appealing to them also. With that, I will leave you with some acknowledgements to Mr. Panagiotis Papachiou for enabling the use of the high-performance infrastructure, high-performance computing of the university. And the study was developed in the framework of this project here, Coastal Environment Observatory and Risk Management in Island Regions. So with this, I will leave you. Thank you.
We have time for a quick question.
What do you do about the boundary conditions for the modeling?
There are several models. Okay, there are several domains. Let me go to the first slide. Yeah. Okay. So this one. Okay. There are actually 5 models running. Okay. There is an overall central North Aegean domain, okay, that uses Copernicus Marine Service, Yanni, correct? Yeah, the southern boundary is open, and here we have exchanges with there's a parameterization of exchanges at the Strait of Dardanelles using Mavericks. Okay. Then you have this domain here. Sorry. We have the Lesbos domain, let's say, that uses boundary conditions from the overall ROMS model. It's a nested simulation. And then we have the models running, the Gulf models, let's say, that use boundary conditions from ROMS. So it's a nested simulation, actually. Okay, thank you.
Okay. Oh, very cool. We're out of time.
Sorry about that, but I cannot help it. Can I ask you a question? Why did you use 4 days forecast lead time and not another one? Like say 3 days or...
Okay. Okay, this depends on the source of atmospheric forcing we're using. Okay, there are the ICON model, for example, the atmospheric model ICON supplies 5-day periods, 5 days ahead. Okay. So we can have simulations for these 5 days. The OpenWRF model, atmospheric model, gives only 2 days ahead, but with higher spatial resolution. So it's depending on the atmospheric forcing, you can have 2 or 5 days within that range, let's say. Okay, so the 4 days, the wave modeling, Yanni, is it 5 days? Okay, so if I wrote 4, it was a mistake. 4.5, okay. So it's basically what the atmospheric models can do. Okay,
okay, in the break we can. It's more. Okay, next. Next speaker is Dr. Mars. It's
the title of the presentation is Combined Eulerian and Lagrangian Perspectives on Water Mass Transformation in the Aegean Sea, of consequence for the wider Eastern Mediterranean. The floor is yours.
Thank you very much. Just going to talk about some new work, which has been a little bit inspired by meeting old friends, Vasilis and others, and finding out something about the Mediterranean. From my perspective, this is a learning exercise emerging from other research, which is based obviously on other parts of the world's ocean. So how do I go forwards now? Just hit the button. Do I progress the slide with a...
Maybe your mouse?
Yeah.
Click. The left click.
Which one? Okay, yeah, okay, thank you. Yeah, I'm just going to go through these points. So this is a preliminary analysis of an ocean model homecast using a particle tracking method to focus on one particular phenomenon, which is the outflow from the Dardanelles, which is fresh, easy to spot in the model. And so I like to use a temperature salinity diagram, like we heard a minute ago from Nico. And there are two perspectives here, which is Eulerian, which is looking at a single place or many places, but varying through time, or Lagrangian, which is following the water, which is a natural way to think about our seas and oceans. And now I think there are ways to further, this is a summary slide. I've just realized I've gone right to the end there, haven't I? Sorry about that. Something went wrong. The outline is that I'm going to be tracing these different flows in, first of all, the Atlantic. Then I'm going to mention how those results relate to the changing of the water masses through what's happening at the surface and what's happening underneath the surface through mixing. And I'm briefly mentioning that there's a change going on in the Atlantic, which is probably not so different than what's going on here in the Mediterranean. So then this is all analogous to thinking about how fresh and saline flows interact in the Aegean. And I take a quick look at how we can use that towards understanding the big changes that have happened in the last maybe 30 years. And I'll be using this eddy-rich ocean hindcast and then concluding with some thoughts about where we go. So this is a movie, and I let it just run out. It introduces the idea, which is that we're following here from a cold, fresh place, which is Fram Strait in the North Atlantic, a particle of which there are going to be many, as it goes south and it interacts with the warmer, saltier water. And in these diagrams, which I'll let them run through again, the particle is recording its temperature and salinity and density. And this is telling us that there are some abrupt changes. So you see the jump, which has just happened in the salinity, because this fresh water has just met some salty water and exchanged salinity, basically. So this is mixing. And this is the idea which we developed to understand changes in the North Atlantic. You do that many, many times, and you can construct these sort of composite integral maps of water movement northward and southward. So the southward water is the fresh, and the northward moving water is more saline. By doing this, we're understanding how the flows are interacting in different property space. So that's the temperature and the salinity and then the density. So these orange and blue streamlines are kind of meeting each other, and the waters are blending together as we understand is going on. And this is an exploration of the overturning circulation. Now, I think that we can move this towards a kind of other context, which is going to be the Aegean. The last thing I show here is some temperature salinity space analysis of changes over 100 years in climate model projections, which are illustrating how temperature and salinity change kind of together. And what's going on here is complicated, so I won't go into that detail, because I want to talk more about the analogy with the Aegean. So it's about understanding the change in temperature salinity space in the big Atlantic. But this is my attempt to make the analogy. On the left-hand side is the work we did already, which concerns the interaction between the fresh Arctic and the more saline Atlantic. So you see this kind of colors are supposed to represent the meeting of the saline and the fresh waters, and a kind of a vertical mixing south of what we call Denmark Strait. And that's kind of interpreted also in a temperature salinity space here by moving from the bottom left to the top right of the temperature salinity diagram. So that's going from a kind of cool, fresh to a warm, salty water mass. And then on the right is my understanding, which I'm learning, of the interplay between the Black Sea water and the Mediterranean water. So moving from the Dardanelles to the left, we have this amazingly fresh water, which meets this really salty water, and it's more like a horizontal mixing, I guess. And again, on the bottom, on the right-hand side is another TS illustration of the much more dramatic transition from cool, fresh to warm, salty. So I wrote in the boxes that although these are very different in scale and different latitudes, and even the orientation of the mixing, conceptually in TS space, it's sort of the same thing. It's just you have to think in a common way. So there's an analogous sense of water mass transformation in temperature salinity space. So as the submitted abstract, this was kind of very recently proposed idea, I guess. We would like to use available data sets, ideally the ROMs at the bottom here, which is from Yanis and others, which will be a really great opportunity for us. So I started off with a quick look at high resolution eddy-rich global hindcast. I looked specifically into the East Mediterranean. I could also in the second row there look at a very high resolution climate projection to 2050, which is something which is easily available to us. And then on the far right column are the different packages that we can use to follow water. Ariane, TrackMass, these are ways we can do additional calculations with these big models. I'll just show you quickly then, I cut out, this is the Mediterranean every month in 2021 from the hindcast with this ocean model. So obviously that's all of the East Mediterranean temperature at the surface, salinity at the surface, currents at the surface. It looks okay. I'm not going to claim that this has all been validated by me, but this has been run by colleagues in the UK. Then we cut out a little bit more the smaller area here of the Aegean with a little box there. Now this is the sea surface salinity. I don't know whether the very fresh signal from the Dardanelles is entirely realistic, but there's a huge contrast of salinity, which I'm pretty excited about because you don't normally see such a big contrast in such a small region like this, of course. Everybody here will be familiar with that. Then what I do is take a look into that little box and just gather together all of the T and the S. This is the Eulerian monthly 2021, just this example. Movement, if you were to play a movie here, you'd see this kind of swarm of TS locations kind of going up and down with a seasonal cycle. So I just emphasized, obviously we have a seasonal cycle in temperature, a little bit less in salinity. This is my sort of Eulerian perspective on the TS behavior in this little region. Now this is the attempt to, the first test trajectories following from the highlighted mini section with a very artificially widened Dardanelles and an extremely widened Bosphorus, by the way. So this is modeling, not perfect, but this is what the very high resolution global model does to the Aegean region. So nevertheless, we have a nice little flow of fresh water there. And all these black sort of spaghetti lines are a few particles. I actually use a lot more particles to do the calculations later on. So I'm following the first two levels of 11 locations across a little transect, and I'm going forwards for 6 months, and I'm saving positions every day. And this is using what's called Ariane, which is a particle tracking software you can use with ROMs or NEMO models. And then I've increased the number of particles and done some statistics with a bit more analysis here of the character of the flow. As you go away from the Dardanelles, I'm looking at the fraction in the top left, and then top right is the average travel time in 1/10 degree boxes. And that travel time increases from 0 to 180 days as you go further away from the source of the particles. So it goes from blue color, which is 0 days, to the orange color, which is 180 days, as you go around Greece, I guess into the Adriatic. And then the water mass, which I'm interested in, is warming, which is meaning that the mean temperature in the bottom left is gradually increasing from sort of mid-teens to mid-20s, and then the salinity is also increasing substantially through the 30s. And this is done with like not averaging, but sampling the entire seasonal cycle. So it's a very averaged looking picture. This is for 93, 94, right? So it chose this time because we know there was a transient, and there's a few points to make there, which is that particles sometimes concentrate in certain places. They maybe take about 50 days to transit the Aegean, and we have this kind of increasing TNS. And I did the same thing for a more recent year to look at the difference. And there are a few differences immediately to spot, which is that particles concentrate in different places as a kind of recirculations going on. More particles reach the eastern Aegean. Particles take a bit longer to go through the Aegean. And there's a bit of a change in the pattern of the increasing TNS. So what I want to finally do is to show you that you can take all that data and just throw it into a TS diagram like this. And now I'm plotting these dots. Each is a particle location with its TNS and the density lines in the background. But the color of the dot is the number of days since it left the Dardanelles. So the pink color is kind of 0. That's the beginning of the journey. And then they end up being this kind of red color. And there's a big arrow indicating that the sort of timing drifts the points towards the higher salinity, a little bit towards higher temperature as well. So the axes say SST, SSS, it should be TNS. These can be going down as well as moving at the surface, but they mostly stay near the surface. And so I did this quick analysis also for the later time. And if you flick between these two pictures, you see that the hoop of the higher density, higher salinity particles is shifting a little bit to higher salinity, and even maybe about the same density. So this is consistent with what Nico said earlier about the increasing salinity. I'll just finish with a summary, which I already started with, which is to say that we did this preliminary analysis with the hindcast of just tracking the fresh outflow because it's a big signal in the model. You can look at the TS analysis in Eulerian or Lagrangian way. I think there's a lot of scope to go a lot further to separate the effects of surface fluxes and interior mixing on the water mass transformation in this kind of framework. I think this is a novel way to take a relook or revisit of transient changes in the region. And we would ideally do this with the much higher resolution ROMs. So I feel like we have tools to bring to examine what's been going on in the Mediterranean in the last 30 years and maybe keep looking as it changes into the future. Thank you.
Thank you very much. We have time for two quick questions.
Very nice, Dr. Mars, Bob. It's very nice having you here. But I should stick to the question, I guess, not say more about that.
I'll get to the answer as well, hopefully.
So the question is, I noticed that you refrained from using a fully Lagrangian module. You used the surface Lagrangian. I mean, so, or you at least kept it within the first 2 layers of the model?
Yeah, but then the particles can go anywhere. They can go down. They can come back up. They can move 3-dimensionally.
Okay, that was my question. Yeah. Thank you very much.
Not a problem. Okay.
Any other?
Can we use such models for future predictions to predict what
Yeah, with a climate model, which has a nice ocean, we can do the same experiment with particles in the future ocean to see how the future Aegean is behaving. Yeah.
Okay, thank you.
That's what we plan to do, I think.
Okay, thank you very much.
Thank you.
Okay. Yeah. Last question.
Sorry, Dr. I'm sorry, Dr. Mark. Just it's a question which is in the periphery of what you're saying. It was coming back to the North Atlantic, in the Labrador Sea, and then the Fram and Denmark Straits. And it's I was always wondering. Now the new information we have for the thawing of East Greenland ice, how is going to affect this kind of changes and this kind of overturning in that area?
Yeah, so you're talking about the increase mass flux from Greenland into the subpolar gyre. That's gradual. It's not dominant at the moment, so it's a small few percent of the freshwater budget. Maybe in the next decades it will be more, and then it can gradually accumulate, and so the freshwater can eventually stop the overturning, and we have a big problem.
So you don't expect any changes over the strait, over the Denmark Strait, to affect this kind of.
Well, I mean, if the fresh water is accumulating at the surface, it's gonna
go down.
Gradually stall the overturning, but the East Greenland current will continue to leave the Arctic because of other reasons, right? So, but whether the water overturns or not is a separate issue, and that may eventually stop if there's too much fresh water.
Okay, thank you.
Okay, let's move to the next one. The title of the next presentation is a method to assess the rate of dilution of marine scrubber water from water quality model results, application in the Saronikos Gulf, Eastern Mediterranean. It will be presented by Professor Zervakis.
Can I do it, miss? Because I want to go to my first page, please. And what's it all? Can I? No, there's a presentation mode, which is slide by slide, not by. Let me see if it works. Oh. Let me take the control for a moment. Υπάρχει presentation mode που βλέπεις τα slide by slide. Okay, today we're presenting methods. So I'm going to change a little bit the overall scope, I guess, of these presentations, because I'm going to turn to a more practical problem than just physical. I mean, right now we'll go to the limits between physical oceanography and maybe engineering, because we want to address a pollution problem that is related to the use of marine scrubbers in the marine environment. As we all know, shipping is a significant contributor to pollution, both in local scales as well as global scales. And due to that, the International Maritime Organization has introduced new requirements about air emissions for maritime shipping. And these requirements are that there should be what is called the cap in the emissions, a global limit of 0.5%, and there are also some emission control areas like the Baltic and the North Sea, where the limit is 0.1% since 2015. So the obvious solution is to use different ways of moving the ships, let's say, alternative fuels, or low sulfur fuels. But another solution that has been adopted by the maritime industry is the use of marine scrubbers. Now, marine scrubbers spray water in the pipes, thus arresting the pollutants and transferring essentially to the sea. So this way we kind of move the pollution from the air into the top layers of the marine environment. This is what is called the use of open loop scrubbers, and it can be a significant threat to the marine environment, as we will show later. So there was this big EU project, Horizon project, that was related to this question. It was examining the fate of the various scrubbers to the marine environment and the impact they may have. And as a response, as our department's response to the requirements of this problem, we developed a system of models that were necessary in order to assess what the fate of each pollutant is on the marine environment and obtain eventually spatiotemporal maps of the pollutants on Saronikos Gulf, which was our case study. So you see that there was a model that quantified the effluent, the liquid effluent of ships to the marine environment. And then there was a circulation model and a wave model that estimated the circulation in the Saronikos Gulf. All these were contributed to the water quality model that we used, together with lateral pollutant fluxes. This was a big part of the project because when you work in the Saronikos Bay, you have many different lateral sources of pollution. And also environmental boundary conditions had to be introduced. So all these were introduced to the water quality model. There is a presentation by Caterina Masiotti about that in this conference, a much more detailed one. And then this is a typical output of a water quality model, spatiotemporal maps of pollutants along in the area. So this is a pretty a concentrated diagram of the approach, of the modeling approach of the marine environment. So you see here the map of the fluxes of pollutants by the ships along the shipping lane of as it is called the Piraeus approach. The hydrodynamic, and this is the domain that we had of the hydrodynamic model, and the same domain was the base for the water quality model. And then this is the example of the maps that we got of the distribution of the different pollutants, whether they were heavy metals, as in this case, or aromatic hydrocarbons. Thank you. And this is a typical example of the results of the model using all these forcings. And we see that the pollution is very high, especially along coastal areas, which means that the lateral pollution sources dominate the distribution of pollutants throughout the Salonikos Gulf. You see that the most pollutant places are LFC's Gulf and the region of the refineries here near the Corythus Canal. So we had some quite useful results regarding the distribution of pollutants in the Salonikos Gulf. However, we needed to assess the impact of scrubbers. That does not solve the problem. So we had to relate to the output of several ecotoxicological studies that took place, which were actually laboratory experiments. When you do this kind of studies, they follow what is called the wet approach, which is they examine the toxicity of the whole effluent and not each compound one by one on the marine environment. Because when you have the effluent, the marine scrubber effluent, it's a mixture of toxic compounds. So the impact on an organism of a mixture could be quite different than the impact of one per one compound by itself. So the wet approach was designed and devised in such a way so as to assess the impact of the whole effluent on the marine environment. And this constituted of diluting at several dilution rates the marine scrubber water, and then examining the impact on little, let's say, the parts of the ecosystem, let's say, or on several organisms. So several contributors to the project did various experiments. So several concentration thresholds were measured, like the lowest observed effect concentrations. So these were the concentrations that an observed an impact was recorded based on the lowest concentration achieved on the experiment. This is not necessarily the absolute lowest concentration that could have an impact. As you understand, it is based on the impact on the design of the experiments and the lowest concentration that you can reach. So another method had been applied, and and has been published, which leads to the predicted no effect concentration based on the above results. So you see here that the lowest observed effect concentrations range between 10 to the -4 and 10 to the -5. So, which means we take the pure marine scrubber as it comes in the ship and dilute it 100 to 10,000 times, and then 100,000 times. And then there is the predicted no effect concentration, which is 2 times 10 to the -8, which is a very, very low concentration indeed. But that is the actual concentration that we expect absolutely no impact, no toxic impact on the marine environment. So we had to devise a way to combine the results of these ecotoxicological results, the results of these experiments, with our results, which only contained compound by compound concentration maps. So the way we did it, first of all, since we knew that the lateral pollutant fluxes was very strong, were very dominant, we had to cancel this part of the equation. So we run again all the models without taking into consideration, for example, the oil refineries, the urban pollution from Citalia, the work in the shipyards around Saronikos. So we only forced all the models with the steam model, which is the marine scrubber waters. Now, the crucial point on which this method was based was that we assumed that the marine scrubber, once it falls into the marine environment, there's the first stage of the decay of concentration, which is turbulent. When the mixing is turbulent, the decay is a fully mechanical process. And during that process, the relative ratios of concentrations between the various compounds remain the same. It's something very similar to the Marceppe principle that you all know applies on the salinity of the water. So through the first stage of the outflow of the marine effluent of the marine environment, we expect that the relative ratios of the compounds should be constant. After that stage, the different biochemical properties of every compound take over. For example, the hydrocarbons, some part will evaporate, some part will start to sink. Different heavy metals behave in different ways. So the relative ratios of the concentrations will start to, well, so I have to accelerate. Yeah. So we devised, first of all, a numerical way to assess the concentration, which is based on the dilution rate, which is based on the concentration of each compound. And then the criterion for the extent of the turbulent stage was the variance of the different concentrations of the different compounds. Now, then it was pretty simple. This is the result of the concentrations of each and every compound along the shipping lane. And here is the scattergram between the variance that I said previously determines which is the stage at which turbulent mixing is dominant. And this is, you see, throughout turbulent mixing, the variance remains low and constant. And then, after some time and at higher dilution rates, the variance starts to rise, which means that the biochemical processes take over. So you don't have anymore a single mixture. So you cannot apply your results to compare your results to the results of the ecotoxicological experiments. So these are the typical results. So we have, we find that actually we have high concentrations, quite high concentrations relative both to the lowest observed effect concentration, as well as even to the no effect concentration rates. And eventually we estimated the percentage of the time that the water exceeds the lowest, either lowest observed effect or no effect concentration. And we actually find quite high time, high percentage of time that pollution exists. So this work shows that marine scrubbers could have a very strong impact on the marine environment, and even at quite large distances from the shipping lane, which distance is of the order of 10 kilometers. I guess I should stop here. I'm not gonna say much else. Thank you very much.
Thank you very much. Two questions, quick ones. maybe during the break so we can move. Okay. Yes. Oh, yeah. I'm sorry. I didn't see you.
He has the last question.
Yeah. Okay. Okay.
Apart from the fact that, of course, thank you very much for your presentation. I know a little bit about the MERS project, but apart from the fact that it seems that things are not the way they look, or basically they make this kind of developments without having tested how much is going to be the effect on the environment, and if it is better to be in the air or better to be in the sea.
I cannot hear very well. If
I will
please.
take that out.
Thank you.
I say, apart from the fact that the scrubbers, from what you're saying there, they seem to have more influence on the environment than expected before. So basically, we have to consider what we are doing with these very expensive modifications on the boats. It just, while you were talking, I was thinking, do you think that if you go to an area that there is a strong tidal signal, that will change, and towards what direction will change it? Just.
Well, I don't think that the tidal signal will change that much the results, because the tidal signal just brings backward and forward or remixes the water around the same area. And we saw that actually the same methodology is being used at a larger scale, at a regional scale, by a colleague in eMERGE. And I think he has similar results also for the for the North Sea, where the tidal signal is strong. Yeah.
Okay, thank you very much. Okay.
I'm not very familiar with this technique with scrubbers, but I'm wondering what is happening when they want to get rid of all this. Where do they throw away the?
Well, yes, that was a good question. That is a good question because I did not describe very well the scrubber technology. There are two kinds of scrubbers: the open loop, which actually take all this effluent and throw it immediately in the surface water, and there's the closed loop, where there is a filter and a tank within the ship, and they keep all the fluxes there, and they recirculate the water, all the compounds there, and they recirculate the water, and then when they reach a port, they store it someplace. I don't know really where. The problem, I guess, is that the second approach, which is much more ecologically friendly, I suppose it's very expensive because at least most of the ship owners choose the first one.
Thank you.
Okay, let's move to the last one. Thank you, Professor Servakis. It's a Central North Aegean forecast system, a high-resolution forecasting system for Central North Aegean system description and validation, presented by Dr. Mamoutos.
No, they fix it on the... Greetings to all. I'll try to be quick and not tire you more. So here we're presenting a new high-resolution forecasting system for the North Central Aegean Sea. There is a small system description and some validation. The group members are the same with the presentation that previously done by Dr. Vasilis Kolovogiannis. As you can see, only Yannis has affiliation until now with Aristotle University of Thessaloniki. All others are here in the Laboratory of Physical and Chemical Oceanography on our department. So let's start. Can I have a pointer somehow? Anyway. So, in this panel, we see the extent of the Aegean Sea and the, let's say, neighboring basins. As you can see here is the Aegean Sea. The black box denotes the center-north Aegean Sea. Here is the domain of the model. We actually have here with the green and yellow color the positions of two Argo floats that actually now providing us with data. Why we need a forecast system for the North and Central Aegean Sea? We have a complex coastline bathymetry, different water types from other subbasins of the Eastern Mediterranean, let's say, meet in the area. Black Sea water from the Dardanelles Strait and the Levantine water from the south part of the Aegean here. It is home to a large fishing fleet. It's an area with a heavy merchant ship traffic. And of course, we have, because of these conditions, we have a high possibility for a marine accident. Now, the forecasting system has two components. One component, an ocean component and a wave component. We produce 5-day forecast every day at 04 UTC time. There are every day available here in this site, in this address. So a very short description about the components. The ocean component is ROMS model version 3.7. We use a daily data simulation scheme using the native capability of the model. It's an overlap technique. Anyone has a question about overlap, I will explain. We use all the available information from all platforms, satellite products, such as sea surface temperature, sea level anomaly, and in situ platforms, mainly the two Argo, now 3 Argo floats. The boundary conditions for the southern part here comes from a basin scale Mediterranean Sea model from CMEM's service, the MEDMFC. The atmospheric forcing is from hexadral icon meteorological model that is data is freely provided from the German Meteorological Service. The assimilation window is 3 days, and we forecast for 5 days. The outputs are 3D fields such as temperature, salinity, velocity components, and sea level. So the wave component is based on the SWAN version 41.4, no data simulation. The boundary conditions, it's 2D relative spectra, comes from an in-house developed model of the Aegean Sea. The wind forcing here again is from ICON, and we give a 5-day forecast with these, let's say, these variables, which is significant wave height, mean wave direction, swell, mean period, and peak period. So this is a short description of the operational product chain that runs every day. So as you can see here, the first step is to download the icon data from DWD at 2145 UTC. And then we have in 2 different machines, the operational chain, what is happening until all models are finished, and then all the forecasts are available in 4 UTC at this page here. So this is example, as you can see, it's almost 15 days ago, but this is the time when I had to send the presentation. As you can see, here is the output for the Central North Aegean. This is the surface salinity, this is the surface temperature, and this is the mean wave direction with the significant height. Also here we have, as Dr. Kolovogiannis said earlier, we have a local model for the Aegean, for the Lesbos region with a 500 meters resolution, and we produce the same fields. Here is the sea surface salinity, sea surface temperature, and significant wave height and direction. Now, of course, when we run, when we do operational forecasts, and especially when we use data simulation techniques, we need to see the validity of the model results. So, as you can see here, this is the latest version of our operational system. This validation covers the period from July 2023 until the end of February 2024. So you can see here is the mean salinity profile, the mean temperature profile. The shaded area is the, let's say, conference interval for each depth. It's 1 standard deviation. The black line denotes the observations. It's a temporal spatial mean from all Argo profiles. The red line is the analysis, and blue, yellow, and green line is forecast day. The first forecast day, the second forecast day, and the third forecast day. This is the leading hours, analysis plus 24 hours, 48 hours, and 72 hours. So as you can see here, in terms of mean profiles for temperature and salinity, we have a good match almost in the full extent of the vertical distribution of the seawater. Here for the salt RMSC and temp RMSC, we have for analysis and almost for all forecast days, we have some discrepancies, especially in the upper, on the surface layer and the upper intermediate layer, which is more or less common in the for operational models, especially for this region. It has to do with Dardanelles parameterization. Again, for the setup, for let's say, the input that we use, and for the complexity of the region, the model reproduces the variability of these fields, and more or less the error in terms of bias and RMSE are in, let's say, in the magnitude that we expect for its resolution, horizontal and vertical. So here, it's the same thing, showing the salt bias and the salt and temperature bias and RMSC per cycle. Again, the colors are the same as the previous figures. The red line is for the analysis. The analysis, let me here to clarify, it's the outcome of the assimilation. So it's when we use all the data to correct our model and produce the optimal initial condition. And the other colors are for forecast day 1, 2, and 3. As you can see here, when we started the initialization, when we started the new version of this operating system, we have higher, in case of salinity, higher bias and also highest RMSC. But gradually, as the model evolves and assimilate more and more data, we now have, let's say, a very stable overestimating in salinity. And additionally, we have a slightly underestimation in temp bias. The good thing is that the forecast skill is good because there is no big differences between the forecast days error. So here is a validation using Sentinel-3B data. This satellite, we use it as an independent source of information about sea level anomaly. And here we have taken all the tracks from the swath of this satellite and make bins of 10 kilometers and calculated the correlation coefficient, the RMSC and the bias in terms of sea level anomaly. For the first column here is for the analysis, and then forecast day 1, forecast day 2, forecast day 3. As we can see, in most of the cases, the analysis product and the analysis outcome is better and much more precise comparing to the forecast. But the good thing for the 3 days forecast is that more or less we have the same error magnitude for the sea level anomaly. And this is crucial because the sea level implicitly corrects the circulation of the model. And having small errors, or let's say acceptable errors in sea level anomaly and in general in sea surface height, we actually correct the outflow of the Dardanelles because we use a climatological approach because we don't have at the moment real-time data. So here is the validation for the wave component of the model, again for the Central North Aegean, again from using in these panels here, using 3 different satellites, SWOT, newly introduced satellite with a big band, Sentinel-6A, Altica, Haiyang 2, and this is for day 1, day 2, day 3. Here's a scatter. We see the correlation coefficient between the observed and the model significant wave height, and here we have a time series for RMSC bias and scatter index for each of the overall metrics for each satellite concerning the wave component. Now, okay, the validation is okay. Let's say it's acceptable. But in real time, in real life, can this model predict something? Yes. The first, let's say, example is the last summer heat wave. Here you can see the verified forecast from the model, the operational model. The SST, this is the surface, sea surface temperature, and this is the recorded sea surface temperature for the 24th of July 2023. It's a level 3 product, means that all the available satellite tracks are co-located and then super-collated to produce one map. As you can see here, the model predictability is more than good. A second example for a combined analysis forecast, mean the analysis we run every day to produce the initial conditions for our forecast, and the forecast is has to do with a deep water formation event, small one, not compared to the Eastern Mediterranean Transient, of course, or previous year deep water formation events. During the winter of 2022, it's also a published paper, work from Potiris et al. 2024. So as here we see the depth, the salinity, and the temperature of the isosurface of the isosurface with potential temperature of 29.1 before the deep water formation event. This is the first 15 days of December 2021, and here is after the formation event, or after the formation event, the last 15 days of April 2022. This data was, let me say that this result was verified by the by the very little amount of observation we have from the Argo floats, but again, it's still valid. So, some concluded remarks, and I'm finishing quickly. The modeling effort to simulate, let's say, an area such the North and Central Aegean Sea with unique and distinct features for both hydrodynamic and wave characteristics is for our group an ongoing and non-trivial and demanding task. The results indicate that our system is capable of producing high quality and mostly valid analysis and forecast, and feeds high resolution coastal application with boundary and initial conditions. Again, here, this must be mentioned because the biggest bottleneck of our system, especially for the ocean component, is the use of climatological values for the outflow of the Black Sea waters into the North Aegean. At the moment, we are continuing the development of the model, and the idea is to use, let's say, data from a global scale model. Now, the only available through CMEMS GLORIS from Mercator, and let's say to adjust the fields or the output of the GLORIS for our case, using climatological assimilation technique in order to adjust the surface, the sea level, and the surface salinity fields. Another crucial, according to my point of view, because I'm mainly involved with the assimilation processes, to go from a strong constraint approach to a weak constraint, but this requires to estimate the model error rate, and this is a very not so easy and non-trivial, let's say, procedure. But in order to, if actually we're able to go to a weak constraint, now we are introducing a bias correction scheme using a relaxation technique during the analysis phase. We are at the moment, let's say, to check the stability of the solution and how balanced are the solution. And last but not least, I hope, the wave components, we are now running sensitivity tests with different physical setups for wave growth and decay, wind drag and friction. But most importantly, we are in, let's say, in process, run pre-operationally, fully coupled the region. Fully coupled, I mean the ocean and the wave component. And our intention is in the next 2 or 3 months, let's say after summer, the model to be fully coupled, so the wave and the ocean component run online coupled. So many thanks for your attention. I hope.
Thank you, Yanni. That was very good. Do we have any questions from the audience?
I wonder if your domain model include the Marmara Sea. Does your model include the Marmara Sea?
How I'm... If it's included. No. We have eastern boundary in, let me show you. No. Unfortunately, we're not including Marmara Sea. We have an eastern boundary here, and we're prescribing the flow using Vladimir Madrit's data, hematological data.
Yes, because my second question was, what's your plan in order to improve this?
The improvement has to do with taking boundary from global Mercator and try to correct or adjust it here, let's say, in some using the idea is to do an climatological assimilation to construct a climatology for all this region from the Mercator dataset in order to, let's say, relax the boundary here when using the Madrit's time series. It's a bit complex, but I can explain outside if you want.
Another question from a student of our department, Marina.
Hi. First of all, I would like to say congratulations. Your work and your colleagues' work is really inspiring. My question is about the validation of the model. I cannot comprehend how that happens using Sentinel-3B data, right? My thought process is that the orbital cycle of the Sentinel-3B is over 20 days, if I'm not mistaken. And I cannot understand how the validation happens.
Okay. So we take the track of Sentinel-B, the Sentinel-3B swath, whenever, let's say, cover an area of this domain. And we then we construct bins. We make spatial binning from the swaths with the model to have, not bins. First of all, we collocate the swath data in our grid, and then we construct bins, and then using these lines, these tracks, we interpolate in time because we don't have the exact same times, the satellite and the model. And then we calculate the RMS, see the bias and the correlation coefficient. It's, let's say, a loop.
Okay, it's a technical issue.
It's a technical issue.
If you are not satisfied, you can discuss it later, Marina.
I would like to say one more comment about the presentation. It's although it covers a part of the Mediterranean Sea that is small, and there are operational models, many of them that they cover Aegean. This is an important step because it focuses in an area where we have the exchange with the Black Sea. And most of the approaches until now, in my point of view, this approach is state of the art inclusion of this exchange, and it can be, of course, can be improved.
Always there is improvement.
Other questions? It is the last talk of this session, so do you want to say something more, anyone? If not, yes, let's go out for a coffee break. Thank you.
Thank you.
Ευχαριστώ, Γιάννη. Εσύ κάτι μπορούσες να δεις. Εγώ από εκεί δεν μπορούσα να δω τίποτα, ούτε από το πλάι.
Dear delegates, good evening. A warm welcome to this afternoon session, which is about aquaculture. We have 3 presentations. So let's invite the first speaker, who is Apostolidi, Miss Apostolidi. It's a study entitled Development of qPCR Assay for the Detection and Quantification of Tenacibaculum maritimum.
Good afternoon. My name is Apostolidi Evangelia, and I'm here to present you the development of a qPCR assay for the detection and quantification of Tenacibaculum maritimum. I come from the Veterinary Research Institute of Thessaloniki. Regarding the pathogen, is a gram-negative filamentous bacteria of the Flavobacteriaceae family. It affects a large range of species with commercial value, such as sea bass and sea bream. As you can see in the photos, it produces distinctive gross lesions on the external body of fish, such as mouth erosion, frayed fins, ulcers, skin necrosis, and tail rots. It results in substantial losses across the global aquaculture sector, including, of course, the Mediterranean area. The diagnosis is based on culture-dependent techniques. However, the isolation is not always successful, and definite diagnosis requires colony isolation on specific media, such as Flexibacter maritimum medium, FMM, or media supplemented with antibiotics. There are available PCR tests. However, they have limited or not at all capacity in quantifying the pathogenic bacterial load in infected fish or in environmental samples. Therefore, the aim of the present study was the development of a novel, rapid molecular method using real-time PCR with the use of a Tachman probe for the specific detection as well as quantification of Tenacibaculum maritimum. For this purpose, we used the gene bank databases and online available softwares in order to choose the most specific gene area. We were between 6 other areas, but finally we selected the translation initiation factor IF2 of INF-B gene. Therefore, we designed a primer pair along with a Tachman probe, and the method amplifies part of that region that is 161 base pair long. The reaction was performed in a final volume of 25 microliters containing kappa probe fast qPCR mix, 300 nanomi of each primer and probe, and approximately 20 nanograms of bacterial DNA. The thermocycling conditions were optimized, and you can see them in the slide, and the analysis was performed using CFX Manager software. As controls, we used non-template controls in duplicate in its run and negative control samples that were originating from fish pathogenic and non-pathogenic bacterial strains that were previously identified by DNA sequence. This regards the species Vibrio, Staphylococcus, Photobacterium damsellae, Escherichia coli, Pseudomonas, and Lactococcus garveae. As positive control, we obtained a DSM reference strain. We started with the cultivation of the reference strain in selective media FMM, and then we quantified it using the standard plate count method. The quantified bacterial culture was then used for the extraction of quantified bacterial DNA. The final concentration was approximately 10 to the power of 7 copies per microliter. In order to create the standard curve for the quantification and to estimate, therefore, the amplification efficiency, the dynamic range of quantification, and, of course, the detection limit of the method, we used the serial 10-fold dilutions of the aforementioned quantified DNA extract. The standard curve was performed at least 3 times. The PCR products of the method were also sequenced by an ABI genetic analyzer for the evaluation of the method's specificity. As you can see, the method showed a very high sensitivity of 1 gene copy. That means 1 bacterial cell of Tenocybaculum maritimum. The method showed a linear range of quantification over a 6 log 10 range and a high amplification efficiency of 98.3%. The specificity was confirmed by the absence of amplification products, either on non-template controls or in the negative control samples we said in the material and methods. The PCR products that were sequenced showed a very high homology, and the sequence has been already submitted to GenBank. So why is this method useful? Recent studies show that tenacibaculosis is one of the most important bacterial diseases along with vibriosis, especially for European sea bass, leading to severe mortalities. In addition, since the common treatment strategy is based upon the use of antibiotics, that can lead to potential presence of antibiotic residues, antibiotic resistant, and environmental pollution. A tool of early diagnosis can help the early treatment of the disease and therefore have a positive economic impact for the fish farm industries. Moreover, nowadays with the climate change, heightens the demand for the development of early diagnostic tools and management strategies. In conclusion, the advantages of the developed method when compared to traditional microbiological methods is its speed and reliability. When compared to the gel-based PCR assays, the higher sensitivity and specificity, and when compared to other available real-time PCRs, it provides the benefit of quantification of the pathogenic bacterial load, either in infected fish or in environmental samples. The gene area we targeted of INF-B gene provides superior sensitivity of 1 copy per microliter whereas the other two available real-time PCR methods that targeted the 16S rRNA gene of the pathogen provide a sensitivity of 4.8 copies per microliter and 2.22 copies per microliter. Its future perspective is its application of qPCR in environmental DNA samples. And I could say we have already started doing that with really good results. I would like to thank you for your attendance. And I have to say, this was funded by the within the framework of the program Prognosis, Correlation of Abiotic and Biotic Factors for the development of a disease prognosis model in fish farming units, funded by the Fisheries and Maritime Operational Program. Thank you again.
Thank you very much for this very interesting presentation. I would like to hear some questions from the audience, please.
Please don't be shy.
Oh, yeah.
Make a question, please.
No, okay. Well, I was wondering, what is the cost for each sample in order to test the cost, economic cost, in order to test a sample?
In order to test, what is the cost?
Cost in order to test a sample.
Yes, it's the DNA extraction. Of course, it's around, it depends on what scale you do that.
What per sample?
Yes, per sample, it's around, let's say, €6 the DNA extra, 12 euros, I would say. 10 to 12 euros.
Okay.
It depends. If you are research, if you do it on research, or it's the common practice of the laboratory.
Let's say that you have a laboratory and you do diagnostic services. Yes. So what would be the cost?
I think around between, I would say a range of 8 to 12.
Okay, this is a very good price. I think so. I thought that it would be higher.
Also, its application on the environmental DNA samples. That
Yeah.
means from the water around the farm.
Yes.
The fish farms. So
Okay.
you can predict, since you have
yes,
such
this
high
is
sensitivity,
very important.
You can predict.
Okay, for future application, this is very important. And concerning the eDNA you just mentioned, do you know how many samples do you need for the eDNA in order to have a reliable result?
We tested some areas.
Okay.
We had at least a triplicate in the start of the farm, of the fish farm, in the end, and also you always need a control point.
Yes, of course. Okay, so okay with me. Any more questions from the audience? No. So thank you very much.
You are welcome. Thank you.
So we can go on with the next presenter, who is Mr. Mitsopoulos. Yep, hello. The presentation is entitled Starvation and Refeeding of European Sea Bass and Baltic Brown, co-cultured with glasswort in a polyculture aquaponic system.
Μία Άγγλο μαζού. Τις διαφάνειες αλλάζω μόνος μου. Όχι, ευχαριστώ. Good evening. My name is Ioannis Mitsopoulos, and I am a PhD candidate of the University of Thessaly. My PhD thesis is about multi-trophic aquaponics system with brackish water. Today, I will present my study with topic starvation and refeeding of European sea bass and Baltic prawn co-culture with glasswort in a polyculture aquaponic system. This study aims to evaluate the effect of starvation and refeeding on the growth and survival of Baltic prawn and European sea bass in a polyculture aquaponic system with brackish water when co-cultured with glasswort. And what is aquaponics? Aquaponics is a sustainable method of food production that combines aquaculture with hydroponic cultivation. Aquaponics is consistent and eco-friendly and innovative production system holding significant ecological importance due to its minimized environmental impact and controlled water usage. The impact of climate change poses new challenges in the agriculture sector, emphasize the necessity of integrated agriculture and aquaculture into alternative forms such as aquaponics. This is why we need to maximize the output of aquaculture systems. For this purpose, the self-sufficient integrated multitrophic aquaponic system aims to drastically reduce nitrogen byproducts dissolved in water produced by aquatic organisms, thus decrease pollution. In general, polyculture in aquaponics is characterized as an activity that combines ecology and sustainable development, ensuring environmental, social, and economic benefits. This present slide shows a number of researchers who have studied multitrophic aquaponic system, being freshwater, like Alias in 2022 with Herochromis niloticus, Leptobarbus noeveni, and Barbonimus goniotus for aquatic plants, and Osimum basilicum for plants, or brackish water, like Waller et al. 2015 with the Kentrarchus labrec and Sparus aurata for aquatic organisms, and Tripolimum panoticum, Plantago coronopus, and Salicornia dolithostachia for plants. In this study, the feeding protocol used was subject to the compensatory growth technique, process characterized by an increased growth after a prolonged period of starvation. The growth of fish previously deprived of food is similar to that of fish fed on a daily basis throughout the period. Feeding withdrawal periods can also be used to improve the quality of fish, avoid excessive lipid accumulation, and reduce food and labor costs and minimize any feeding errors. Within the3 body, and specifically in aquarium of the fish, we've placed 135 European sea bass juveniles with an average body weight of 3.39 grams and average body length of 7.02 centimeters. We're also placed in 3 separate aquariums with cages, 27 Baltic prawns with an average body weight of 1.21 grams and average body length of 3.34 centimeters. In the hydroponic parts of every system, we've placed 8 glasswort or Salicornia europaea plants. The fish were fed with 3 different frequencies. Specifically, System A was fed daily, System B was fed 3 days and week and 4 days of fasting, and the third system was fed for 7 days and fasting for 7 days. The feed was commercial with 45.75% of crude protein and 15.3% of lipids. As we can see in this slide, in the present slide, the polyculture system that we use in the experiment. The system separates in 4 parts: the NFT hydroponic unit, the number 2, the fish tank, the number 4, the pro-rearing tank, the number 5, and the sump filter. The water flow was designed to flow by gravity from the hydroponic cultivation NFT, the high point of the system, to the pro-rearing tank, the middle point of the system, and enter the sump filter. The SAM filter was designed as an upflow and downflow filter and consists of three parts. Sorry. The mechanical filter, the number 6, the biofilter number 7, and the water pump number 8. From the pump, the water goes again to the fish aquarium in the hydroponic unit. Water was added to the filter of the system every day, and the mechanical parameters of the water was checked twice a week. Every 10 days, we measure the morphometric characteristics of the plants and height and new branches. Every 15 days, we weigh the fish and measure its length. Finally, we took tissue samples for our fish and total chlorophyll measure for our plants only at the beginning and at the end of the experiment produced. The result of the present study shows a statistically significant difference in the final weight and the SCR index of the sea bass, where the highest value are found in treatment B. In contrast, the statistically maximum value of the SCR index is found in treatment A. As for prawns, the SGR index shows statistically significant difference, with the maximum value being in treatment C. For glasswork plants, final height and yield showed statistically significant difference, with the highest value in treatment B for both index. Compensatory growth is considered a promising tool for increasing production in aquaculture and aquaponics, with value in farm fish for its faster growth rate and better feed conversion. The fasting period used in this study is relatively conserved compared to previous study involving Oreochromis niloticus and Sparus aurata. The results of this study are consistent with those of Eroldogan et al., in which gillhead sea bream with a mean initial weight of 5.85 grams had a higher mean final weight after 8 days of feeding, followed by 2 days of starvation. The results of the present study do not agree with those of Pastoraki et al., in which Baltic prawn with initial weight of 0.50 grams had a higher mean final weight cultivated in a RAS system. The SGR of prawns observed in treatment B was significantly lower than the SGR of prawns reported by Vlachos et al. For Palemon and Spursur culture at 75 degrees Celsius in a RAS system. To date, the optimal growth diet for Baltic prawns is unknown. Glasswork growth under treatment B showed higher yield and height compared to the other two treatments. The yield achieved in treatment B was significantly higher than the yield reported by Pinheiro et al. For the Conchogeric Sarcocornea ambiqua in co-culture with Pacific white shrimps and 16 ppt salinity. For the conclusion, the polyculture system is sustainable and increases the animal production and plant biomass. The development of compensatory growth benefits the growth of European sea bass and glasswork. Thank you for your attention.
Thank you, Mr. Mitropoulos. Is there any question from the audience? Yanis, I have to make a question then. What do you think? How you can interpret these results? I mean, why the B system? Why that gave you the higher plant growth?
Salicornia europaea is a halophyte plant that absorbs a little less nitrogen, nitrate ions specifically, than the other plants.
So a system that was
she can't afford very
too much.
Thank you.
Too
much of nitrate ions in the water.
So that means that it does not, for its best growth, let's say, it does not need too much feed input that ends up in its root.
Yes.
Right. And also it should be not. It should have a continuous flow of nutrients in the water compared to the sea system that was too much feeding and then too long period for starvation.
Too less nitrate ions in the water.
So that limited the growth of the plant, right?
Yes.
Okay. So, and considering the fish growth, why and fish growth and prawn growth, right? Your best results, if I not wrong, was
in treatment B for prawns and sea bass.
Both.
Yes.
No, it was not in a different.
The FCR index for sea bass is higher in treatment A.
Right. So you had a higher SGR value in the
FCR. Sorry.
FCR. Right. What do you think is how you can interpret this? What this means to us?
Sorry.
Why that happens? That's my question.
We caused the gluttony of sea bass because of the compensatory growth, the feeding and the starvation protocol. We gave food, and after 4 days of the experiment, we starved the sea bass. After that, again, feed the sea bass.
Right, and that resulted in better results than feeding them continuously.
Yes.
Right, and compared to feeding them for 7 days and stop feeding, right?
Again, it's too we are minimized the food.
Because I saw some low FCR values as well.
Yes. In treatment A, the highest.
Because below 1, right?
Yes. The daily feeding.
Right. Okay. Thank you for your answers. Is there any other question? Thank you, Mr. Mitsopoulos.
Thank you very much.
Right, so the final presentation is from Mr. Famulari, right? Entitled on a native population of ruddy tapes, Decunsatus, if I said it rightly, from the lagoon of Capo Peloro, Messina, Italy, a multidisciplinary study.
Good afternoon. Can you hear me? My name is Sergio Famulari. I'm a technologist at the University of Messina in Italy. And today I'm pleased to present some preliminary results on a multidisciplinary study focused on different biological and ecological aspects of a native population of Ruditapes decusatus from the lagoon of Capo Peloro. The grooved carpet shell clam, Ruditapes decusatus, is a bivalve mollusk belonging to the family Veneridae. It is an edible species of high commercial interest, widespread throughout the Mediterranean basin. The farming of these mollusks has deep roots in the local culture of the city of Messina, boosting a century-old tradition in this area. Following the introduction of the Indo-Pacific congenetic species Ruditapes philippinarum into the Italian market and aquaculture, the native populations of Ruditapes decusatus were considerably reduced. This is probably due to the greater resistance of the Alloctonus species, as well as frequent hybridization events. Although we have some discriminating characters that can allow us to detect some differences between the Ruditapes species, the most striking of these interspecific characteristics is concerning the siphons, and in particular the morphology of the siphons. Even if we can detect some differences in shell sculpture, the well-separated siphons of the Ruditapes decusatus can allow us to identify the species. While in Ruditapes philippinarum, these siphons are fused at the base and just separated at the end. In this research, the main goals were to study the population structure of a native population of Ruditapes decusatus present in the lagoon of Capo Peloro through a multidisciplinary approach, including the genetic identification of the species through molecular analysis and an innovative use of ShapeR package to detect some interspecific and intraspecific differences regarding the shell shape of these mollusks. And then a study of gonadal development through an histological assessment. And finally, the age estimation through the analysis of the internal depositional feature of the shell of these animals. Regarding the study area, the lagoon of Capo Peloro is located in the northeastern corner of Sicily, where the Tyrrhenian and Ionian Sea meet. These particular habitats was identified as an oriented natural reserve established by the Sicilian region. Two brackish ponds in communication with the Strait of Messina are part of this lagoon. This communication with the Strait of Messina, globally known for its peculiar habitats, it's a really important point of this transitional environment. In particular, small daily water exchanges, depending on tidal currents, occur every day. And this is due to the presence of a system of canals that allows the constant communication with the surrounding sea. In particular, the Lake Faro covers a 0.26 hectares area, reaching a maximum of 30 meters depth in its central part. And two sampling points were identified in the canal that connects the Lake Faro with the Strait of Messina. And all the samples collected were harvested there. As pointed out before, this is a site of traditional activities related to the shellfish farming in this area. To avoid affecting the delicate balance that governs this ecosystem, it was decided to adopt non-invasive methods, just using traditional tools like gloves, thigh-high boots, or shovels and sieves to fully respect the environment. Periodic samplings were carried out from September 2022 to September 2023, and a total of 300 clams were collected. Specimens were immediately separated from the sediment and were kept alive by immersion in water taken on site and transferred to the laboratory. Each harvested clam was isolated for morphological species identification based on the morphology of the siphons. And several measurements were made to describe the morphometry of the collected bivalves. Regarding the shell shape analysis, this type of analysis was performed using ShapeR, an open access package that ran on the, on the R platform. And this package was specially designed and originally designed to study the otolith variation among Teleos population or species, but it can prove useful to study any two-dimensional object. First of all, k-means cluster analysis was performed to identify three size class, as you can see in the present slide. Regarding the shape analysis, the first step was to divide the valves and classify them into right and left valve. Each valve was then photographed individually, and each photo thus collected were processed using ImageA software to calibrate them and binarize them for the subsequent analysis. The shell outlines were detected and linked to the dataset containing the morphometric measurements of the analyzed specimens. And in addition, Vovlet and Fourier coefficients were calculated and standardized to define the allometric relationships between clam measurements and valve shape. Regarding the histological assessment, soft bodies were extracted and immediately fixed in a 10% solution of formalin and before being banded in paraffin. From the paraffin blocks were obtained serial section, and stained with hematoxylin and eosin in order to be analyzed under a light microscope with built-in digital cameras. All the gonadal development stages reported in a scale of reference were detected during the reporting period, and all the samples were classified according to this scale. Regarding the estimation of the age, the acetate peel technique allows the analysis of internal dark lines called annuli, alternating with light increment deposits that represent a well-preserved record of the growth pattern of these animals. For this reason, a total of 45 valves were first sectioned with a special saw machine with a diamond blade and then embedded in epoxy resin. After an overnight hardening period, each resin block was ground and polished using progressively finer grits and diamond particle solutions. The polished face was fluted with acetone and covered with acetate sheet to obtain a print of the cutting surface. This can better expose these internal depositional features. Regarding molecular analysis, 3 specimens of Rutitapes decusatus and 3 specimens of Rutitapes philippinarum were allocated for this type of analysis, and total DNA was extracted from muscles and the foot. In particular, the amplification reaction was obtained using the 16S mitochondrial DNA primer for the PCR reaction, and the results was analyzed on agarose gel. The sequencing of the DNA was performed by an external company, and the results were analyzed with the nucleotide blast search against the NCBI system. Regarding the results achieved, it is possible to say that several measurements were taken for analyze the population structure as shell age, shell width, soft tissue weight, shell weight, and shell length. These variables were also used to calculate the sharpness index, like convexity index, compactness index, elongation index, as well as 3 different ratio, based on the shell weight and the 3 main measurements of the shell. It's easy to deduce from the result of the cluster analysis, as well as from the other graph here proposed, that the most abundant class during the reporting period has always been the class 2. In addition, the smallest individual with 10 millimeters of shell length and a minimum mean shell length of 25.03 millimeters, both these parameters were recorded during spring. It appears that spring was strongly influenced by the event of recruitment. Regarding the results of the analysis of the shell shape, as you can see in the present slide, we can observe some comparison between left and right valve, in particular for the mean valve shape of each samples belonging to different size classes. As also demonstrated by the results of the ANOVA analysis performed, slightly but significant differences were detected between left and right valve and between samples belonging to different size classes. Then linear discriminant analysis were performed for both right and left valves. As you can see in the graph on the right, in the case of right valves, the specimens belonging to class 1 were moderately separated from the specimens belonging to class 2 and 3. Similarly, in the case of left valve, specimens belonging to class 3 were significantly separated from specimens belonging to the other classes. Regarding the results of the histological assessments, a total of 100 specimens were analyzed, and the 54% of these specimens were identified as male, while 46 were identified as female, based on the presence of spermatozoa or oocytes. Regarding the sex ratio, in our case, we detect a sex ratio of 1.17, and this is not really different from the value reported in literature of 1:1. In addition, as you can see, a similar trend of sex ratio were observed in autumn and summer, while opposite sex ratios were documented during winter and spring. Regarding the gonadal development stages, as you can see in the present graph, you can see that each stage reported in the scale of references were documented during the reporting period. In particular, ripe specimens were sampled mainly in winter and early spring. This is probably due to the lower temperature and the resulting natural metabolic slowdown. Regarding the half-full maturing stages, this was documented mainly in late spring as temperature rose and breeding season began. For the breeding season, Summer was the only season showing a greatest variability of this stage of gonadal development. And this is probably due to the peak of breeding season and to the higher temperatures. Regarding the results of age determination, the range detected was from 1 to 8 years old, and the youngest specimen was of class 1, while the oldest specimens belonged to class 3. It should be noted that despite you can observe the exponential trend line between shell length and estimated age, in some cases, smaller clams were found to be older than the largest one. Regarding the results of DNA sequence amplification, it is possible to state that the results confirmed the identification of the species based on the morphological structure of the siphons, showing even an identity closer to 100, like in the case of Ruditapes philippinarum. Regarding the results achieved, it is possible to state that this study provides a solid database for future research, undoubtedly required to better understand several biological and ecological aspects of this natural population of Ruditapes decusatus present in the Mediterranean Sea. Regarding the literature available, research performed on the Italian coast are still scarce, and even in Europe, the available literature on native European population is limited. In addition, to the best of our knowledge, this is the first multidisciplinary study on a native population of Ruditapes decusatus from the Lake Faro. Moreover, the innovative use of our software for shell shape analysis allow us to detect significant differences in right and left valve, and these slight differences found could be related to a selective pressure from the environment. Regarding the class comparison, results confirmed that the differences between size classes related to the growth patterns is really strictly correlated to these patterns of growth. Regarding the results of the rest of the analysis, as reported by several authors, the reproduction is affected by external factors like temperature and salinity related to the transitional environments. Regarding gamete maturation, as already pointed out by several authors, the peak of breeding season, even in our case, was recorded in summer, while a metabolic slowdown was detected in winter. The event of recruitment occurred during the reporting period in a generalized way, although it showed greater influence in spring. And the sex ratio could be due to higher proportion of indeterminate specimens during cold months. Regarding the age determination, as already pointed out, exponential trend line between shell length and age can be detected, but with some exceptions. In conclusion, the native population of Ruditapes decusatus from Lake Faro is in an overall good physiological condition. The grooved carpet shell clams showed robust reproductive capabilities in the studied area. This bivalve mollusk seems to be well adapted to the environmental conditions prevalent in this transitional environment. The scarce presence of individuals of commercial sites suggests a lack of control over artisanal harvesting practiced by locals. A greater awareness and consciousness on the part of the entire community would be desirable, essential for the proper management of this important resource for the territory. A closer cooperation between local shellfish farmers, the region of Sicily, and the world of scientific research could lead to the development of the expertise needed to preserve and fully reintroduce this species into the national shellfish farming scene. And I would like to express my sincere gratitude to my research group and to all of you for your interest. Thank you very much.
So, any questions? Yes, of course. The microphone, please.
This is the first time that you apply a shape analysis on the shell?
Yeah, to the best of our knowledge, this was the first study applying this type of shell shape analysis, because the package ShapeR was originally designed to study otolith variation in Teleos species, but we found this type of application for the same package, and it appears to be promising. Okay. So we will see. Thank you. Thank you very much.
Can I make a question, please? Very interesting species that I suppose that most of us are not familiar with this species. It was very interesting the study you made. You actually did a lot of work there. I was wondering if there is a law, Italian law, forbidding the fishing of this in specific periods, or are you suggesting?
Yeah, I'm suggesting that a closer cooperation could be a better way to avoid the harvesting, illegal harvesting of this species that is really important in our area. And the presence of the alloctonous species is really dangerous for the Rhodopes degustatus. So we have to fix this situation, and this is what we can do. Just controlling the harvesting by locals, because it's out of control.
And do you have any plans to continue this research? I mean, are you doing this?
These are just preliminary results from my PhD thesis. Right. So I would like to continue this study over the years.
Right, I see. And you mentioned that this species is cultured.
Right? Yeah, I mean, it's not cultured.
It's cultured extensively, you mean, right?
Yeah, the farming of this species in our area is just providing for fattening, but it's related mainly for mussels and Rhodopes philippinarum.
I see.
Rhodopes degustatus is not farmed, but it's still present in natural population in these transitional environments. So that's the reason why it's really important to preserve it in order to fully reintroduce in the panorama of the shellfish farming.
Excellent.
However, does it have the potential to be farmed?
Yes, okay. But it is not easy to find the seed and some specimens. So that's the reason why it's endangered.
Okay, any more questions? No.
Okay.
So yeah, we can close this
session.
Close the
session. Thank you very much for your attendance. Congratulations to the presenters and all the best to your research.
Thank you. Thank you very much.
Finalizing your PhD thesis. And have a nice evening. Let's remind that there is a dinner. The
official dinner is tonight, so you're more than welcome to attend there.
So Rob will catch you there.
Okay.
Bye-bye.
Bye.
Thank you. Thank you. Thank you very much. It was a pleasure. Thank you.
Steve will go there. For us, it's okay. I mean, they can hear us. This, okay? Not. And they hear us when we speak from here in the streaming. My good friend Nikos. They can hear us, but they cannot see us. Okay? Yeah, okay.
No,
but
it's okay. No need for us to be in the screen, just to hear us on
or presenting our keynote speaker, we are here. So since time has passed and there was a delay also in the different sessions, okay? So and we are in the Mediterranean, Steve. That we are in the Mediterranean, so that means it's a lot of increased temperatures and so on. So okay, we call in the floor our guest, invited the speaker for a keynote speech.
We can come here until you're ready. I'll just make an introduction. Professor Steve Fletcher from the University of Portsmouth is a professor of ocean policy and economy. He will present today the unlocking the transition to a sustainable blue economy. He is an ocean policy, blue economy, and plastic pollution specialist, and is focused on delivering actionable research to support positive outcomes for people in the ocean. He has more than 100 peer-reviewed publications and research reports, and it is one of the top 5 most cited researchers in the blue economy field. He has worked during his career at UNEP, to prepare in the G20 meeting within World Bank and with a lot of projects. Now, I think is the director of Center for Ocean, Plastic Ocean. Plastic Ocean. So, Steve, the floor is yours, and thank you for being with us today.
No problem. Thank you for that lovely introduction. Can everybody hear me okay? Yeah, wonderful. Okay, so before I get started properly, I'd just like to thank the organizing committee for inviting me to this event. I'd like to thank Artion for the logistical support they've provided. It's wonderful to be here as well at the University of the Aegean, so thank you so much to our hosts here as well, and it's great to see so many students in the audience as well, in general, I mean. And the other observation I would make before I get started, it's been really fabulous to watch so many presentations presenting such great research. And I've been struck that a lot of the work was done during COVID times, which is, makes it even more impressive, actually, that there's such research productivity during those challenging times as well. It's hugely impressive. And the conference itself, I think, is such an excellent platform for you to share your research, but also to elevate it, to show it to its best effect and to the right audience. So congratulations to everybody for that. Most of all, though, I'd like to thank Michael for the personal invitation, and we share I guess a connection to Portsmouth, where Michael studied for a while. So it's great to be here and great to maintain that connection. Right. Okay. So down to business. What I'm going to be talking about in this session is how we unlock the transition to a sustainable blue economy. And okay, pressing the button, but nothing's happening. Try the mouse. Okay. I've got 3 questions really that we're going to tackle in this presentation. The first one is, how do we respond to the ocean crisis? And I'll talk a little bit about my interpretation of that in a minute. And I'm really going to ask us to reflect a little bit on our role, both as individuals and as a group of scientists. What also is the role of the sustainable blue economy itself in tackling the ocean crisis? And how do we generate the necessary transformative change to move to a more sustainable blue economy? So the underlying question really beneath all of this is the sustainable blue economy a solution to the ocean crisis? And if it is, how do we get there? And that reflects very keenly the theme of the conference overall. So the first thing I want to show you is something that perhaps you've seen before, this idea of the triple planetary crisis. This is the idea that we have climate change, biodiversity loss, and pollution. And these huge global challenges interact with each other. But just think about pollution, for instance. Pollution, you know, exacerbates climate change, and it also generates biodiversity loss. And there are lots of interactions between pollution, biodiversity loss, and climate change. And increasingly, well, traditionally, I should say, these have been discussed as a terrestrial problem, a land-based problem. And really, they could apply and do apply equally to the ocean. And increasingly, we're hearing discussions of the ocean crisis, and the ocean crisis having three elements: biodiversity loss, pollution, and climate change. And I think we can probably all see that. But the question is, how do we respond to that? So we're all scientists of different types in the audience here. How does your research, how does your practice respond directly to the ocean crisis? How does our collective work respond to the ocean crisis? And by collective work, I mean perhaps your department, your research group, or perhaps we mean this group of scientists gathered here for this conference. And how is this conference directly contributing to tackling the global ocean crisis? It's an interesting question, isn't it? Because it challenges us to think about the role of our research in making a difference to this urgent global challenge. And sometimes I hear people say, well, we need more research into this, we need more evidence, but we're in an urgent situation. And I just wonder sometimes whether we do need more evidence, or really we need to take some bold steps to figure out what some of the solutions might be. But we'll come to that later on. So I think maybe some of you have seen diagrams like this before. What we're doing here is combining the idea of planetary boundaries. Those are the things around the outside edge there: climate change, ocean acidification, pollution, and so on, with ideas of donut economics. Okay, so we'll come to that in a second. So what we're saying in this diagram is that for people to have a healthy life, we need a social foundation that uses the world's resources to provide us with the things we need, like energy, housing, social equity, a vote, education. And we use some of the world's resources for that. And there's a minimum level we need to function as a healthy society. That's called the social foundation. But we know that if we use too much of those resources, we overshoot, causing things like ocean acidification, chemical pollution, climate change, and so on. So what we're aiming for is this kind of donut, this circular zone, where we meet the social foundation, but we don't exceed the ecological ceiling. And that's referred to as the safe and just space for humanity. Okay? And that safe and just space is regenerative, and it also is distributively fair. Okay? So resources and benefits are distributed equally. Just by the way, where we fall below the social foundation, that's where we have poverty, hunger, food insecurity, those sorts of things. And where we go past the ecological ceiling, it's where we get things like horrendous plastic pollution, biodiversity loss, and so on. So getting in this space is really important. The question is how? Because what we can say right now, given the ocean crisis and this triple planetary crisis, whatever we're doing right now is not working. And that's the horrible truth of it, which is we can't for a moment think that our global approach to tackling the ocean crisis is working, because things are getting worse. So what do we have to do to get to a more positive space? The green donut, in effect. So when you look at conventions like the agreements like the Intergovernmental Panel on Biodiversity and Ecosystem Services, Paris Climate Agreement, the Treaty on Plastic Pollution that's being negotiated right now, they all talk about the need for transformative change. This idea that we're shifting from the current model of production and consumption and environmental management to a new system that is better, a new system that probably employs a circular economy and a new system that is low in waste and so on. But how? But how is the challenge? And this idea of transformation or transformative change is central to all of those ideas. Well, I mean, perhaps we could just simplify it a little bit. You know, the picture is we're going from a coastal zone that is polluted, degraded, you know, a bit of a mess, to a coastal zone that is biodiverse, that has nature-based solutions to tackle sea level rise, that has clean energy. You get the idea. So what we're trying to move is from the left to the right. So let's have a think about what transformative change actually is. Well, according to the Intergovernmental Panel on Biodiversity and Ecosystem Services, they define transformative change as follows. They call it, and just listen to the words, they're quite, you know, difficult sounding words, fundamental system-wide change. Well, that's a pretty big ask straight away. That includes consideration of technological, economic, and social factors. And then the wording is a bit strange, but then talks about paradigms, goals, and or values. So it's talking about changing the fundamental model that we use to manage the world's resources, that incorporates technological approaches, economic and social approaches, and talks about redefining, you know, the paradigm, the overarching you know, way in which we're tackling these challenges, but also our values, Because in essence, isn't it our values as a society that's driving these challenges at the moment? Potentially, you could argue. So there is an opportunity, according to the transformative change idea, to change the model of production and consumption and change the values that are within it. Now, you may still be thinking, well, what's that got to do with me? I'm a scientist. Okay, well, just hold on. So in the UN Decade of Ocean Science for Sustainable Development, which I'm sure you've all heard of and you're aware of, and you kind of have a sense of what it is, this decade of investment in ocean science to really make a difference at the global scale. That's the point of it. And they talk about transformative science. Okay, interesting. So they describe transformative science as inclusive science that spans marine and social sciences. When you dig down a bit more, it's more than that. It's arts-based sciences as well. It's arts-based approaches. It's economics. It's, you know, all sorts of psychology. It's all sorts of things. So basically, transformative science is inclusive. It includes every discipline that has a relevance to the ocean, which of course is every discipline. Okay? So that's an interesting challenge. It's not just for marine scientists to solve this problem, but for a wider group of people. But the research that we're doing is co-designed and co-produced. Okay, what that means is that the work we do is targeted to a particular audience that needs our research. So that we're not just pushing out research that we find interesting, but we push out research that is useful and meaningful to the global ocean crisis that is urgent and critical for us to address. Okay. Just as a very quick side comment, Michael said that I used to work for UNEP, UN Environment Programme, and I did. And people used to send me papers, just scientists would send me a paper, say, Steve, I've done this piece of research. I think it'd be really interesting for you. At the time I was working on marine protected areas and marine spatial planning and things like that. And the papers were good, but the metrics were slightly wrong sometimes, or the aim was slightly wrong, or the framing of the policy questions weren't quite right. So it was great research, but it was no use to me. And so this co-produced and co-designed idea is so important to make sure that your work lands with the audience that you need it to land with in a way that is usable and has traction. What else? So that the transformative science uses Agenda 2030 as a framework for priorities. Agenda 2030 is the Sustainable Development Goals, basically. So it uses the Sustainable Development Goals for priorities. And I find this one really interesting, emphasizes solutions for governance. Yeah? So interestingly, there's been a real mix at this conference because I've been looking out for that last bit. And you know, I'd say about half of the presentations I've heard have got a section at the end which says, and here's the so what, here's what we do as a result of my science, which is great. Others just end after the series of observations or cause and effects relationships without going that next step. So there is a challenge. And this is not a criticism of this group at all. This happens all over the world. And in fact, you know, there's probably more emphasis on solutions here than you'd normally see at any other conference. So this is really good. But it's interesting that the science we do doesn't just end at the observations and the analysis and the cause and effect. It then goes a step further into, well, what do we do about it? And that's really an interesting shift. Right, so how do we generate transformative change to achieve a sustainable blue economy? So, right, brace yourselves for this, right. So what we have here is a model of how to achieve transformative change. Okay, first of all, I should say there are lots of different models of this. There are models for rapid and deep transformative change that's more radical and fundamental, and there are models for more slow, shallow, bit by bit forms of transformation as well. But this is a model that sort of kind of brings both together, and I will just try to explain this really straightforwardly if I can. Oh no, didn't mean to do that. Okay, I'm not quite sure how to get the little. Anyway, so what we see here, could I actually just look at the regime row at the moment? What we have here in this space here is the current system, the current governance system, let's say for a coastal space. It includes things like science and culture, you know, skills, policy, investments, all that stuff. All the stuff that helps us determine how we use space and how we use resources. Okay? We know that that is not working. We also know that coming from above at the landscape scale, say the national or international scale, there's lots of pressure on that system. So it could be climate change, could be biodiversity loss, could be population growth, could be a economic challenge, or something like that. And we also know at the bottom here, there are people like us who are doing good science and good research, and we're innovating. We're suggesting new ways of doing things. We're saying, well, you know, we found that there's this particular pattern of a fish stock change. If we just manage the fish stock slightly differently, then maybe we'd have a more sustainable fish stock. And these are called niche changes. And so what we see here is now looking at the sort of the vertical axis, we have ideas that are emerging. Then we have the ideas as they diffuse into society a bit more. And then we have the reconfiguration of the system. Now, I'll just talk that through. So we just have a look at the niches idea. So you could say this whole conference is about describing and showing niche changes from your research, okay? Those niche changes over time, they grow, they support each other, they generate a critical mass for change. And what we begin to see is the regime begins to break down. Sorry, didn't you see that? The regime here breaks down. And then there comes like a critical moment where the niches are so strong, the niche change so strong, that they form or reform the system into this new shape here. And part of showing this to you is to give you encouragement that the sort of work that we're doing in this niche change is important because it all fits together to help generate this change in the regime. But also we need to recognize that there's this top layer as well, which is also pushing down on the regime or the governance structure that is also driving change as well. So imagine, just imagine, if we intentionally made decisions that support niche innovations and that intentionally cause tensions in the regime and that intentionally enable these landscape pressures to push against the regime. So the point I'm making here is this is not an accident. We can control this and use it to generate system change and transformative change in a way that we want to see. Now, what we've got here is what's called a decision space. And the big blue square represents sort of a mix of decisions that a country could take. And all of these, the blue nodes here, are decisions, okay? And we've got country A and country B and their starting points, okay? Country A and country B are different. Maybe one's an island, maybe one's a country on a continent, maybe one has a degraded coral reef, maybe one is investing in wind energy. You know, who knows? They're different, that's the key point. And over here, what we're trying to get to is a vision for their sustainable blue economy. Okay? And we know that because they're different, they will have different decision points. So as they just work through these decisions here, and this could be, you know, what marine spatial planning approach they take, it could be what types of habitats they want to protect, it could be how they support rural coastal communities to you know, be affluent and to have, you know, good health outcomes and all this sort of stuff. The point being, just looking at country A, it makes a series of decisions that create this sort of haphazard looking journey through the decision space. But at the end, it comes out with its sustainable blue economy vision. Country B has exactly the same process, but the decision points are different because they start in different places. And so again, that comes out with its blue economy vision. And I should have said that we've got time across the bottom here. So these decisions are taken over time. So now let's just go back to this one for a second. If you notice again at the bottom, we've got this idea of the emergence of ideas, where the niche ideas just come out, then they diffuse into the wider society and the wider regime, and then the system or the regime reconfigures itself into something better, hopefully. So what we can then do is start to overlay that idea on the decision space. So this is the emergent space, this is the diffusion space, and this is the reconfiguration space. So what we have here is, you know, we can intentionally support country A by investing in innovation. And then we can support country A or B, in fact, by then working to help diffuse those ideas into the wider regime. And then we can support the reconfiguration of that regime as well. So what we're doing is recognizing that countries have different starting points and they have different endpoints, but the process they go through can be similar across those three transition or transformation stages. It's slightly complicated stuff, but hopefully it kind of makes sense. But the point is, we can support countries to make those decisions. These are not random events. These are active choices that we make to support transformation to a sustainable blue economy. Right, which raises the question, what is a sustainable blue economy? Now, I've heard it mentioned quite a few times in this conference, and the shorthand is, it's the balance between economic development of the ocean and its conservation. Maybe. What I'd like to see is a more nuanced definition, I think. And I just want to show you a definition that myself and some colleagues have been working on with the World Bank, actually. This is not published, this is just sharing some ideas with you at this point. So a sustainable blue economy is a regenerative approach that restores healthy and resilient blue ecosystems, generates equitable and inclusive benefits for people, and contributes to the integrated delivery of tangible outcomes to meet the challenges of climate change, marine biodiversity loss, and pollution. So the simplistic view of the blue economy as economic versus conservation interests, forget that. Blue economy is whatever we want it to be. It's whatever principles we insert into it to make it deliver what we want it to deliver. Okay? And so this definition sees the blue economy not as a trade-off between economic and conservation interests, but as a vehicle to regenerate and restore coastal and marine ecosystems, that builds resilience, that has benefits that are equitable and inclusive to coastal communities, and that supports confronting the ocean crisis. So the understanding of the sustainable blue economy, I would say, has evolved quite a lot in the last few years to really see it as a vehicle to deliver change. Not everywhere, not always. You can be skeptical about that, but it could be seen that way. So the critical point amongst all this, though, is how do we make the sustainable blue economy a principled thing? Okay? And I'm just sharing with you now some work that me and my colleagues have been doing with UNEP. Again, it's not published, but it does sort of illustrate some of the principles that we've been discussing. So, for example, there are 5 principles down the right-hand side there. I'm sorry, that might be slightly hard to see, I'm realizing, but the first principle is that the sustainable blue economy protects, restores, and regenerates healthy ecosystems. The second is that it delivers equitable and inclusive processes and outcomes, enables climate stability and resilience, delivers sustainable consumption and production, and it applies circular economy approaches. Okay? So let's just pause for a second. So any approach to managing marine resources is basically an empty box. It's just a process. Like marine spatial planning is just a process. Integrated coastal zone management is just a process. But it depends how you do it, right? It depends on the principles you apply to that process and the decision-making criteria you use whilst you're delivering that process. So a sustainable blue economy could be just a vehicle to make money. It could be, if that's the approach you want to take. It could be just a vehicle to conserve, if you wanted it to be. But what I'm presenting here are 5 principles that together give a different perspective of what a sustainable blue economy could be. So it's a circular ocean economy that supports climate change action, that is inclusive and generates benefits for people and protects and restores healthy ecosystems. And that feels like a good thing, a good set of things to be asking for. Right, so what we can then do, and I shan't spend too much time on this, is look at each of those principles and think, well, what does it deliver? And it's all the stuff you'd expect it to deliver. So restoring and protecting, regenerating healthy ecosystems, supports an ecosystem-based approach to ocean and coastal policy, delivers equitable and inclusive processes and outcomes, delivers increased representation of women, youth, and other marginalized groups. So what we start to get is a series of outcomes through the application of these principles. Okay? And I shan't do any more, we're a little bit short on time. What we can then do is look at and see how those principles relate to the Sustainable Development Goals. And this is a piece of analytical work that we've done, and each of those principles generates quite a contribution to multiple Sustainable Development Goals. Interestingly, in my time working with UNEP, I used to work to support countries to deliver SDG 14, but where a coastal or marine policy supported the delivery of other SDGs as well, that was the holy grail. That's what the countries really wanted. They wanted efficiency of delivery of the SDGs. So if there was one policy they could put in place that delivers 10 SDGs, or delivers contributions to 10 SDGs, that's brilliant. Okay? So just by applying these principles to a blue economy approach, you start to meet multiple sustainable development goals. So what we've been working on with UNEP is a sustainable blue economy framework. And it's this idea that there's a process that UNEP can help support, can help countries guide through a process to deliver a sustainable blue economy at the end of that process. And it's based on the principles, but it's also designed to be practical, achievable, and respect and reflect a country's own needs. And this is it, basically, boiled down into a diagram. And the basics of it are that there are 3 phases. There's a phase 1, which is understanding the system, which is understanding how a country currently manages its marine resources, what its institutions are, what its legal framework is, all that stuff. Then there's a part of the process, phase 2, which is about strategic policy development. Okay, so that's about identifying a vision for the sustainable blue economy for the country, for a given country, and by having a vision, the vision is kind of the end point, the understanding the system is the start, so you can begin to figure out how you transition and what institutions you need, what policies you need, what investment that's needed, what alternative approaches to community engagement might be needed, all that stuff. And that's in the strategic policy development phase. And phase 3, delivering change, is basically the implementation of that system through an integrated marine governance framework. So that is not treating tourism as tourism, fisheries as fisheries, but trying to bring them all together into a single integrated approach. Now, if you just sort of ignore these principles for a second, you could get anything out of that process. I mean, the process isn't particularly remarkable. The process isn't particularly mind-blowing, but it's the principles that sort of supercharge that decision, or that process, sorry. So when we understand the system, we're trying to figure out how those principles are already embedded in the system. When we're doing strategic policy development, any policy we produce has to adhere to those principles. Okay? And the delivering change then becomes the embodiment of those principles. That's why the principles are so important. Otherwise, we could get any random outcome based on the politics of the day. I'll talk about the cross-cutting actions. These are very low down at the bottom here, because these are quite important as well. I'm just going to skip through these. So basically, this is a breakdown of the activities in phase 1, and there's lots of baseline assessments, basically, good research-based activities, working with countries, working with stakeholders, and so on. Phase 2 is about developing a vision. It's about developing policy. It's about expenditure planning, all that. So again, working with a country to really understand what its needs are and how and what needs to change to help it deliver its vision. Okay, that's pretty straightforward. And then phase 3 is about the integrated marine management, capacity building, monitoring and evaluation, all that sort of stuff. Who needs to be involved? Well, remember the transformative science, inclusive approaches. So we need all sorts of partners. I mean, academic institutions, researchers, us are one of them for sure, but there are lots of others as well that need to be in the mix, because whatever blue economy strategy we come up with needs to work for all of those stakeholders, or increasingly, as they're called, rights holders, people with rights over the marine environment in various ways. Right, we've come very close to the end, Michael, you'll be glad to know. So I just want to just say a couple of final things. The shift to the sustainable blue economy is not just this process. We have to think, well, what enables that process? What sort of oils the wheels of that process? Okay, otherwise, it's just a dream, okay? It's just a fiction. So what we need, and this is all debatable, but all good stuff, we need strong leadership, we need clear and appropriate stakeholder involvement in various ways. There absolutely has to be sustainable finance. We can come up with any plan we want. It's not finance, it's not going to happen. So, much as, often, in an audience, there's a lot of pushback on the finance. Like, oh, I don't really understand that. But actually, it's critical. It's so critical to this whole thing. We need coalitions for change. So, people working together, building a consensus about what needs to shift, building a consensus about what the vision is like. And sound science. I would actually not say sound science, I'd say sound evidence, because we're not just talking about science, are we? We're talking about the arts, social science, economics, and every other discipline you can think of, some of whom may not wish to be considered as science, but sound evidence and evidence providers, I guess, is what we're talking about there. And of course, good governance around transparency and accountability, and so on. So we've got the process, we've got the principles that drive the process, and we've got these enabling conditions. And of course, the way we deliver those is through working in country with governments to support their engagement with these ideas and making those ideas their own within the context of a national setting. I'm going to draw things to a close with just some concluding reflections. I think the first one is that the ocean crisis does require urgent action. I mean,
if nothing else, the presentations in the last 2 days show us that. We've heard about, fish stock collapse, habitat destruction, species disappearing. It's quite depressing, actually. If you sort of bring it all together into a single storyline, it's basically in the Mediterranean, things are pretty bad. I mean, that's the horrible summary of it. So there is an urgency to this situation we need to tackle. We know that transformative change is possible, but it requires intentional choices. So it's not, I'll come to this point in a second. It requires intentional choices at all levels to move towards that new system. A model exists to unlock the shift or the change to a sustainable blue economy. We just talked about that. But the final point I'd just like to talk about a little bit more is that this, as in the transformation to a sustainable blue economy, really does need to be supported by this idea of transformative science. And that's where you and me and most of our colleagues come in. It's about doing research that really supports this idea of transformation, particularly through solutions-focused action. That's it for me. If anybody would like to follow up with that, obviously grab me now or grab me at dinner. But if not, this is my email address. Feel free to get in touch. Thank you.
Dear Professor Fletcher, thank you very much for this inspiring lecture. We can understand that the blue economy concept is really new. We discuss about blue economy, we are discussing about sustainability or sustainable blue economy, but a lot of people is not sure on what exactly is about this. And especially if we go from the science that are working on this issue to the government, to the responsible policies, then it's even more difficult to work with them. This kind of procedure gives us a roadmap on how we can work as a scientist to show the way to the politicians and to the managers, I think. So this is my first impression of this speech. I don't say that it's an easy path, but it's a path. And can we address to the public?
Let Steve answer in that, because my question is also this one. It's we know the pathway, we know the model, we know the basic terms we have to follow. The scientists suggest, but as you very well know, and I think this also happens in the UK, as it happens everywhere in the world. The question is, who is going to take the decision to apply this, let's say, initiatives? Because everybody speaks for that. There's a need to crisis. We speak the scientists. We speak about overfishing, about pollution. You saw it in the first slides of your speech, okay? But we are not taking the decision. So that's a critical point. That if we do have till now at least some, let's say, successful implementations, are there any examples of we have managed to go further in a positive way on this issue?
Okay, gosh. So how do we move, how do we persuade countries to move to go through this process? Okay, so I know that the World Bank is generating a new program with investment to support poorer countries to go through a process to develop their blue economy approach using the sort of principles that I just talked about. I know that UNEP, UN Environment Programme, are doing the same. They're putting a program together to support, again, poorer countries that are biodiverse to support their approach as well. There are big international funds like the Global Environment Facility and the World Bank in a separate part that are working with coastal communities around the world to try and determine their priorities to put in place sustainable blue economy approaches. In fact, there's a whole division in the World Bank focused on blue economy. So, you know, there are big, powerful players who are providing financial support and a process for countries to go through. I mean, then you've got places like, you know, we've got the EU, for example, which is really pushing not so much blue growth anymore, but a blue economy type approach. And again, I'm seeing, if you look at the Green Deal, for instance, if you apply the Green Deal concept to the blue economy, you're getting something like I was just describing. So I take your point. It's difficult to see how countries might voluntarily take up some of the ideas that I just spoke about. But it's usually in the self-interest of most countries to go down this route. And if they have some financial support to help them, then that is likely to just push them into action, I think.
However, in this discussion we're having, we can see the example of even a level of the European community. We've seen recently, this year actually, okay, there were, you know, big protests all over. We have also European elections for the European Parliament in a few days, okay. However, what we see is that, okay, we speak about Green Deal, green blue economy, blue growth, blue economy, but I think that's my feeling, at least, that we're ready to go some steps behind, because and also we shouldn't forget that the different political parties are taking, you know, to express different issues, sometimes not going ahead but going back. That's my feeling, at least.
Yeah, oh gosh, okay. Well, you know, I don't have all the answers to this, you know, but politics is critical to all of this, right? And so, you know, if maybe there's a stronger role for scientists, you know. So, for example, in the plastics treaty, okay, right now a new treaty is being negotiated to end plastic pollution, and in that treaty process, there's something called the Scientists Coalition for an Effective Plastics Treaty, and that's a group of about 500 scientists globally who are feeding in real-time evidence into the treaty meeting. They're fact-checking statements that countries are making within the negotiations and responding to them in real time within that negotiation block. They're putting out policy briefs, they're putting out position papers. It's a quasi-campaigning science organization. I mean, they would say they're not campaigning. They would say they're just providing independent evidence. It's a sort of difficult point. But we don't have one of those for the ocean, do we? Don't think so. So, you know, why not? Maybe this conference could be beginning of the Mediterranean Scientist Coalition for a healthy Mediterranean, where, you know, you lobby and you push hard at any negotiation that's happening around the state of the environment or blue economy in the Mediterranean. It's an idea, it's an option, it's something you could in theory do. And there's a model already in the plastic space that appears to be really pushing countries to rethink their relationship with plastics. So it's...
I know that time has passed, but we have still some minutes, okay, for some questions extra from you. Adonis, and but ladies, we give the floor to Ioanna, and then Iphigenia, and then Adonis.
Yes. Okay. Concerning blue economy, I think that this is a political decision that has to be based in stakeholder engagement and involvement, scientific analysis, participatory processes, collaboration with investors, and 3 stages have to follow: planning, implementation, and monitoring. Thank you.
Okay, that was a question. Okay, so you're probably right. I mean, the 3 stages, planning, implementation, and monitoring, are not that different to what I showed. I think there's probably a stage before planning, which is understanding the current system, although that could be planning as well. It depends what you call planning, I suppose. But yeah, you're right. Those 3 stages would work.
I mean, everything's a political decision, I would say. So certainly the blue economy is. And the blue economy is not something that, as scientists, we could just decide to do. It of course, requires political support. But the question is, how do you generate that political support? How do you generate that need to change? And that is something we can influence through our work and how we share our work at events like this. You know, just as a small side comment, in tackling the ocean crisis is urgent. It's urgent, you know, tremendously now to make a change. So what do we keep doing? Do we just keep on pumping out the same research and hoping something changes? Or do we step in, do we lean in to try and make a difference in the political space, to try and push governments and politicians to change? You know, for decades, we've been very polite. And where's it got us? So there's I'm sounding like a kind of rebel suddenly. Okay, but you know, I do think there's a moment when, you know, enough's enough, and we have to lean in and say, you know, the science is telling us, and the evidence is telling us very clearly, we need a new form of marine management and marine governance that regenerates marine ecosystems and supports communities. You know, I think probably now is the time to do that.
Thank you for your presentation. Coming back to the comment made by Professor Koutroubas, I would like to ask you, do you really believe that there is a close link between the scientific community and the decision makers? Because to my perspective, to my opinion, at least at the Mediterranean, there is a huge gap between the scientific community in terms of their scientific reports, their everything, and the way and the reason that the decisions are made. And I think that it's no matter of funding, or for if the European Investment Bank has the money. It's a matter of the structure of our administration, of our community, of how we understand the concept of the ecosystem services. This is my opinion. Thank you.
I understand. Can I ask you a question back before I answer? You might need the microphone. Which is why is there a gap in your opinion? Why is there a gap between scientists and politicians?
Because to my opinion, there is another agenda. Okay. There is a second agenda, a back agenda. So, for example, you presented us the SDG goals. Okay, Greece is lower in terms of achieving the 14th, the 15th SDG it's classified among the 5 countries with the lower scores.
Yeah.
Okay, so it's something something doesn't make sense.
Yeah. Okay. What is the alternative though of trying? What is the alternative to trying to close the gap? We just let things continue and get worse. I mean, I'm not saying you're wrong, but I'm saying, what's the alternative? I mean, what is our responsibility as people who know this stuff in this space? Is it just that, oh, there's a gap, never going to make a difference? Or do we lean into that gap and find new ways of communicating, find new processes? I don't know what it would be in the Mediterranean, but I don't think we can just say, I'm not criticizing what you're saying, but I don't think we can just say there's a gap and so we can't do anything. I think we have to find alternative ways. But I'm sure you're right though in this context, but I don't know.
I can see, and to proceed to the next question, that we have a problem that the old situation, the old regime, the old status is fighting trying to avoid any changes, and we have to fight back. And one way is building a network of scientists working together and promote the best solution. It's not always working, but according to your presentation also, after a few years, a few efforts, a few back and forth, we'll manage to have this critical mass that will have this transforming, generating transformation procedure happen. So, Professor Velegrakis.
Yes, hello, good afternoon. Very nice talk of yours. And of course, we have to be optimistic because basically if we start considering what the situation is getting worse and worse. I just want to say two things. From your representation, I focus on what you said, urgent. And urgent, of course, is how much time you need for the change compared to how much time we have. And there the situation has become very, very dear, very difficult, because of course we don't have time to do that. So basically we have to be fast. And of course there is a roadmap there. The problem is that in many cases, we do not understand what we have to fight against. For example, you said about the plastic thing. Well, in my opinion, and probably I don't know if I'm right or wrong, but in my opinion, the plastic pollution is a supply side generated problem. So basically, nobody woke up one day and said we need to have all our meat in plastic. I remember sometimes, because I'm older than I look, and I remember sometimes that we don't use so much plastic. And who started that? Well, I didn't demand that. Nobody demanded that. Somebody had an idea how to get some more profits from petrochemical companies. So how do we make sure that the petrochemical companies stop having this, and how because they have basically diluted all their ideas and whatever that it is something that we need to do and cannot do without it, how do we change that? I don't think we can change that with NGOs, and I don't think we change that with everything else. We need to change, certainly, the way the government are thinking. And this is a very, very crazy thing to do. And because, for example, you are from Britain, and for example, we know what the situation with 400,000 releases of sewage, untreated sewage in the seas it was on. I was living in Britain for many years. And I thought that the British people will never go for it, will never support that. And I cannot see anybody talking about because they say it's an economic problem. And we should continue doing that the way we're doing. We do the same here. So basically, I thank you very much for this optimistic point of view. There is a problem with the finance too, very much a big one. For example, just to say that when in 2009 in Copenhagen, they pledged 100 billion each year for adaptation of climate change. How many of those billions have ever been given to the developing nations? 75% of all the money anyway, it's in the form of loans, not in terms of grants. So it's a very difficult. I don't want, there is nothing for you to understand. We see the same thing as we see. And thank you very much for an effort and your efforts to try to make sense out of this crazy world. But I don't know what to say more about that. Thank you.
I just make a couple of quick comments. Your first question about the timeline, I don't know either. It's very difficult to figure out, how much time transformative change takes versus how much time we have. I don't know either, but so I agree. Just on the plastics thing, you said plastic pollution is supply side. Just to explain what that means, it means the production of plastic in the first place is the problem, not how we deal with plastic waste. Dealing with plastic waste is dealing with the problem once it exists already. It's kind of too late by that point. What we really need to do is deal with the production in the first place. That's supply side, right? And interestingly, in the plastic negotiations, the Scientists Coalition that I mentioned before have just published a paper proving the link between plastic production and plastic pollution. For the first time, this has been properly proven, although it feels obvious, right? And so the evidence is now on the table for negotiators that if they want to reduce plastic pollution most efficiently and most effectively, just cut production. And probably about 100 countries are now behind that position because the scientists put that option on the table. So it may not pass. I mean, it may be negotiated out. But again, it's this idea that, what is the role of people like us? It's to bring the evidence, put it on the table at the key moment, and make the case. Yeah.
Go to the last question, Professor Verilis.
Dr. Fletcher, thank you very much for your interesting presentation. I really think that you are optimist, but I am pessimist. In the middle of your presentation, you saw a slide with a very big diagram showing how to reach blue economy and a very little box down saying that we need good governance.
Yeah.
This is the bad thing. The good governance is the big problem.
Yep.
This is the big problem. For example, thinking about greenhouse gas. The limit, I think that for EU to reach, the limit is 240, if I'm right. I think that's too late. And the steps are too little about that. Yeah. In my opinion, the only way to reach blue economy is to have an education for that. Our generation not have educations. I think that our children must have a better education and better information about this blue economy. But I'm really pessimist that it will be too late.
Okay, I'll come back to the optimist point in a second. So the point about good governance, I mean, just to be clear, I mean, the whole transition framework is about, sorry, the transformation framework is about governance. It's about shifting the governance system. But you can have bad governance processes and good and bad quality governance. We have good quality governance. And good quality governance is transparent, it's accountable, it's evidence-based and all those sorts of things. So again, it's a little bit like saying, well, what sort of governance? Because the whole thing is shifting the governance arrangements. The other point you make about me being an optimist, I mean, I am an optimist in life, but I don't think I'm an optimist on this, actually. It feels to me that I'm advocating that we need to lean in to make a difference. And if we do that, we can begin to make our science and make our evidence more impactful, more meaningful, have more leverage on the outcomes that we want to see in the world. And that feels realistic, because, I mean, why are we doing our science? Why? We're not just doing it to idly find things out and say, oh, that's interesting, and never think about it again, are we? I mean, I'm not. I'm doing it to try and drive change, to make a difference, to stop the planet getting wrecked. And so I don't think that's optimism. That's sort of empowering our own science to try and make a difference, I think, and finding research, areas of research that help us understand how to generate change and how to move that change. I mean, loads of people in the world are researching transformative change. This is not something I've invented. There's a whole discipline of transformative change. And these are academics and researchers who are working with practitioners to try and unlock processes of change to generate better outcomes for the world. And we sort of somehow need to kind of connect our marine science with that idea of transformation so that we can sort of, I can't quite find the right word. We can sort of empower or electrify our research to have more impact. I sounded very optimistic then. I do agree with you, but I'm trying not to be. I'm trying not to be. Yeah, I see your point there.
Thank you very much. Thank you for joining us. Thank you for the.
Oh, okay. Thank you.
This is the book.
Oh, wow. Okay.
Theophilus is a local painter from his island. Not a big painter. It's an older one. He has a lot of traditions.
So nice.
This is a big book of his paint.
Yeah.
This is just
the t-shirt. Yeah, I saw some people.
We hope that it is okay. This is extra large. This is extra large. I think we cannot have a large. For you.
Wonderful. Look at that. It's very kind.